rahmatullahi wa barakatuh. Esteemed guests, good morning. And of course, everyone who's watching us from around the world. Hello and welcome to the second day of the Riyadh Global Medical Biotechnology Summit 2023. We have a very packed program for today, engaging agenda and sought after local, regional, and international speakers, industry leaders who will be with us throughout the day from the strategic field of biotechnology. We are here in Riyadh and we have audience members from around the world, over 50 countries. This is truly a global event. To start, it's my pleasure to welcome Engineer Sulaiman Al-Mazrou, CEO of the National Industrial Development and Logistics Program, Saudi Arabia, sharing with us how the Vision 2030 is transforming the kingdom into a leading industrial powerhouse. Welcome to the stage, Engineer Sulaiman. It's always great seeing you. The stage is yours.
Your Highnesses, Excellencies, and honorable guests, Assalamu alaikum. It's my great pleasure to be here at this important gathering to say a few words on how Vision 2030 underpin our thriving economy. But first, let me welcome all of our guests to Riyadh at such a key moment of its history. And for the audience who join us virtually, we invite you to join us next year and enjoy the beautiful Riyadh. For right now, the kingdom is experiencing a wave of economy, an explosion of investments, an expansion that it has not seen for several generations, if ever. And possibly uniquely, it's led by both the private and public sectors. And it's been felt across all the parts of our country. For many around the world, watching Saudi beat the champion Argentina in the World Cup last month may have been the first time they experienced the new Saudi. But the truth is that we have been the world beaters in growth, reforms, and investment for several years now. All success stories start with a vision, and successful visions are based on strong pillars. These wise words by His Royal Highness Prince Mohammed bin Salman, the Crown Prince of Saudi Arabia, explain what Saudi Vision 2030 is all about. Vision 2030 is ambitious yet achievable blueprint, which was built on strong pillars. It starts with our status at the heart of the Arab and Islamic worlds and our determination to become a global industrial powerhouse. It sees us leveraging our strategic geographic location and transforming ourselves into a global logistic hub, connecting 3 continents, Asia, Europe, and Africa. The National Industrial Development and Logistic Program, NIDLIP, is a great example of the impact created by this great vision. NIDLIP integrates the key sectors of energy, mining, industry, and logistics via more than 300 game-changing initiatives to achieve clear and well-defined goals and targets. At NIDLIP, we're proud to have an investor-centric approach that cater for both local and international investors. We do this through the fundamentals of risk and return, minimizing the risk and maximizing the return to investors, all in a sustainable way. To that end, through Vision 2030, the Kingdom offers a well-developed infrastructure, including digital infrastructure, plug-and-play value parks, establishing regulations and provide sustainable and business-friendly environment, plentiful resources, raw material, skilled and qualified human capital, and world-class research and development centers. And finally, access to local and international markets through a state-of-art multi-mode logistic hubs, optimized routes, and local offtakes and international agreements. I hear you say, what will that look like? Think about the possibilities. The power that will come from integrating 4 core sectors: energy, mining, industry, and logistics, combined with the power of the 4th industrial revolution, or a smart mine supplying an automated factory that is connected in a state-of-art industrial city, run on energy from efficient grid, and moving goods and people via best-in-class multi-mode logistic services, maximizing clean energy generation and utilization, achieving the shortest and most direct routes. These, ladies and gentlemen, are just examples of what the transition is looking like. A transition to safer, cleaner, and more productive, fully integrated network of sectors. And as I say that, this is already happening. Furthermore, the impact of Vision 2030 are not limited to the Kingdom. By becoming an industrial powerhouse and a global logistic hub, we will open opportunities for new products and markets, improve connectivity between continents, and enhance international trades, benefiting the entire global economy. So this is not a domestic initiative. We invite the international community to be part of our journey toward creating a prosperous future for the human around the world. Allow me now to shed some light on our national industry strategy. The launch of the National Industrial Strategy by His Royal Highness the Crown Prince is yet another milestone toward transforming the Kingdom into a leading industrial powerhouse. The strategy has 3 main strategic objectives. These objectives are intended to position the Kingdom among the top 15 industrial nations in the world. These include building an industrial national resilience, becoming an integrated regional manufacturing hub, and expanding global leadership in specific segments. The strategy aspires to empower the private sector, increasing the flexibility and competitiveness of the industrial sector, and ensure continuity of access to important commodities for the well-being of citizens and the continuity of economic activities. The Global Biotech Summit is another showcase of Vision 2030 to contribute to the world development by addressing essential themes such as gene therapy, vaccines, and genomics for rare diseases. I look forward to participating in fruitful discussions with such a fantastic mix of experts, data scientists, researchers, technologists, government officials, and entrepreneurs working together to benefit the humanity. Thank you.
Thank you, Engineer Sulaiman, for setting our event off to a great start, sharing with us the ambitious vision of transforming the kingdom into a leading industrial powerhouse. Very promising. And now it's my pleasure to welcome Dr. Stephen Lockhart, Vice President and Head Europe and Asia Pacific, Vaccine Clinical R&D, Pfizer UK. Welcome, Dr. Stephen. Dr. Stephen is about to come onto the stage. Dr. Stephen Lockhart, our second keynote speaker for the day, and he is co-chairing the next panel discussion as well with, we have here Majid Al-Ghuraibi. But first, we will hear from him. Welcome to the stage, Dr. Stephen. It's great having you with us. I'll pass the mic to you. Thank you.
Thank you very much. Thank you very much. What I'd like to do today, thank you for inviting me and thank you for coming to listen. I want to talk a little bit about our experience with the Pfizer-BioNTech COVID vaccine and the rapid development. And I want to tell the story, not really for its own sake. You can read the papers and the history, it's all very public. But I want to talk to why the experience gives some guidance about how you need an ecosystem to do something like this, and what some of the key elements are. Of course, this is the disclaimer. I work for Pfizer, so you have to understand my position, and I've been intimately involved with working on this vaccine. Now, this is the conventional timeline. The timelines show us starting work at the beginning of 2020. The genome came out in January, and everybody was starting to look at what you could do in terms of vaccines, and BioNTech started making some constructs. And by December 2020, we'd been through the clinical development enough to have emergency use approvals, in very first one in the UK, then rapidly in the US, Europe, and of course quite early in Saudi Arabia as well. That's a lovely story, except of course it's complete rubbish because we didn't really start in early 2020 at all. What we built on was the fact that BioNTech had been working on mRNA vaccines for over a decade, built up a lot of experience in how to make them and how to use them and what the immunology was. And Pfizer has been working for a very long time to create what I call a machine to develop products. So we can plug new products into that existing machinery, which has a global outreach in terms of where we can do things. And Pfizer also has decades of experience with manufacturing vaccines all the complexities. Now, this was a different type of manufacturing, but still all the infrastructure underneath it, and the distribution and the regulatory side and so on was very highly developed and ready to work as fast as possible. So in a way, this story I'm telling you here is a bit of an exaggeration. Nonetheless, in that 1 year period, there was a huge amount of work to do, and that work has continued. And I talk about the evolution of the vaccine because, as you know, we're still working very hard to change it and bring it up to date and get it to the world. So this timeline is nice, but it's only part of the story. Now, not really only for us, but for other people, there were really three fronts in the development of treatments and vaccines for COVID. One was the science. We were very, very fortunate in the timing of COVID, of the COVID outbreak, because not only had BioNTech been working on the mRNA vaccines for a long time, we had actually also started working with them. Okay, so we'd started work in the previous year on a flu vaccine based on mRNA. And so we already had a relationship. And the one thing that you can't do overnight, even in an emergency, is build a relationship. You have to have that experience of talking it through, working out how you're going to work. We're very different companies. We're a big machine. They were a very small biotech startup. The one thing we shared, of course, was huge enthusiasm to take this forward for infectious diseases. But that relationship was absolutely critical. And the technology was critical. So I don't think there was a single company that brought forward a new technology during the outbreak that actually brought it to the vaccine market. You actually need that sort of background to do it. The second area that was important was people and practices. So we already had a very, very sound quality system for doing work on vaccines. We had all the arrangements, all the processes and systems to do it safely and effectively. But we had to do it much, much, much faster. And we were only able to do it faster because we had that base to build on of our experience. And then the collaboration and regulatory framework. And I can't really overemphasize the fact that it wasn't just Pfizer and BioNTech. We worked with a lot of other organizations. We worked with numerous investigator sites around the world. We worked with CROs, and then in terms of manufacturing and distribution, huge number of other sites, especially BioNTech worked with, to get these things done. And the regulatory relationship was absolutely critical. Not surprisingly, probably our biggest relationship was with the FDA, and I really admire what the FDA did during this period because they rapidly put out guidance. Of course, they didn't know any more than we did at the time, so we were all learning together because this was a new virus. So they very rapidly put out guidance to help make sure that people would work in a way that was aligned, but also we had discussions. We had a lot of discussions, and there were various groups. I worked on a COVAX clinical working group that talked about how you can bring these things, how we could manage the trials globally, both small companies, big companies, low-income countries, high-income countries, and that was a very effective forum. Now, what do we do? Now I'm going to tell you about how to make a vaccine, in a sense, from a clinical development perspective. Of course, you have to do phase 1. No matter what the crisis, you have to take the first step of putting a vaccine into human beings. And we actually put 4 different constructs into humans. Between BioNTech and Pfizer, we put 4 different constructs because we didn't know which one was going to be appropriate. We put in a basic uridine RNA, which was unmodified. We put in the modified RNA, where some of the uridine constructs are replaced by the nucleotides you put into the medium. And we also put in a self-amplifying RNA, which everybody thought would be wonderful, but really hasn't proved itself yet by anyone really during the pandemic. And so we selected the mod RNA construct, and we used the the so-called P2 mutated full spike protein. Now that didn't come out of nowhere either. The NIH had been doing work based on SARS, the original SARS, for a very, very long time, and had come up with this as the best stable construct, whether it was a protein or an mRNA or whatever, as the way of getting a stable presentation to the immune system that would induce the best response. So there's another piece of work. Background academic work was absolutely critical to moving us forward, and I think most of the companies use that particular construct. Now, phase 1, we had a great result. We were very, very pleased. There was some reactogenicity, but manageable. And also we found, you can see here, don't look at the figures, but basically it showed that we didn't get much response after a single dose, but after a second dose, 21 days later, we actually saw very, excuse me, we saw very good, what we thought were good antibodies. We didn't know, like any phase 1, excuse me, you don't know what antibody response you actually want. And so we actually used the human convalescent sera. So we said, if it gives you a response that's at least as good as you see in people who've had the infection, that's a pretty good starting place. And all the other companies use that too. So that was very encouraging. We also had to show that we got a T-cell response. And this is important because the big risk, you know, before you go into humans, of course you do a big risk evaluation. What are the risks? What could go wrong? And one of the concerns with new virus vaccines is that you might actually make the viral infection worse at a later point when someone gets a natural infection. And that's a bit of a theoretical risk, but at the same time it's been seen before. RSV virus was discovered the year I was born. Everybody thought, great, there'll be a vaccine within a couple of years. They did trials with an inactivated vaccine, and a couple of children actually died from having worse disease when they then got naturally infected. So it's a real risk. One of the things that the regulators asked for, and we thought was appropriate too, was to show that you get a T-cell response. So that's a background response. And that's partly because vaccine-enhanced disease, when it occurs, is usually related to weakly binding antibodies. So first of all, we found that we had very potent neutralizing antibodies, which was encouraging, but also that we had this background T-cell response for a particular type. We didn't actually have this, of course, until after the phase 1. So we did the phase 1 in a group of very healthy people in case this was a factor. But as soon as we saw this in those people, and we saw the good neutralizing antibody response, we were very encouraged to move forward into an efficacy study. So we started a phase 2/3 efficacy study in July 2020, which that was really only a few months after we'd started the phase 1. I mean, we wouldn't normally move quite that fast, but obviously this was a special circumstance, and the phase 1 data was really closely examined and was very encouraging. So we started this study. We didn't. Usually when you start a phase 3 study, you know a lot about the disease. You know how common it is, you know where it is, you know how it manifests. Well, we knew a little bit by this stage, but it was changing very fast, and especially what we didn't know was how many cases there would be in the places where we were doing this study. So we chose quite a large number. It was largely theoretical, and we aimed to enroll about 40,000 subjects, 20,000 to active, 20,000 to placebo. We gave 2 doses, 21 days apart, and then we started following the subjects. This is the detailed safety evaluation. This is absolutely typical for any vaccine. So, you know, if you're doing a vaccine efficacy study, this is the kind of thing you might do. You know, when you actually get started on it, we can come and talk about all these things. Now, we actually decided to do this as a global study. I have to be honest, most of the enrollment was in the US. And that's partly because of the ecosystem that exists in the US. In the US, I hasten to add, I'm British. You may not tell, I mean, some of you can tell from my accent, but I'm actually from the UK. I'm not American at all. But I'm a great fan of the American ecosystem for doing vaccine studies because they have a lot of clinical trial sites. Now, we did our phase 1 in very specialist academic sites. with people who were very, very knowledgeable about vaccines and about COVID. But at the same time then, to get 40,000 subjects, you can't do that in academic centres. And in the US in particular, there are a lot of sites out there set up to do trials. Some of them just vaccine trials. Many of them will do practically any trial that you need done. And they will do it very, very efficiently, and they will do it very compliantly with all the GCP regulations. Of course, you have to keep a close eye on it. There are always going to be centres that won't do it properly, so you have to get on top of them. And for that, we actually have in the ecosystem, we have a lot of checking, and the FDA does a lot of checking. So that background stuff is extremely important. But anyway, we went to all these very large number of sites and follow the subjects. The other thing that's important in a phase 3 efficacy study is to know exactly what you're looking for. There's a big difference between defining a case in clinical practice, where you have to decide how to treat a patient, and a clinical trial. In a clinical trial, you need a case definition that's going to be very, very highly specific. Because as soon as you start saying, maybe it is and maybe it isn't, you start then getting false positives in both the placebo group and the active group. And that really messes up the assessment of efficacy. It's a statistical problem. And so you need a very carefully defined, highly specific definition. And to be fair, the FDA agreed on this definition, which made a lot of sense to us. Certain symptoms of COVID plus a validated PCR test for the SARS-CoV-2 virus. And that's a classical vaccine efficacy definition. Symptoms, because you have to have, you're not looking to prevent just asymptomatic carriage, plus a defined microbiology endpoint. And we actually did that PCR in our own laboratory to make sure it was absolutely standard, that there were no issues about that. And I've just got to show this slide because it's so beautiful. We got a complete surprise towards the end of 2020, where when we looked at the efficacy results, and we'd actually gone on a lot, it turns out we'd actually gone on a lot longer than we really needed to, because when a vaccine is highly efficacious, obviously you need less subjects in the trial. We didn't believe it would ever be 95% efficacious. And this flattening out, and the other thing that surprises us here, and you're always looking out for surprises in vaccine studies, because it gives you some clues to how it's working. I showed you that the big antibody response comes after the second dose. The second dose was way after the curve in the active vaccine group flattened out. It started flattening out at about 7 to 10 days, which didn't fit with what we knew about the antibody response. So that means that antibodies are probably not the only part of the response that's critical for that early control. What we don't know, and of course the big question, and we didn't have, we'll probably never answer that question now, it's too late, is what would have happened if we'd only given the first dose? Would we have seen the same flag? Maybe not, you see. Another problem with vaccine trials is it's one thing to show protection after a couple of weeks, but what the healthcare system wants to know is, will this vaccine still work in 3, 4, 5 years' time? Am I going to need to give another dose? Because that completely changes the economic value of the protection you get from the vaccine. So persistence is a very, very important question. I just want to, before I go on to that, just to tell you about how we went from that first study was mostly in 16 upwards. In the US, it's possible to include 16-year-olds in what are basically adult trials. And obviously, they're a group that does a lot of mixing, so we wanted to control COVID. But then we went down to younger age groups. And we did actually we ended up adding a few 12-year-olds into that efficacy study that we later looked at. But we moved down in age. And this is just an example of the kind of bridging that we do to other populations. And because you can't repeat an efficacy study in every population. So you actually need to show, well, if the antibody response, and you can see in the blue, the antibody response in adolescents, children and adolescents 5 to less than 12 years of age, if their antibody response was at least as good as we saw in the adults in the efficacy study, there's every reason the regulators will presume that the efficacy will be as good. And just to add to the complications, you can see here, we actually did the 12 to 15-year-olds with a dose of 10 micrograms because we decided the 30 micrograms was a little bit too reactogenic. We tried bringing it down, and we went with that dose, and this bridging proved perfectly that that was an appropriate dose. And just to back it up, that's the statistics, that's a bit boring. Just to back it up, we also followed efficacy in that age group, and we saw practically the same Kaplan-Meier curve as we saw in the adult population, which supports. But the reason we do non-inferiority, immune non-inferiority rather than efficacy, is because you know when you take the blood sample, you will get the result. Whereas if you wait for efficacy, it can take you months and months and months. So we wanted to know sooner and get on with treating that population. Now, sorry, this is a slightly dull slide, but I just want to show you here. If you follow that orange box down, about halfway, you see that more than 7 days after the second dose, we got an efficacy onwards for about 6 months of 90.8%. And actually that was changing. So between 7 days and 2 months after the second dose, it was 96%. But by the time you got beyond 4 months, that efficacy had dropped to 80%. So that shows that the vaccine protection is wearing off, and that was something to deal with. So what did we do? We looked at a third dose. And you can see here the antibody levels in this study had dropped at day 1 before the booster dose. This was about 7 months after they'd received their second dose. But it jumped back up even higher than it had been originally with the third dose. So that really was the data originally supporting using a third booster dose, which I think everybody's implemented now. Just to be certain, we actually did an efficacy study looking at the efficacy of a third dose. Did a third dose actually prevent disease? And you can see, actually, we were surprised how well this went. And in a way, it's a bad thing, because the fact that the third booster dose produced such good efficacy means that the efficacy of the first 2 doses was really starting to wear off. But this was real evidence that we needed that third dose. Now, the reason for doing a booster dose is not only because the efficacy of the first 2 doses might decline. We all know that we've seen this big swings in new serotypes coming out. Sometimes you might think this is to do with vaccines, but in particular here, what was probably driving this was the normal human immune response, regardless of vaccination, around the world, because there were a lot more people getting natural infections than were actually getting the vaccine. And so we had to deal with this. And I'll show you this. Every time a new strain came out, a new variant of concern, we actually looked at how the serum we'd already taken from people after 2 doses of vaccine actually worked in terms of neutralizing that variant. And mostly it looked very good all along. The beta was a little bit lower, but we did have evidence of protection against beta. They all looked pretty good until we got to the Omicron BA.1. And then the antibodies really were the antibody activity was much, much lower. And that really showed that 2 doses was an issue. And this is just to show you different. We've looked at 2 doses. Here we've shown it in the younger age group, the less than 5-year-olds. And here you'll notice we'd actually gone down to 3 micrograms for this age group because we really didn't want to have young children getting fevers and reactogenicity and so on. We know with meningococcal vaccines that can be very confusing in the clinic, especially in the middle of a pandemic. So all of the age groups, including the older adults on the right-hand side, we saw lower activity after 2 doses against the Omicron. After a 3rd dose, we saw a higher level of antibody. Again, we don't really have a correlative protection. We don't know how much antibody activity you need, but generally speaking, more has got to be better than less. So that was really the reason why a booster dose of the original vaccine was recommended, not only to sustain protection, but also to start protecting against the Omicron variant, as well as the original variants that we'd seen. We actually did do a clinical trial, and we compared, we decided to go, it's not just us, of course, there's a lot of discussion with regulators and experts and so on. We decided to try out a variant vaccine where the spike was actually replaced with the Omicron variant, and we mixed it 50-50 with the original vaccine, because we know that the original vaccine gave pretty broad protection against most of the variants we'd seen up to that date. And so we put the 2 together in order to see how that would work. And that's this type of bivalent vaccine we've moved forward with. And just to cut to the chase, you can see for the Omicron variant, the geometric mean titer for this bivalent, the bottom row shows that we got a better GMT against the Omicron variant, geometric mean titer of neutralizing antibody, than we did with the original vaccine, the ratio 1.56. And that means the response was superior, as defined by regulators, against the Omicron variant than the original vaccine. And importantly, if you look at the antibody response of the bivalent and the original vaccine against the original strain, the Wuhan strain, you see they were almost exactly the same. So we didn't lose anything on that, but we did gain on protection against the Omicron variant. Importantly, we showed that the reactogenicity for the bivalent, which was 15 micrograms of each to make up a total of 30 micrograms, was exactly the same, pretty much the same as the original 30 microgram vaccine. Now, by the time we published those results, in fact, those results came out last week in the New England Journal. It shows you the journals are way behind reality at the moment, unfortunately. What did that do for the BA.4/5? Because by the time we actually did that trial, BA.1 had been replaced by BA.4/5. And we showed that although we did get antibody against that, it was lower than the antibody against the BA.1. So this is where we try to gain speed, because it takes us, even the best eco-structure in the world, it takes us at least 8 months to make a new construct, to actually get that vaccine out to clinical trial sites, consent and dose people, get their antibody response following the booster dose. But in a mouse, we can do that in 3 months. So that's about a 5 to 6 month gaining in terms of getting the information in a mouse compared to a human. Now, I'm not going to show you the data here, but we've actually, through the pandemic, we've seen that the pattern of responses against variants has been very similar in the mouse to humans. I mean, obviously the details are slightly different, but the broad pattern of response is much the same. And this is a mouse that's already received 2 doses of the original vaccine, then received a booster dose with either the original vaccine on the left or on the right with a BA.4/5 bivalent. And this shows that the response to a range of Omicron variants was substantially better than we saw using the original vaccine as a booster. So this really provided some justification for using a bivalent that contained not the BA1, with which we'd done the clinical trial, but with BA.4/5. And that was the basis, both the clinical safety from the bivalent with a slightly different construct, and this mouse data of the immunogenicity was the data that really justified to the FDA and other regulators moving forward with the bivalent BA.4/5 vaccine. I hope that makes sense. I'm going very fast. There's lots of abbreviations. But that was the fundamental justification. And this is the difference in timelines. So you're saving 5 months. Now, to us, that's a model. That's a model for potentially moving forward with newer variations, because already the BA.4/5 is out of date. So what do we do next? And, you know, throughout this pandemic, we've been making new constructs all the time. We looked at the beta, we looked at the delta, we've looked at the BA1, the BA.4/5. We've made new constructs and tested them, and generally found that, you know, the pattern, we've done some of them in humans, but by the time we'd got the results, we'd already moved on, and so we threw it away. That's one thing we've been doing through this pandemic, is doing a lot of work at risk. So we did a lot of work thinking it might be necessary, and then we threw the results away because it was the only way to get data to fast enough. Now, if I could just to say, there is a discussion today, I don't know if many of you know of the VRBPAC, which is the FDA's Vaccine Advisory Committee. They're actually meeting today to discuss whether they think we should move forward with the bivalents to completely replace the original vaccines. I don't know what the outcome will be. I'm sure many of you will be listening in later, but I think that will give us some good guidance. And we're not stopping still. So this is, again, a part of the ecosystem. We have to constantly try and be ahead. We're looking at a couple of different approaches. We're looking at modifying the spike insert to try and get better stability for the immune presentation. Early days, when we find one that works, we'll shout about it. And we're also looking at broader protection. So like many other, I mean, we're not alone in this, nothing, we're not unique in this in any way, but just to say, we're looking at using T-cell epitopes to try and get a broader response, so we can give the T-cell vaccine and the spike-based vaccine at the same time to try and give an added level of protection, particularly where we're a bit short in terms of protection against new variants. What helps with this is what we call the light speed paradigm, but I think the principles work pretty well for anything that you need to do in a vaccine ecosystem. A lot of surveillance. We worked with lots of people out there doing surveillance, and we watched very closely and worked closely with groups around the world looking at what was coming new, because you have to know what you're dealing with when you make a new vaccine. And in many vaccines in the past I've worked on, we actually set up the surveillance because it didn't exist in an adequate form. We've done a lot of parallel testing. I said we've done a lot of things in parallel, thrown the results away. Obviously, dose selection we did from experience. Very difficult. A lot of people thought our 3 microgram for children was a bit too low. It's a value judgment, really. Streamlined governance. Like every big company, we're full of committees. So we actually really worked to, we did everything appropriately in terms of safety governance, but in terms of other things like investments and so on, we moved much, much faster than usual. And I think that's a good experience in general. You have to be, what is the governance committee there for? It's there to make sure you're doing the right thing. Does it have to take a month to do that? Not necessarily. A lot of at-risk investment, especially on the manufacturing side. Now, obviously, for a normal vaccine, that's a value judgment, you know, how quickly do you move the investment forward. But also, the much better regulatory dialogue. And, of course, you know, we've communicated with regulators around the world, including the SFDA. We've had a lot of communication. Of course, they come back with a lot of questions, and we work really fast to try and get those responses back to them. We have a lot of people to thank for an activity like this, and that just shows you actually the size of the ecosystem you need to support vaccine work. It's not a small group, it's actually a very inclusive, wide group. Clinical trial participants, we had over some 60,000 clinical trial participants who gave their consent knowing that this was an unknown, to some extent they're going into. A lot of sites and investigators and their staff around the world, a lot of CROs and clinical trial partners, very, very important relationships with governments and with the regulatory authorities as we move forward. Not easy because everybody has their own views on this, and yet we don't want to make different vaccines around the world. So there's a lot of dialogue was very important. And of course, a lot of colleagues at Pfizer and BioNTech. So just a little glimpse into the making of a new vaccine. Thank you very much. So I think we now want to move on to a policy discussion.
Yes. Hi.
Oh, sorry.
I'm here. I'm back. No worries. Thank you, Dr. Stephen, for your greatly informative presentation. May I kindly ask you to stay with us on stage, and you can have a seat, because you're going to co-chair the next panel discussion. So ladies and gentlemen, we will dive now into something very interesting: vaccine development and manufacturing, enhancing success rates with comprehensive tools, innovation, and expertise. It's my pleasure to welcome the co-chairs of the session, Dr. Majid Al-Ghuraibi, Chairman, Infectious Disease Research Department, King Abdullah International Medical Research Center, KMARC. Welcome to the stage, Dr. Majid. And again, Dr. Stephen Lockhart, the Europe and Asia Pacific Vaccine Clinical R&D Advisor, UK. Looking forward to hear your in-depth insights about this promising topic. Thank you.
Good morning, everyone, and welcome to the Riyadh Global Medical Biotechnology Summit 2023. My name is Majid Al-Ghuraibi. I'm the Chairman of Infectious Disease Research Department at King Abdullah International Medical Research Center. I am delighted to co-chair today's sessions with my dear colleague, Dr. Stephen, the Vice President and the Head of Europe and Asia Pacific Vaccine Clinical Research and Development at Pfizer. We are delighted to have all here as we come together to discuss an important topic of the vaccine development and manufacturing that are of such great importance in the world today. With advance in medical biotechnology and the various vaccine development technologies, we have the potential to create more effective and safer vaccine than ever before. We look forward to engaging in meaningful sessions and exploring the potential of these new technologies to improve global health. It is wonderful to see so many professionals from different backgrounds here today gathering together at the summit to discuss some of the latest advancements in the vaccine development and manufacturing. We are very pleased to have a distinguished panel of speakers with us today. Our panel of speakers bring wealth of experience and knowledge on vaccine development and manufacturing. We are looking forward to hearing their thoughts and engaging in meaningful discussion. With great pleasure, I would like to introduce our first speakers of the session. Dr. Paolo Martini is the Chief Scientific Officer for the Rare Disease Hematology and Rare Alliance at Moderna, with more than 20 years of experience in drug discovery and development. Dr. Martini will be discussing the design and manufacturing of mRNA-based vaccine, and we look forward to hearing from Dr. Martini and learning more about the potential of mRNA vaccine to improve global health. So, without further ado, please join me in giving Dr. Martin a warm welcome.
Good morning, everyone. Yeah, you can hear me, okay. So today I wanted to give you an introduction of why messenger RNA is actually potentially the new methodology or technology to build quite potent and powerful vaccine. I just saw, unfortunately, the early presentation of Dr. Lockhart, but I just want to give you a little bit of an overview why we think messenger RNA could be a great technology for a vaccine. One of the things that it's important is obviously building the antigen. And one of the things that messenger RNA gives us is the opportunity of multiplex. What does it mean? It means that we can combine as many messenger RNA in a single lipid nanoparticle and create, at this point, a spike protein, or the protein that potentially our virus would present to our immune system in a more complex way, so that our immune system will react with a much larger repertoire of different antibodies against the spike protein. And you can see here that we can start to build something that is a little bit more complex. It's not like the old technology where obviously you build a peptide or you build partially a piece of protein that would represent the antigen, but not necessarily with the right conformation or post-translational modification that you needed to build the right antibody. And that's why when we saw the first data in the clinic and we saw that we had 94-95% of neutralization of the antigen, we were actually pretty happy, meaning that the design of the spike protein was actually really interacting with our immune system, activating B cells and T cells for memory, and then obviously starting to produce antibody. This is how basically it works. In the lipid nanoparticle, you can basically encapsulate, as I said, as many different messenger RNA as you want, and that's why you can start to create something that is a little bit more effective in terms of building antibody. Once the RNA is delivered to the cells, it actually goes to the ribosome where it gets translated, and the spike protein gets basically presented to the cell surface, and the antigen presenting cells, and then activating basically an immune response from B cells, precursor cells, and the T cells, also creating a memory. And when we were pleased from the very first immunization, we actually saw that we were stimulating, in fact, T memory cells, suggesting that potentially these vaccination could last a longer time. So that was a great impact that we had. And to be honest with you, obviously we don't have the longevity, neither the experience of Pfizer. We started in 2011. Yeah, we thought about vaccine. We started to build some vaccine, but COVID came as sort of a novel vaccine for us to build. But if you look at the timelines, it only took altogether 42 days to not only design the vaccine, but to produce it, purify it, and actually to scale it to a point that we were actually able to produce the first vial that were delivered to NIH, which was collaborating with us in order to start a clinical trial. So in a way, it was the fastest, for us, the fastest drug ever produced. And I think in the history of drug discovery and development. So one of the things that also gave us the opportunity to get to these timelines is that we really started in 2011 building a messenger RNA technology, building not only understanding the RNA, understanding lipid nanoparticle, delivery, etc., but really building the manufacturing, the scaling up of the system. And if you control all these aspects, you can actually have a better understanding of the drug that you are going to produce and eventually to scale up for obviously clinical trial. And so we can certainly say that all our manufacturing facility now can manufacture messenger RNA vaccine. Obviously, this was a pandemic, so you can imagine that although we are able to support not only vaccine from our manufacturing, we are also therapeutics. when we have a scale of a pandemic, you need also to collaborate with CMO that allow you to mass produce the vaccine. But in our facility, we actually can produce every single drug that eventually goes in a clinical trial and to support the post-marketing based on our facility in the United States. And so this is what, let me see. This is what it looks like, in fact, when we produce our vaccine. And more importantly, these are our bioreactors, how it happens, the encapsulation, and more importantly, the quality control. Sorry, this went a little too fast. But one of the things that is important that all our vaccines do not utilize any animal or human product, which means that the process is semi-synthetic and there's no cells involved, there's no animal product involved, so everything is pretty much synthetic of origins, chemistry and synthesis of chemistry, and more importantly, the vaccine is in an aqueous buffer in the end with a very little impact on the human body. And this is the sorry, this is the scale. These slides are going by themselves. I've never seen such. This is the scale of a normal bioreactor, and this is, I don't know what's happening, it's moving by itself. I swear I'm not doing anything. And this is basically what we have. This is for research production, and this is actually for large-scale manufacturing. So you see there's a huge differences in scaling of what we need in order to produce basically a massive amount of drugs and then to ship it. But one of the next important things is that mRNA technology for us is a fully integrated ecosystem, as obviously Dr. Locker was talking about. You know to know every aspect of it, from starting from the messenger RNA, and going into chemistry, bioinformatics, understanding how to design the spike protein, the process, the formulation, etc., and getting really to then eventually build a business strategy around it. So it's an integrated ecosystem and have everything happen at Moderna. And I want to stop here. I think this is my last slide. Everything is also digitally performed. So we have the aid of artificial intelligence that help us not only in building the right spike protein, but actually in all the aspects of the production of the vaccine. And with this, I think I will stop here, and I thank all my colleagues at Moderna, and I'll be happy to be part of the panel. Thank you.
Thank you very much. That was very interesting and looked very familiar to me. I'd just like to now invite our next speaker, Dr. Mohammed Basaid, who's the Associate Director of Clinical Trial Services at Kamar, infectious disease consultant at the Department of Medicine at the King Abdulaziz Medical City in Saudi Arabia. And he's going to talk to us about the challenges of performing a phase 1 vaccine clinical trial in his unit. Thank you.
Thank you, Dr. Stephen. So it's a pleasure being here, and I would like to thank Dr. Lasker, Dr. Howdy for great organization for this event. So I'm sure you have seen this, and what we are going to talk about is the phase 1 clinical trial. And as some of you would know, that traditional vaccine development will take years and years. And that's because of a lot of things you need to do in the lab, moving to the clinical phases, and then to the approvals, and then the post-marketing studies. But recently, in the last 3 years, and you can see here Dr. Stephen and Dr. Paolo from Moderna and Pfizer changed that. So we had an accelerated timeline. We have an expedited process we can work on, and we can actually have an emergency vaccine available in a very short time. And that's what KMARC and National Guard are trying to do. And we are trying to build this ecosystem where we are already having the experience, and that was one of the things that we were focused on, which is the phase 1 clinical trial. We know the challenges. So we know that we need to focus on funding, infrastructure, the culture and belief system, recruitment and consent processing, and that's in general the community engagement in clinical trial. And then we need the experience, the investigators who are able to conduct such a clinical trial. So I'll just take you through our journey. So it's a phase 1 clinical trial. We had experts from Oxford, part of the team, and also we engaged the Saudi FDA to build such a unique and first phase 1 clinical trial in the country. So after a lot of animal studies, lab studies, and discussion with our colleagues and collaborators in Jenner Institute, we started in 2016. So as early as 2016 up to 2021, we had the discussion showing that we are interested to join the early phases clinical trial. And then later in 2017, we had our discussion. We shared our interest with Saudi FDA, with all other partners, and then in the next year, we started working on our documents. We worked on our experience, our dossier, and that took around 1 year. And by the end of 2018, we were able to submit our papers to the Saudi FDA, and then another year of discussion to improve the submission, revision. A lot of constructive things have been done. And fortunately, by around the end of that year, we received approval for a unit that's ready for phase 1 clinical trial and a phase 1 clinical trial for Chaddock's MERS vaccine. We worked at that time with our medical services in King Abdulaziz Medical City. We have a unit that's dedicated for KMARC, for phase 1 clinical trial, fully equipped, short and quick access to all emergency things that might be needed. We worked on our team, either internal training, or we sent some of the teams to Oxford University for further training. We also worked on outreaching the community. And that was interesting because we did not know how people would accept that. And we had very interesting and positive feedback. A lot of people are willing to volunteer in a phase 1 clinical trial. And then we again worked on our team, the patient journey. A lot of exciting stuff were happening at that time. I'll not take you through that, but we have 3 doses. We are trying to make sure that everything was done as per the protocol. So the vaccine was shipped, and on December 2019, His Excellency Dr. Bandar Ganawi was there to officially open the first phase 1 clinical trial in the country. And we started our phase 1 clinical trial on the 17th of December. So another major obstacle. So early 2020, we had the pandemic. And with a great team, we were able actually to continue making sure that the patients are safe, and we are following our patient till the end of the study. I will not take you into the details, but it was finally published in The Lancet with the support and recommendation to move to a phase 2 clinical trial. So in general, it was a great experience. We are sharing this experience with all other stakeholders over the last 2 years. I think we are showing case that we can do a phase 1 clinical trial. Now we have the team, we have the place, and with the cooperation with all stakeholders and vaccine developers, sponsors, and the regulators, I think this is doable. Thank you.
Thank you very much. I'm sure we'll be able to tell us a lot more about that in a while. And our next speaker is Dr. Neil Donegan, who's Vice President of Business at an Australian company called VaxMed. And he's going to be discussing a completely new technology which may make a big difference in future, synthetic vaccines. Thank you.
I'd like to thank thank you for the introduction, and so thank you for the organisers of this fantastic meeting. You've done a great job. So VaxMed was founded by a couple of guys who had actually worked on vaccinia and MVA for actually more than 20 years. They were the first to sequence MVA and have essentially reverse engineered it to be a synthetic vaccine platform. It's fully synthetic, a non-replicating double-strand DNA. It's around 90% of the MVA genome with some genes removed. From the safety profile, it derives from vaccinia, which has been in around 300 million people, and MVA has been in around 300,000, and there's no adjuvants. On the efficacy of the vaccine, it's got a 30 kilobase payload. Obviously, when you're looking at vaccines, it's a numbers game. The more epitopes you've got, the more or the broader B cell response and the T cell response you can generate. On the preclinical, there have been successful challenge studies in some rodents and non-human primate. On the manufacturing side, it's a suspension culture with replication in a rescue cell line, serum-free, essentially producing thermostable sort of VLPs that are then introduced to the cell. The advantage of having suspension culture is that essentially with the roller bottle culture with MVA, you could only produce at max 50 million doses globally a year, but within a 3-week period, or we're able to produce up to 1 billion doses on this process. So we've got 5 vaccines have been synthesized in silico, a second generation SARS, respiratory syncytial virus, so universal influenza virus, also staph pseudomonas and African swine river African swine fever. We've got, as all biotechs have, a bunch of collaborators. We have the DNA synthesized by Thermo Fisher in Germany, and then we stitch together in the construct. We've introduced 12 some cloning sites, and so we can drop in a whole bunch of antigens. At the moment, we're involved in a capital raise. We're looking to raise $20 million US dollars to fund our phase 1, and we're looking to run a phase 1 in New Zealand, and we're in discussion with groups here with the idea of having essentially co-phase 1 in Australia and Saudi. Okay, thank you very much.
Thank you, Dr. Neil, for this great presentation. Our fourth speaker for today is Mr. Justice, the Vice President and the Head of R&D Strategy and Planning Office at SK Bioscience. Dr. Shaw will be discussing SK Bioscience core capability in vaccine, R&D, and manufacturing, including their versatile platforms to address both pandemic and ordinary needs. The stage is for yours.
Thank you very much indeed for the kind introduction. So I choose to stand here to hide my valley, and I'd like to also spend some time about SK Bioscience and its recent efforts around COVID and vaccine development and manufacturing. So first of all, I'd like to express that it is my great honor and privilege to be a part of Riyadh Global Medical Biotechnology Summit, and I would like to extend my sincere gratitude for allowing me to speak on behalf of SK Bioscience especially in terms of vaccine R&D and manufacturing. So my name is Justice Hejenjo, the head of R&D Strategy and Planning. Today I'd like to deliver a couple of key messages that we have learned from pandemic, which can be topics to further consider in the light of building capabilities within the region, starting from our introduction and experiences. So the name of SK might be unfamiliar to you, but basically SK Bioscience is a leading vaccine company. It's a fully integrated vaccine pharmaceutical company. And since its foundation of R&D center in 2007, SK Bioscience was able to build up rather comprehensive capabilities in process development and manufacturing. At the same time, SK also gained multiple global partnership experiences, not only with private companies, but also with philanthropic organizations, including the Gates Foundation and CEPI and WHO. And based on those experiences, we were able to successfully develop and launch the CEPI-funded, the first homegrown COVID vaccine in Korea, and also acquired European GMP certification through CDMO and CMO services for other COVID vaccines. These contributions were realized based on two core capabilities. The first, process development capability powered by a variety of different platform technologies. And secondly, manufacturing capability featured by the combination of single-use system, multimodular designs that enable to manufacture many different types of bacterial and viral vaccines, as well as different types of formulations. And on bottom of these, speed and quality of execution for the seamless development and commercialization is a part of our DNA. So based on those core capabilities, during the pandemic, we were able to successfully serve as a manufacturer and played a critical role as a developer at the same time. This is surely a good example to show that the importance of flexible and versatile platforms in terms of manufacturing that can be useful not only in pandemic time, but also in peacetime. Influenza vaccine is a good example. So we switched influenza vaccine manufacturing suite to COVID vaccine manufacturing suite very swiftly. During the COVID pandemic, SK was able to serve as one of key players in R&D, process development and manufacturing, and rolling out of vaccines in need. But we also realized that we need to get better prepared for the next one. So to make a long story short, these four things are the topics worthy of further consideration to get better prepared for the next pandemic. The first, vaccine candidate and libraries for potential pandemic threats studied in advance. And second, platform technologies for rapid response to be secured. Thirdly, expansion of manufacturing, expansion of infrastructure for R&D and manufacturing. And last but not the least, active engagement and close collaboration with key stakeholders in vaccine industry, such as CEPI and Gates Foundation and WHO. So in order to get prepared in peacetime and pandemic time as well, we are currently building up a brand new research and process development center in Songdo, Incheon, Korea. It is supposed to be completed in the first half of 2025 to come with CGMP standard pilot plant and open innovation hub to invite global partners to work with us and train talented people to develop and manufacture vaccines. We are also expanding our commercialization manufacturing capabilities in order to properly responding to the needs of vaccines by making best use of our versatile and flexible manufacturing platforms. So at the end of my presentation, I'd like to spend some time on the initiative to share our capabilities in terms of process development and manufacturing, which is called the SkyShield. SkyShield is basically maximizing the advantages of flexible modular manufacturing infrastructure to the regions or countries by transferring SK's technology. So it's about building up manufacturing capabilities to supply vaccines in needs. So for example, if a country of interest is in need of PCV and influenza vaccine, we shall plug and put the same suites we have in Korea, together with appropriate formulation technology. SK will also dispatch people to make tech transfer, also invite some people to make some trainings, and they will be equipped with manufacturing capabilities. So SkyShield represents a social and enterprise value and next pandemic preparedness and business sustainability as one solution package. So my belief is that this experience of success can be transplanted to the places with strong commitment on national security by working together with SK as a one team. This is the end of my presentation, and thank you very much for your kind attention.
Thank you, Dr. Justin. I think virtually we'll have Dr. Scott with us. Dr. Scott is the General Manager of Nucleic Acid Therapeutic Business for Cytiva, leading the global commercial go-to-market strategy. He will be talking today about making innovation reality strategy for future manufacturing requirements. I think we'll have Dr. Scott soon with us online.
Good morning.
Can you hear us?
I can hear you well. Can you hear me?
Welcome to the Global Summit, and we look forward to hear about your experience in vaccine development and manufacturing.
Thank you very much. And first off, my apologies for not being able to be there in person, but very much wish you a successful summit. I'm going to share a few observations from Cytiva in terms of how we see future manufacturing strategies for both COVID vaccines, but also for future both communicable and non-communicable diseases and manufacturing approaches to those. We've had great talks earlier from our colleagues at both Moderna and BioNTech and others around the sheer therapeutic promise of mRNA, and I'm really going to focus here on mRNA. Cytiva has a long history of supporting the GMP manufacture of both monoclonal antibodies, viral vectors, and cell therapies with GMP manufacturing infrastructure. But most recently, I have now started a new division focused solely on the GMP manufacture of mRNA-based therapeutics and vaccines. And why do we see this as being important? What you can see here is there are over 500 mRNA therapeutics and vaccines in development globally right now. The vast majority of those have been focused on infectious diseases. COVID has obviously been the most prevalent with the work of both Moderna and Pfizer, but increasingly follow-on diseases, whether those are rare infectious diseases or more common diseases like flu and RSV. However, I think about sort of future manufacturing strategies for Saudi Arabia and for the world. A big part of what we have to consider is how manufacturing facilities are leveraged, looking at the uptime in those facilities. We don't want to be considering white elephants that sit idle. We really want to be considering manufacturing facilities that are able to both approach pandemic response with mRNA, but also accommodate future needs. And really one of the take-home messages you can see here is, as well as the infectious disease approach, there is a big focus on non-communicable diseases. Oncology is clearly one of those, and I know that is a concern in Saudi Arabia. There are clinical trials ongoing for cardiovascular, and I know oncology and cardiovascular will be other topics in the summit. So as we think about the strategy for manufacturers, how can it accommodate the current pipeline, but also the future pipeline? If we can step forward to the next slide, please. What we've seen in Cytiva is when we look at that in terms of future manufacturing requirements, obviously COVID was a global response. It was really, how can we quickly vaccinate the entire planet against that highly impactful disease? And manufacturing scales there were in the hundreds of kilos to address that globally. But if we go back to that previous pipeline, what we start to see is that for follow-on infectious diseases, we look at local manufacturing, and if we consider the future pipeline for perhaps oncology and others, manufacturing scales start to come down to between a 1 to 50 kilogram range. The other thing that we've seen is, as much as we think about mRNA as a single molecule, there's huge diversity in approaches to manufacturing mRNA. Future mRNAs may be more efficient, such as self-amplifying RNA. So we really have to consider flexibility in mRNA types and also in scale. And at one end of the extreme is personalized therapies. If we go to the next slide, please. So here at Cytiva, we've focused on how we can enable that flexible manufacturing capacity, not just across a workflow, so from DNA template through to final fill finish, including proprietary technologies for both IVT, purification, and mRNA formulation, but also how we can start to accommodate that future pipeline, really enabling manufacturing ecosystems that can respond to both pandemic response. Our previous speaker was talking about clinical trials, which are going to be at much smaller scale. So we've focused on how we can enable both scale for GMP manufacturing across process development, early clinical trials, scaling up to clinical, but also accommodating that entire need. And that's scaling equipment from process development through to fill finish, including flexible single-use components. These are delivered as a complete manufacturing chain, which Cytiva can support the entire project management. In fact, through the advent of mRNA, we've seen a great uptake of this solution globally. I think one of the key things here from Future Supply is that this is built on a very established backbone, because of course one of the considerations there is future supply of those critical components. Having a facility where the uptime is not able to be met is also a challenge. If we can jump to the final slide, please. So just some take-homes from me as we start to think about future manufacturing capacity. I think first off, as we start to think about the future pipeline, it's considering manufacturing at all clinical stages and scales. A flexible facility will see high uptime, that will see the staff remaining skilled, it will see high utilization. The future pipeline is going to be variable, so you need to consider modular platforms based on standard manufacturing components, but highly modular. Digitization and automation are going to be key to consider the number of batches going through these facilities, and also to de-risk from human downtime, and really establishing quality processes relevant to the manufacturing right from the off. So here at Cytiva, we're very keen to support the Saudi efforts to continue to build out flexible manufacturing strategies. And I'm going to pause there and hand back to the panel. Thank you.
Well, thank you, Anthony. I think there's a lot there to talk about, actually. So perhaps I could start off with a question for Dr. Basaid. What would have made your phase 1 study easier that is feasible within the next few years? What would you focus on to make that work better?
Yeah, so you mean at that time or for the future?
Well, from your learning, what are you going to do now to make the next one easier?
Yeah, a lot of things, I think. And I think when you go in and mix your hand dirty and know what is a really obstacle and what is really not, and then now we can focus on things that I think we were not maybe aware that it's maybe more demanding. So I think we need to work more on awareness, not only for community, because I don't think community was our problem. I think even within the healthcare system, people need to know more about the importance of having early phases clinical trials. And then also building experience is really important. What helped us a lot also is the support. So I think always engaging people very early on is really helpful. And I think that's the best thing that we did within our clinical trial. So if you can see that we started 2016, and since then everybody was aware about our intention of doing a phase 1 clinical trial. And that made it easy rather than coming with a vaccine ready and say, you know, I want to start tomorrow for a phase 1 clinical trial. So I think these are the things that we learned, at least from this.
Great, Dr. Mohammed. I think clinical trial phase 1 was a milestone in Saudi Arabia, and I think that's a starting point to kind of expand our experience in clinical trial and vaccine development. Going back from the clinical trial to looking deeply in the infrastructure and planning to build a vaccine development and manufacturing. Going to Dr. Martini, with your experience working with several projects here in Saudi Arabia, collaborating with them, what is the needed resources that you think it's very important nowadays to be a kind of a leading country in the region to be manufacturing a vaccine in Saudi Arabia?
So obviously, I think the most important thing is understanding, first of all, the structure of the antigen, right? So bioinformatics, artificial intelligence that can really help. And I know that you guys have an incredible program with artificial intelligence here at KMark particularly. And those are important tools to build the right structure and potentially to start to predict future mutation that could change the conformation of the protein, which will allow for potentially further infections unless you have an incredible repertoire of antibodies that neutralizes the protein. So to begin with, obviously, it's understanding how to build the spike protein and how to build the best spike protein. Then obviously in the case of COVID, we have seen that the Omicron variants actually collapsed quite a bit the structure, and that's why we needed to create a novel vaccine, and so what we call the bivalent, which was a combination of the ancestral sequence plus the Omicron 1, and now next step was to add also Omicron 4 and 5. And sometimes when you have changes in the structure of the protein, that's when you see less ability of the antibody to really neutralizing the vaccine. So starting from the sequence is the most important things. And then in terms of manufacturing, it's not difficult to manufacture RNA. As I said, it's a semi-synthetic process, and I think obviously Scott from Cytiva mentioned that they are also starting a process for building a large quantity of messenger RNA. It's all about understanding, first of all, how to create a 100% reliable transcript from a DNA template, how to cap it and how to polyetail it, so you have actually a sequence that is absolutely highly comparable to the wild type sequence, and more importantly, that gets translated very well. In the manufacturing, obviously, the modification of certain enzyme, like the T7 polymerase, the capping enzyme, or the polyetail enzyme, are critical to create that 100% pure sequence. Unfortunately, small fractions of RNA could cause further immune response and potentially side effect, and that's why it's important to have a fully sequenced and fully integrated messenger RNA that then is encapsulated. Then, it's pretty simple compared to other technology because in reality, the immunogenicity is induced not only by the messenger RNA itself, which is not modified, and therefore is going to be recognized in fact as a virus, because our body tends to recognize RNA as viruses. And more importantly, to use lipid nanoparticles that do, in fact, induce an immune response at the site of injections. You don't need any adjuvant, you don't need anything else. As I mentioned, our vaccine, in fact, is a trace buffer, right? It's nothing very special. So controlling the manufacturing and all these aspects of creating the right sequence, it gives you sort of the fidelity of a highly purified, highly active molecule without using extra materials.
Definitely, with the new technology and sequencing, understanding the dissemination of the variant, knowing the right target, will make it easy for using the right vaccine technology to upgrade even. We are seeing now upgrading the vaccine to cover more variants of COVID. And that will be something incredible to see that happening quickly.
And you can start to do prediction, right? Prediction of the possible potential mutations and the possible changes in conformation of the spike protein, and start to build out of that. Unfortunately, we didn't expect that the COVID-19 would mutate so fast and would create so many different variants in such a fast manner. On the other hand, now we have a technology that allows us really to build something very quickly against all these other variants, and that we were just discussing before, I have a 5-shot in myself. I'm 100% Moderna, just to say, to be fair to my company. But, I actually had COVID, and I didn't know that I had it. I had no symptoms, I had nothing. I just got tested because at Moderna we were tested on a daily basis. And I realized that I was positive, and I didn't even know. So sometimes, building the right spike protein and the right molecule, it does certainly help.
Thank you. I agree. I think it's been a great and continuing competition. It's kept us both on our toes very well. So it's been very successful.
And I love that, right? Because in the end, it pushes us for a better and better product all the time. And so if there was no competition, I think that would sort of relax a little bit the system, and I don't want that. In reality, competition is helping in creating even better drugs for the future. So I'm glad that this is
I agree. It's been very successful, and of course now everybody's after our lunch as well. So even more competition, which is always helpful.
Right.
So let me move on.
Yes.
Yes, Scott? I just wanted to comment briefly there, and my colleagues from Moderna and Pfizer made a great point there. I think they've clearly brought to market the first generation COVID vaccines at sort of record speed, but as the colleague from Moderna was describing, there is great science going on now looking at variants of capping strategy, enzyme evolution, sequence design, and formulation. So going back to sort of manufacturing strategies, what I think is we're going to see is future developments that are going to see variants on mRNA, and any future manufacturing strategy has got to consider how it will accommodate the science that Pfizer and Moderna will drive. So I think a common strategic backbone, but building that flexibility into the facilities is going to be absolutely key.
Yes.
Yeah.
Agree. Well, and I think being pathogen non-specific actually makes that a whole lot easier. And I'd also just add, and a great point, Scott, of course, we're talking today about vaccines, but vaccines is probably just the very, very beginning point for the RNA revolution. So let me move on, Dr. Donegan, because you're on a slightly different side with the DNA, as I recall. How transferable do you see the DNA manufacturing being your synthetic vaccines? Because a lot of talk we hear about now is about local manufacturing and people wanting to have the capacity in their own country for various reasons.
It's certainly transferable to a wide number of infectious diseases. The fact that our Synvax platform has got a payload of 35 kb, there's the potential to drop in that's up to 12 antigens, and we know that as I mentioned, essentially more is better when it comes to epitopes. Looking at the safety profile, it's a non-replicating virus. It actually doesn't detour to the DNA. Everything's done in the cytoplasm. So with respect to specific countries, the manufacture requires a footprint of about 50 square meters using existing bioreactors. So in principle, like a country without a sophisticated vaccine platform that could become involved. But I think looking at one of the slides I put up where we've got in Australia maybe 15 collaborations on the technical side and probably the same number on the commercial side. So I think if a country's looking to develop a vaccine platform, as they're doing here in the Kingdom by running phase 1 trials, they're establishing the diagnostic support around those. And so over time, you build up the different components. And like in Australia, we're not synthesizing as I mentioned, we're not synthesizing our DNA. We're getting that done by Thermo Fisher in Germany. So as a platform, it's certainly something that can be dropped into different countries. And I think it's in, say, North Africa, for example. I think the Kingdom is actually well positioned where they can look at niche infectious diseases and looking at the cost economics of actually producing DNA vaccines on, for example, the Synvax platform. There's not the same infrastructure. Everything can be done in an S1 lab. So I think it's and certainly the enormous development that Pfizer, Moderna, and everyone that you guys have done has kind of led the way to bring a global focus back onto the vaccines and the importance of having vaccines and being in a position where you can actually get ahead of a virus and not be chasing it. So I think there's just, in principle, there's no reason why small countries can't set up their own industries around that.
Thank you. Thank you.
Yeah, that's a very kind of interesting journey to see how different platforms kind of forming the future of biotechnology, of vaccine development. But going to Dr. Justin, through your experience in SK and looking for the pandemic globally and working in developing a vaccine, what's your perspective of transferring that knowledge and technology to Saudi Arabia? What's your recommendation through your experience?
So basically the points I wanted to highlight today is the versatility and the variety of set of manufacturing platforms that can be responding to many different types of antigens that include viral and bacterial. And also in terms of drug product perspective, we also need to get ready for manufacturing vials and prefilled syringes and fried rising formulations and even for oral droppers. So that was the point that I wanted to highlight. And I believe during the COVID pandemic, we were able to be responding to the other companies' requests. For example, we also manufactured and supplied AstraZeneca's viral vector vaccines by manufacturing the products in our facility, and we supplied those products to globally and to domestically, of course. And we also had a CDMO contract with Novavax to manufacture their products, the protein recombinant protein vaccine with adjuvants. And we were also able to manufacture the products to supply those, to responding to the pandemic threats. So that was possible because we had already built a certain level of infrastructure to be responding to process development and manufacturing. And I believe those kind of success stories can be realized in other places, and that is the concept of Sky Shield. And we are very much committed to doing that initiative in other places, and we also have many requests and proposals from other countries and governments. So I think that's something that I wanted to highlight.
I can see here you highlighted the importance of multi-sectoral, different kind of collaboration with different manufacturing and companies work together toward very effective vaccine, starting from the raw material, from the research, the raw materials, and also to have the vaccine ready to develop to be reach the end of the journey for clinical trial. Also, you have mentioned here, it's very important that what I see as an infectious disease scientist here developing the vaccine for viruses, but also recently there is an interest to develop vaccine against bacteria, especially for the antimicrobial resistance. And I have lovely discussion with Dr. Stephen yesterday about that and the potential of developing vaccine of what we think is the next pandemic, which is the antimicrobial resistance. And that will be a great something to consider for moving all that experience and manufacturing to antimicrobial resistance.
Yes, that's a wonderful plan, and we'll be delighted to have such discussions, of course.
Great. I think we are on time, and I think all that experience we have here on the stage, different vaccine platforms, we have Moderna, Pfizer, and different technologies that is trying to provide the services that will be able to make a very effective and safer vaccine more than ever. Thank you for your time, and I would like to thank our distinguished speakers of that very interesting discussion, and all of you guys here having with us to hear about the vaccine and manufacturing. I'm looking forward to transfer all that technology and knowledge to Saudi Arabia to have a reliable and sustainable vaccine and development in Saudi Arabia. Thank you for your time.
Thank you, co-chairs and esteemed speakers, for a great session focusing on the valuable contribution toward vaccine R&D and manufacturing. Ladies and gentlemen, we'll now take a quick break for 10 minutes. So stay connected and we'll be back soon. Thank you. Welcome back, everyone. Ladies and gentlemen, our next session will bring together our international speakers to discuss in-depth therapeutics, development, and manufacturing. It's my pleasure to welcome onto the stage the co-chairs of this session. One of them only is joining, and the other co-chair is joining virtually. Dr. Badr Al-Muzaini, Deputy Chairman, Medical Genomic Department, KMARC. Welcome to the stage, Dr. And Dr. Linda Chin, President and CEO of Apricity Health, joining us from the US. And Dr. Anita Ben Taylor, Dr. Hani Gabra, Dr. Mohammed Bouzain, and Dr. David Erby, and Dr. Steve Rees. Welcome all. Dr. Badr, to you.
Thank you so much. So, good morning, everybody. So now for this session, we will have like the major topics. It's going to be transforming disease treatment with the new therapeutic modalities. For today, we will have like 6 distinguished speakers. We will start, at the beginning with Linda Chen, remotely. Hello, Linda. So we missed you last night, Linda.
Good morning. I'm sure I missed you more.
Yeah, so Linda Chen is the President and CEO of Apricity Health USA. Today she will talk to us about the dynamic precision, the new frontier in precision medicine. Mike is yours, Linda.
Thank you. Good morning. It's a pleasure to participate in this summit remotely. And my disclosure, as you heard, is that I'm the founder and CEO of Apricity Health, and I will be using Apricity as an example of the ecosystem infrastructure that's needed to usher in a new era in precision medicine. Next slide, please. Over the last 2 decades, cancer genomics have evolved from sequencing the bulk tumors at diagnosis to deconstructing the complex tumor microenvironment down to single-cell level, revealing many new and novel points of therapeutic interventions, some of which you will hear about later in this session. At the same time, medicine is being disrupted by digital health technology. We now have the capabilities today to continuously monitor a person's biometrics and quantify their behavior in a health or in sickness. When these two technological trends converge, we can now, for the first time, generate a dynamic living map of the complex genomics and phenomics interactions that is unique to each individual. Therefore, giving us the possibility to gain insight to treat our patients as a person, not just as a diagnosis. Next slide. But to effectively harness such genomic and phenomic data for development of new medicines, we need a different kind of platform, an ecosystem platform that can connect patient care with drug discovery and development, so that the drug developers can learn in near real time from patients continuously to make decisions about what target to drugs, which patient to select for clinical trials, and in what clinical context would the drug be most beneficial. Apricity Health is founded with the goal to build such a platform and therefore to create a virtuous cycle of better care, better insights, and better therapeutics. So let me, in a very brief time, talk a little bit about each of these pillars. Next slide. So on the care delivery side, Apricity has built a virtual cancer center, and its focus is on treatment management by augmenting care of the general practicing oncologist. It is a tech-enabled clinical coverage for the treating oncologist so that patient can have 24/7 monitoring as if they're on a clinical trial or at premier high-resource academic centers. With this AI decision support and a dedicated team trained to manage cancer symptoms, Apricity Care is an efficient way to democratize clinical best practice so that every patient, no matter who and where they are, will receive timely and optimal side effect management. That will maximize their chance of treatment success. And this is very important, particularly when we are talking about novel treatment modality or complex combinations, where clinical experience in general practice may be lacking. So when apricity care is helping the oncologist better take care of their patients, it is at the same time generating and capturing high frequency and curated patient data in context of the treatments. That data allow us to understand who and why some patients will respond and others do not, and some would succumb to toxicity. Such insight will inform next generation drug discovery and development. Next slide. So let me take the example of immune checkpoint blockade. As you know, anti-PD-1 is an enormously successful drug, but nearly 75% of the patients do not achieve durable response. So to develop secondary immune checkpoint for these non-responder, we have deployed dynamic precision analytic to mine longitudinal phenotype and serial serum proteomic data of patients that are receiving anti-PD-1 as standard care therapy. And this have led us to identify non-responder signature consisting of multiple targets with distinct biology. And importantly, each of these targets has a patient selection strategy based on the blood-based signature. And that means higher probability of success in downstream drug development because historic data have shown that biomarker-selected clinical trials have 5 times increased likelihood of FDA approval. Moreover, an advantage of blood-based biomarkers are lower barrier to implementations and therefore faster enrollment and more diverse participants. Next slide. So very quickly, how does this three-pronged ecosystem work? As I've briefly mentioned, the Virtual Cancer Center uses technology to augment care and support providers to improve care of patients today and generate and capture patient data in that process. By leveraging that insight, apricity then identify human relevant target with blood-based patient selection biomarker for downstream drug development. Instead of developing this therapeutic program internally, apricity have adopted a model to create spin-out of NewCo or partnership so that it can maximize its shots on goal while mitigating risk, allowing apricity to pursue multiple opportunities. For example, apricity have launched its first biotech NewCo with some of the target biomarker pairs and to prosecute drug discovery and development with its lead program in this NewCo on track for IND in early 2024. Last slide. So in closing, I'm hoping to sort of give you a sense of how technology and data coming together will drive the convergence of precision care delivery and therapeutic development, ushering this new era of dynamic precision where we won't only develop precision therapy, we will have the insight to prescribe the right drug for the right patient at the right time during their treatment course. Thank you.
Thank you, Linda. So next we will have the Anitab Taylor. Anitab Taylor is a global technical marketing manager, immunoassay platform from Merck, United States of America. Mike is yours.
Great, thank you. Just wait for the slides to come up. Thank you. Thank you, Linda, for the great talk. And I'd also like to say thank you to the organizers of the event and for the invitation to speak at this summit. Even though it is virtual, I'm sorry I couldn't be there. So my name is Anita Taylor, and I am the Global Technical Marketing Manager for Merck, based out of Washington, D.C. And I'd like to take the next few minutes to provide a high-level overview of the drug discovery and development with a focus on R&D. Next slide, please. This is our disclosure slide. The life science business of Merck KGaA Darmstadt, Germany, operates as MilliporeSigma in the US and Canada. Next slide. So to begin with, this slide shows a high-level overview of the drug discovery and development process. So for any given drug, regardless of the modality, before it's approved to be on the market, it can take up to 14 years of research. And the cumulative costs are quite high, averaging up to about $2.6 billion, if not more. Now, for any drug to be approved, it has to be safe, effective, and the dose proposed is optimal for the intended use. The development process starts with the preclinical phase, and this is the first step in the process to generate evidence to support its safety and efficacy, where qualified drugs are put to the test by testing its effect in preclinical models, such as animals. And there are many in vivo models of choice depending on the modality. For example, biologics often use rats, mice, or non-human primates. Vaccines, non-human primates are preferred model of choice. Once safety and efficacy in these preclinical models are shown, the candidates selected then move forward into humans as first in humans. However, before you even get to that process, there is the R&D process. Early in this process, usually thousands of compounds are generated and screened to identify lead candidates that will undergo testing, usually in vitro models, in different model systems. Before it even goes into these in vivo testings, candidates are selected based on efficacy, chemical stability, as well as ADME tox, and so forth. And this process can take up to 6 years, even within inclusive of preclinical. Next, can you click? Nope, you're fine. However, a challenge with drug development is that it's a high-risk process with a high attrition rate. And a lot of therapeutics fail due to the safety. So only about 4% of the therapeutics make it onto the market. And often the drug fails at either phase 1 or phase 2 trials. And so by that time, you can imagine what the R&D spend is. So researchers need to have a solution that will enable them to earlier predict the potential of a failure, ultimately saving time and money. Next, please. Okay, thank you. Now, even though every therapeutic goes through a similar process as I showed on the earlier slide, the integral research process, the steps that go within that process will differ based on the modality. For example, drugs such as mAbs, once the target is ID'd, the antibodies are generated by whatever process, such as hybridoma generation, phage display, and so forth, and they're screened to identify a hit. Promising candidates that then go through an optimization phase to maximize their efficacy and minimize toxicity. Similarly, ADCs will also go through a similar process with like mAbs, only there is a conjugation to ADCs, and these also have to go analytical characterization as well. Similar for vaccines, such as mRNA, you need to synthesize, formulate, pack it into, for example, lipid particles before you can test it. There's a common theme that goes through all of these modalities, so it is modality agnostic. And they must all be tested for safety and efficacy, and extensive analytical characterization and QC occurs throughout the R&D process to optimize these candidates. Some modalities might be more challenging versus depending on the complexity of the molecule. Next, please. Now, as I said earlier, it is well known that drug discovery and development is an expensive venture, and a lot of the cost is associated with R&D. And I wanted to add a couple of quotes that I recently kind of came across, that the cost has risen in R&D more so to the late stage, mainly due to regulatory agencies making it harder to approve drugs. Further, the increasing complexity of advanced medicines and investments into treatments which do not end in success makes R&D even more expensive. So a lot of this has to do with the complexity of the drug, the efficacy and the safety, and the regulatory agencies being more stringent and asking for research as these drugs become more complex, certainly in the next generation therapeutics. And this ultimately drives the cost of the R&D process. Who does this ultimately impact? It impacts the end user, the patient, who will have to pay more for their prescribed therapeutics. Next, please. So since I'm here representing Merck, we are a global life science company, and our goal is to enable our scientists at every stage of their research by providing an end-to-end solution to facilitate some of these R&D steps. So whether you're doing molecular, cell culture, genomic, chemistry, our goal is to help provide solutions. Also, as we continue to learn from our customers and what their needs are, we continue evolving to work and provide a more efficient solution to facilitate the R&D process. Another important factor that I want to highlight here is that Merck, as a company, has an important goal to reduce our carbon footprint and being more sustainable. For example, one area is animal models by providing a go-to model of reuse or reduction. One such example is 3D cell culture organoid models. These are models that are coming of high interest into the industry, and we are heavily invested in offering animal-free options that fall within the global movement towards sustainable and ethical preclinical practice. These models enable you by reducing error and these improved models is critical to improve targeted therapeutic development. And in the EU, they are looking to reduce animal model studies by 70% by 2030, with a view to replacing this with more biomimetic models instead. And so the overall aim of us as a company is to enable our customers by providing tools and solutions to make better informed decisions much earlier on in the process, thereby mitigating some of the challenges that we are seeing today in the R&D process. And with that, I'd like to say thank you to the organizers. Thank you.
Thank you, Taylor, for this presentation. So our next speaker will be from the stage. We'll have here Hani Gabra. He's the founder and the chief scientific officer of Papyrus Therapeutic from United Kingdom. So he will take us through the Papyrus Therapeutic from bench to side. So mic is yours.
Great. So just waiting for the slides, I guess. Fantastic. Well, thank you for inviting me to speak. I have to say I've been absolutely blown away by just how smart and committed and articulate everyone here is. And your first problem or issue around human resources for the development of my medical biotechnology is already met. You're almost there. Keep going. So I'm going to tell you about Papyrus Therapeutics. And this molecule in the background is the crystal structure of a tumor suppressor gene with 3 immunoglobulin domains. So what's interesting about this is that as well as being a gene and then a protein that we identified and we did the crystal structure in my lab, it's also the drug that we are developing for Papyrus Therapeutics. And the only precedents for this have been the idea of taking P53 tumor suppressor therapy into cancer cells through adenoviral gene transduction. However, this is completely different. It's entirely serendipitous because the drug acts at the cell's surface, so you don't have to deliver it by gene therapy, you just have to get it to the cell. And of course, serendipity favors the trained mind. So just to remind you that the oncology market is expected to grow to $264 billion by 2026. There is plenty of space to really accelerate with your venture, your plans in Saudi Arabia. And specifically for Papyrus Therapeutics, we have a very well-structured 5-year plan that takes us from single to double-digit individuals up to around 200 people in 5 years. Our drug, which is also the tumor suppressor, called PYTX-004, is at this point ready for IND-enabling studies after 20 years of research. And as we heard yesterday, this pre-inflection point, post-research, pre-IND enabling studies is the ideal time for maximum returns as you move forward. So in addition to this single asset heading towards the clinic, we also have a platform which I'll describe, which potentially brings 126 follow-on molecules to this first one. And we expect to be in first time in human studies, phase 1 within 2 years, with a phase 2 inflection within years 4 to 5. So Papyrus Therapeutics has developed this molecule, which is an Fc fusion of recombinant OPCML. It's highly effective as monotherapy, and it's synergistic with other modalities such as receptor tyrosine kinases and checkpoint inhibitors. And importantly, it's applicable to almost 50% of all solid tumors. So that is potentially a huge line of sight space. However, we are very focused on ovarian and breast cancer, which of course reflects the previous research that we've done, as I am actually an ovarian cancer oncologist. Importantly, because this is somatically methylated, and the genomic people here will understand this immediately, this switches off the protein, but it also methylates the promoter, which you can detect from blood. So it comes with a ready test from the blood to identify patients who will benefit from this type of therapeutic approach. So the mechanism of action is well understood now, and it is, and I'm not, obviously it's a 5-minute talk, and if you invite me back, I'll bore you to death for an hour about this, but I'll keep it short here. The mechanism of action is about negative regulation of a network of receptor tyrosine kinases. So this drug switches off 9 oncogenic driving receptor tyrosine kinases simultaneously, which produces a massive shutdown in signaling for the MEK-inert pathway. It reverses EMT, it reduces metastasis, enhances chemosensitivity, and engages immunotherapy synergy. And it's well tolerated at effective doses. Here is the killer slide, and on the top left, you can see that this is a mouse model of PDX ovarian cancer, so patient-derived xenografts engrafted into NRG mice. And you can see in the bottom left that there's a dose-dependent massive inhibition, the red line almost going down to 0, when you give this drug intravenously to mice. With a huge cell kill, 93% cell kill, and in the bottom right, no impact in terms of safety on the animals. In green is our current asset. In pink and white are 4 other genes, and by shuffling the domains of these, we can create 126 novel molecules for screening as follow-on for this platform of tumor suppressors. So my final slide, just to say, it's brilliant to be here. I get a sense that you're going to be very successful in attracting biotechs and pharma to this ecosystem, and I look forward to engaging with you more in the future. Thank you very much.
Thank you, Hani. So next speaker will be Dr. Mohamed Bouzayani. He's the CEO of Ceramics Therapeutics, Johnson & Johnson, G Labs, USA. Dr. Bouzayani will talk to us about the universal vaccine against multiple COVID-19 variants.
It's a great pleasure to be here. Trying to move the slides. Next slide, please. So Sanomix Therapeutics is headquarters at Johnson & Johnson, G Labs in San Diego, California. Basically, we've been there for the last 5 years. And the headquarters there is a really state-of-art facility that is FDA approved, GMP certified, where we can make therapeutics and vaccines from phase 1 all the way to phase 4. The company is focused in several projects. We work on next generation vaccines, so we have different platforms. We use mRNA, LPN vaccines. We also have a platform with protein expression, especially using baculovirus to express different proteins and adenovirus. We have a lot of expertise in therapeutic antibodies for cancer immunotherapies, and I will talk about some new delivery platforms that we are using, especially nanoparticles and nanofiber strips. Next slide. So I will try to give one example from each of these projects. So for mRNA vaccine, we are working with the herpes virus. We are also working with COVID-19. And basically, our strategy for COVID is a little bit different. Most of companies, as you know, Moderna, Pfizer, they are focusing on the spike protein. So basically, the spike protein is the key protein that will have the virus, infect the cells and produce neutralizing antibodies. Our goal is to work on conserved region on the virus. So basically, we are looking to all different type of COVID, different variants, and we are taking the conserved region. And we are developing a cocktail of mRNAs, and those mRNA will be like universal. So independent of the mutation on the spike protein, for future pandemic, for example, we will have neutralizing antibodies that will cover a wide spectrum. And here I can show you one example of those cocktails. So basically we have collaboration with the UC California University, where we developed the transgenic mice, triple transgenic mice in BCL3 that we can check all our cocktails. And we have the first cocktail that is working in all the variants, and it's ready to start phase 2/3 clinical trial. Next slide. I will talk about the nanoparticle delivery platform. So these delivery platforms are really important and key. If you look to the nanoparticles, so for example, for mRNA, we use lipid nanoparticles to encapsulate the molecules. Here, we can put any kind of chemotherapy inside those nanoparticles. We can label them, and they will go and specifically get linked to the target, cellular therapy. So I will give you one example here. We work with the Hospital for Clinical Trials on patients with multiple myeloma. And for those patients, usually the treatment that is available is BTZ, the bertozomib, which is the chemotherapy used. So basically what we find out is about 30% of those patients develop resistance to the chemotherapy. So what we did is we developed those nanoparticles here. In this case, those are chitosan, and we functionalized them. So we did genomic sequencing on 2,000 patients, and what we find out here on the left side, we find out that the patients that have the resistance are here. They have an overexpression of the CD47. If you look here to the housekeeping gene, GAPDH, we use it to normalize again, so this is almost same level, and you have this overexpression of these genes. So what we did, we expressed the anti-CD47, and we encapsulated the nanoparticles with the BTZ. This is electronic microscope picture of those nanoparticles. Next picture, please. Next slide. And the results are really phenomenal. So those patients that didn't reply to the BTZ, now if we look to those nanoparticles, if we use, for example, PBMCs as control, we don't see any effect. If we use the nanoparticles just encapsulated with the BTZ, we can kill the tumor up to 70%. And that's in animal models and also in in vitro studies. And now, if you use the specific antibody, the specific CD47, we can destroy up to 95% of the tumor. So this is really excellent result that show that this platform is able to reverse the resistance. And of course, by changing the antibody and the target, we can use it for different type of cancer or infectious diseases. Next slide. And here I will show you another platform that we have. This is called the generation of nanofibers. via electrospinning. I'm not going to go into the details, but basically, we have a formula with polymer and solvent, and we can use any drug or proteins or DNA, RNA, and we can generate nanofibers that are loaded with the drug inside. So it's a brand new, innovative delivery platform. Those are some of the machines that we use at the R&D level or GMP for large-scale production. Next slide. And here I can show you some example of results. So this is the nanofibers with the drug inside. So here's the pictures that we get like with electronic microscope. And then we get those strips here. And those strips, we can put them inside the mouth, either under the tongue or against the cheek. And then what happens is this strip will dissolve immediately and release the API. So by releasing the drug here, we have really nice effects immediately, and we show that we can reduce the dose up to 10 times, and it's a new delivery platform. Next slide. So here's some application of this platform. For example, we generated nanofibers for oral cancer. Instead of using the chemotherapy, the regular way, we put these strips here, and we use like different polymer, as we said, and we use the classical chemotherapy for this cancer, like the 5-fluorouracil, cisplatin, carboxyl, and then we can put directly the strip on the tumor, and we got really phenomenal result in reducing the tumor very efficiently. This is another example with cervical cancer, with HPV. We can here, for example, formulate the strip that will have not only the chemotherapy, but we can also add antivirals against the HPV and some heparin sulfate, for example, to kill out the virus. And again, those nanofiber will reduce the tumor and get rid of the virus. Last slide. Thank you for listening, and I look forward to discussing in more details.
Thank you.
Thank you, Dr. Bouzian. So next will be Dr. David Earl. Dr. David is a senior distinguished investigator in an island company from United States. Dr. Dave will take us through like one of the potential therapy, the RNAi therapeutic, the past and the present and the future. Dr. Dave.
Thank you. I really want to emphasize what we said earlier. We've had such gracious and generous hosts here. It's been a wonderful time. And I think as you'll see as I go through this brief introduction to Alnylam, to RNAi, past, present, and future, I think you'll see there's a lot of excitement locally here about the future. Alnylam actually it's a Cambridge-based company that has now grown globally, but the name actually started with an Arabic word. There's an Arabic word that translates, I won't attempt it, but it translates as string of pearls, and that inspired the name Al-Nilam with an I, which is the center star in Orion's belt. One of our founders, Paul Schimmel, his aunt was an Arabic scholar, and so it was quite intentional that this was named for Al-Nilam. What I'll tell you briefly, though, is a story about passion for patients, focusing on patients, and working hard. And that piece, while we wait for the next slide, or I'll try it here. Yes. That passion for patients and a willingness to work hard, I'll emphasize what we said earlier, you already have that in spades. I've really seen it this week, meeting with your colleagues, and so there should be a lot of excitement here about what we can do together. RNAi will be new to many of you in that, and I'll go through the history briefly, but it was based upon Nobel Prize-winning medicine that harnesses a natural tendency for genes to silence themselves in their regulation. We won't have time to go through the details here, but it's a different modality. Small molecules, antibodies, we've heard about mRNA, vaccines. This is a different modality that harnesses this ability to silence any gene in the genome by cutting off the mRNA and the protein doesn't get made. Alnylam has now used this to build a
portfolio of now approved drugs. So briefly, in the interest of time, something that was first observed in petunias, where they took the genes to try to make the petunia more purple, injected that gene, and something strange happened. On the same plant, you had some that were mixed colors, and you had white flowers. And this is because the gene itself naturally gets cut into microRNA, into small segments, and that then downregulates through the mechanism that we've now taken advantage of, the expression. That work then was translated into C. elegans a few years later. You can see the timeline. Alnylam was founded around 2002 as this work was being made clear that it is in everything. It's in plants, it's in worms, it's in people, and it could be harnessed to bring new medicines. I won't go through the details here, except to note that all of this hard work I can feel very proud of indirectly because it wasn't done by me. I joined the company in 2014 to take this platform and develop one of the medicines that that's relevant to this community and the world. And so we now have 5 approved medicines, and I'll go through those in a moment, that span rare diseases. When you have a new modality, you often start with monogenic, very clear causes because you can have a large treatment effect, but now we've expanded that into more common diseases like hypercholesterolemia. So in the interest of time, so that's the past. And you see, we're finally arriving here in Riyadh, I shouldn't say finally, but in the kingdom to discuss things. But I feel like we've been joined here spiritually, if you will, for some time through this passion for patients and an interest in hard work. This is now the present. This is where our pipeline is. You can see the 5 drugs at the top for TTR-mediated amyloidosis, for acute hepatic porphyria, for primary hyperoxaluria, that's the one that's special to my heart. Also Leqvio for hypercholesterolemia, a more common medicine that's now being marketed through Novartis, and Amvuttra, which is also for TTR-mediated amyloidosis. These are now injections, subcutaneous injections that you give sometimes monthly, sometimes quarterly, and now we're up to biannual and even annual injections. The drug is then targeted to the liver to silence any gene in the hepatocyte as a mechanism. And as you look down this list, you can see we're now growing into hypertension and other diseases. But there will always be an interest in rare genetic diseases, molecularly defined diseases, and this is where I think the research that Alnylam has done was inspired by many of the publications from the physicians and scientists already in the kingdom, and we're working with those same people going forward. So then the future is a bit captured here from an Alnylam perspective, but I think just in the brief time I've been here, I see a future that's very, very much enriched by the opportunity for collaboration. We use human genetics through like the UK Biobank. We're making improvements to the platform. This is how we've got to annual dosing. And then we're also now hoping to get beyond the liver we have our first molecule in the clinic in the CNS, but we're hoping to get to other tissues. These are the types of opportunities we think that we can take this natural mechanism using our technology then to bring new medicines in collaboration. So I'll stop there and just say that it's been an absolutely wonderful week. I think what was emphasized is the most important resource is already here. It's the physicians that are so passionate about the patients. It's the hard work that's already being done. We just now need to put some pieces into place to enhance that further. So thank you.
Thank you, David. So the last speaker for this session will be Dr. Steve Rees. Dr. Steve is the Vice President, Discovery Biology in AstraZeneca. He will talk to us about the transforming disease treatment with the new therapeutic modalities. Mike is yours, Doctor.
Many thanks for the kind invitation to speak at the meeting today. And apologies again for not being with you in person. I had the privilege of visiting Riyadh just before Christmas to spend a couple of days with scientists at KMARC as we explore potential collaborations between KMARC and AstraZeneca. And I have to say I was hugely impressed by the ambition within the kingdom to grow in the life sciences space and grow within biotech and medicines discovery. and by the ability and energy and enthusiasm of the scientists that I met. I've worked for over 30 years in drug discovery now, so I've seen a lot of ideas come and a lot of ideas go, and I can genuinely say that I don't think there has ever been a more exciting time to be working in drug discovery. The technologies, the capabilities that are open to us today to understand disease and identify new medicines are really unprecedented at any time in my experience. We've just had a great example of that from the previous speaker from Analo. If we move on to the next slide really captures where I think we are in terms of transforming medicines discovery. We have a wealth of new technologies available to us to understand disease at the molecular level, at the cellular level. And these are technologies like genome sequencing. You can sequence a genome now for just a few $100s. 20 years ago, the first genome that was sequenced cost $100 million. And whether it's genomic sequencing, it's metabolomics, it's proteomics, these technologies allow us to understand disease and identify drug targets that are directly relevant to human disease. We then move on to new modalities. There are a whole range of new therapeutic modalities, and this will be the focus of this short talk today. New therapeutic modalities that offer the way to treat disease symptoms, but increasingly offer the ability to cure disease. And that's something that we haven't really been able to do in the past. And then, of course, a revolution in personalized medicine to inform, or better to understand how disease breaks down in terms of different drivers of disease, to ultimately ensure that we're giving the right medicine to the right patient, where we have confidence that that medicine will work. And we're seeing a revolution in this space in oncology at the moment, and that will transition into other diseases over time. If we move on to the next slide, please. The next slide captures the different therapeutic modalities that we're actively researching within AstraZeneca. These modalities, of course, are also being actively researched across the field. If we go back 10 years and you look at the pipeline of any pharmaceutical company, it was pretty much small molecules and monoclonal antibodies and not really much else. Across our pipeline today, we have a variety of different small molecules, so classic small molecules, but also compounds like PROTACs. So PROTACs are small molecules that, rather than inhibiting a protein, they degrade the protein, they destroy the protein, and offer potential advantages in terms of efficacy. We have a whole variety of different antibody therapeutics, from the relatively traditional monoclonal antibodies, through antibody drug conjugates, bispecifics and the like. The potential of cell and gene therapies. So cell-based therapeutics, either CAR-T type cell therapies in cancer, but also regenerative medicine cell therapies. Protein and peptide therapeutics, then a whole variety of different nucleotide-based therapeutics, and as I say, we've just heard some examples of these. And it is worth reflecting, 2.5 years ago, nobody on this planet had ever been dosed with a messenger RNA therapeutic. Billions of people have now had a messenger RNA therapeutic in the Pfizer and the Moderna vaccines, which is an astonishing example of how technology can impact to transform disease. And just imagine how this world would have been able to cope with the COVID pandemic had those mRNA vaccines, had the Oxford AstraZeneca vaccine not been there. So we really have this opportunity where these next generation therapeutics are able to transform what we do in terms of drug discovery and move from disease to cure. If we move on to the next slide, and I just have a couple of summary slides really to exemplify this, and I'll move through these quite quickly. Just touching on oligonucleotide platforms, I won't say much on this as we've just heard about it, but the potential of messenger RNA therapeutics in vaccines to treat infectious diseases, but also increasingly in oncology, cancer vaccines, I think will transform our ability to treat these diseases and prevent these diseases in the years to come. Antisense oligonucleotides, siRNAs, these are technologies that have been in development for 15 or 20 years, as we've just heard. They're increasingly reaching the clinic, and again will offer the opportunity to transform a number of different disease states. The next slide just captures the situation in terms of cell and gene therapies, and just simply captures the number of phase 1, phase 2, and phase 3 clinical studies active in the cell therapy space at the moment. And you can see them from the numbers here. There are hundreds, there's something like 800 cell therapy studies in phase 2 clinical trials at the moment. And the FDA is predicting that by 2025, which is just 3 years away, it will be approving 10 to 20 cell and gene therapy products, whether these are cell therapies in oncology or regenerative medicine therapies or gene therapies like AAV, and also the use of CRISPR to repair broken genes. And just moving on to my last slide, which really just captures a lot of what I said. I think we really are at a pivotal point in our ability to understand disease and bring forward new medicines. The opportunity is huge, and the opportunity is huge across the world, including within the kingdom. We have a variety of new drug modalities being developed to treat disease. We need to find ways of delivering those to patients. We need to find ways of segmenting patients and putting in place the appropriate diagnostic and biomarker approaches to do that. We now believe we can find a molecule that can modulate any drug target. So the concept of an intractable drug target is something in the past. As I've said, increasingly these medicines will allow us to cure rather than treat symptoms of disease. And that's particularly true for gene therapy medicines in rare genetic disorders. We do have challenges around how do we formulate these new medicines, how do we deliver them to patients, how do we manufacture them, how do we follow the patients once they receive these medicines. And of course, healthcare systems will have to put in place structures to identify patients and enable the successful delivery of these medicines to them. And that becomes more complex as we move into therapies like cell and gene therapy. So I will stop there. Hopefully I've given you a flavour of the work that we're doing at AstraZeneca, a flavour of some of the opportunities in the field. And I think we have, working together, working in collaboration, a huge opportunity to transform disease as we move through the next years and decades. And with that, I'll stop and thank you for your attention.
First of all, I would like to thank all of the speakers. I mean, like you took us through many strategical platforms for treatment, application, and other approach. I mean, for the new era of disease treatment. I mean, instead of using the conventional medication, now we move to mRNA, to monoclonal antibodies, and others. And as you know all, I mean, here in the country, we are heading to transfer the technology, and this global summit is part of the plan. I mean, to have biotech park. I mean, to extend the partnership with the big pharma company here in the country. So to focus more, or to link the treatment or the treatment modality with our vision here in the country, I would like to start, I mean, to kick the discussion by asking Linda. I mean, first, I mean, Linda, based on our vision and the discussion for today. So, how do you see the potential of virtual cancer center in Riyadh as a medical hub to democratize cancer care to the broader region in case A and beyond? And also, to be more specific, so what the leapfrog opportunity is to seamlessly integrated with the initiatives here in Saudi Arabia, either here in Riyadh, I mean, as a country, or a specific initiative, I mean, as example for that, the NEOM.
Okay, great questions. I think one of our focus in the Presidency is really thinking about how to take the best practice in major academic center, big hospital, and really democratize that to majority of patients who are being treated more in the community, particularly the rural community. And I think not having enough specialty trained expert oncologists, particularly as treatment of oncology cancer become increasingly complex, this resource constraint in terms of experts is real in the US as well as around the world, including in the Middle East. So I think the opportunity with the technology is for us to think about how to maximize the capability and capacity of our existing resource. And that's what the Virtual Cancer Center platform was built to do. With this AI system, it captures the best practice and use that to augment licensed and trained healthcare professionals to deliver standardized and consistent services to maximize the time oncologists can have to manage more complex issues while making sure a patient is getting that 24/7 proactive care, including when their symptoms are mild, because intervention at that time is when it's beneficial. So if one were to think about such a platform in Riyadh, connecting its major academic center, it automatically, immediately extend the capacity and the reach of the specialists in Riyadh so that you can take care of many more patients in outlying area and in the broader regions, so that it creates a system like the matrix that sort of forms the hub, drawing patients to Riyadh where the experts are for the more complex care, allowing them to return home and be in a more local environment to get better care and still be connected to the specialists in Riyadh. So a virtual cancer center like that will create that hub. But importantly, as I try to give the example, is that in that process, you are doing two things. One, it's very exciting that virtual cancer center can very much be built on and sit on the digital health infrastructure that KSA, the kingdom, is building, particularly the effort in NEOM, building that digital twin infrastructure, capturing and aggregating data, but the virtual cancer center has become the effector arm to take that data inside and extend that to the regions. At the same time, that infrastructure allow you to capture data, to link it to this visions of building a robust biotech ecosystem. As we have heard, there are many exciting modalities to create drug, targeting almost any targets in the tumors or in the human. And there are processes that we know how to improve our decision-making to reduce the risk in the entire R&D pipeline. Being able to put the human data in that process as upfront as possible will increase the probability of success. So in other words, I'm hoping to paint a picture that with that digital infrastructure like a virtual cancer center, you can democratize expertise from your expert in Riyadh, and you can serve more patients and capture data that can link to and increase the probability of success in your biotech drug development efforts.
Yeah, I think, Linda, to add in it, having like kind of centralized like data for clinical data, for whatever created from the patient care, like genomic data and others, and to be centralized more for all of the patient, for all of the cancer patient, and then connected with the kind of satellite cancer center. It will be kind of a good model to be established here in Riyadh, to serve all of the kingdom, and similar for whatever, like in NEOM, since they are more interested in and more into population and more into well-being more than cancer research itself. So,
right.
Yeah, thank you, Linda. So I'll move to Dr. Taylor. Dr. Taylor, so within the next generation therapeutic coming out into the marketing, so how do you see de-risking research and development challenge for the novel drug modalities?
Thank you for that question. It's a great question, and I think it really kind of falls in line with what's already been kind of discussed here. And certainly by Dr. Rees earlier mentioning about the different and new modalities that are coming out, and obviously some of those are getting more and more complex. And what comes with more complex modalities is you will need more research that needs to be going, not just the modality itself, but also things like the formulation and things like these are all kind of risk factors that can impact the way a therapeutic is going to respond. And obviously what comes with that is more regulatory interrogation and more stringency around that. And obviously some, with these new modalities coming out, because some of the guidelines, I think, that should come from the regulatory authorities as well. So all of this kind of contributes to that. But there's another component to this, I think, and this is operations itself. In R&D, processes, it's important to have that multidisciplinary approach. Often, R&D tends to work together, but there's also some of this siloed thinking. And obviously, the bigger the company gets, the bigger the challenge. And so bringing together across functions would help this process. For example, sometimes people can't look at biological mechanisms, but they never think about chemistry. You're thinking about manufacturing and QCing. This is all part of that entire process, and should be brought in much earlier, because I think bringing that knowledge base to the table will help assess risk much earlier especially as these modalities become more complex. So it's really going back to your point of having a bit more of a centralized process. Another point that I also like what Linda touched upon is that this digitization of tools. You know, it is important that a lot of the data that's coming in from clinical or human patients often dictate what the models and things like that are going to be in R&D. And I think this is becoming a more important tool. And I think as well as gene editing, that's another big beast here as well, that, you know, with the advent of CRISPR a few years ago, these are tools that are becoming important, certainly in the R&D process, to be able to make earlier predictions, you know, like I said earlier on, things like being able to edit, do some gene, using CRISPR technology to modify your organoids or things like that. And there's probably a whole bunch of other tools that can be used at the same time. And so I think, you know, as you start bringing these together, I think, and it's one of the things that we as a company think about when we think about what are the tools that will help the customers, will also help them earlier on in the R&D process, in the research process, so we can reduce that attrition rate and get therapeutics earlier out onto that market. But again, that comes down to whatever that modality is and what is required and the challenges that come with it. And that's where we kind of think about these things. What can we do to help out? So, you know, I think there's a lot of challenges in R&D. It's inherent in the way that the drugs are developed. And so obviously these are things that we have to kind of address as they come in, so.
Thank you, Doctor. So I think we just have like around 4 minutes. I mean, I will let's be like more hurry up with the questions. So we'll be with for Dr. Hani. So what do you think? What about what's the opportunity and what's the opportunities? I mean, for the development of the tumor suppressor therapies here in the country.
Yeah, thanks. So I guess I have a view on this. I think the opportunities and challenges are both specific and generic infrastructural. You know, so I've kind of, I suppose I've seen it all. I have a very busy clinical practice. I've been director of an academic research center. I've worked in big pharma and biotech. I've looked at both sides of clinical trials as an investigator in an academic center and also as a drug developer in pharma and biotech. So my sense really is that what you're trying to do here is to create an ecosystem, and you should have, you can pick right across the ecosystem, big pharma, small biotech, but the consistent and ongoing infrastructural message is that you have the opportunity to not just have these people telling you about what and how, but actually to develop your own capabilities, to develop your animal facilities, your biological facilities, your genomic facilities as a sort of core that would then be, you know, a self-fulfilling prophecy, attracting more biotech into the environment. So you actually can receive and build and become a reference center for a lot of the technologies. In terms of tumor suppressors, there's a huge educational opportunity, technology transfer, and the training of young people to look at things differently. The idea of tumor suppressor therapy is so novel, it's on none of the generic lists at the moment. And this would be an example of something you could become differentiated and leading in, basically. Yeah.
So I'll move for you to do Dr. Buzayani because of the timing. So when we talk about in your company, Sanomics, I mean, talking about the application advantage and the technology and how it's possible to be implemented here in Riyadh. Can you comment on that, Doctor?
Yes, for the delivery platforms, it's really critical. I mean, the use of those nanoparticles, as you know, it's really important to transfer this kind of technology. I'll give you an example that everybody is familiar with. If we talk about, for example, the COVID-19. So when we do the whole vaccine strategy, I mean, when I did my PhD, the technology is not new. It's not really, it's like over 30 years old. I did my PhD working on mRNA to inhibit the integrase, for example, of the virus. What is new is the development of those lipid nanoparticles. So if you transfer the whole process, and if you don't have like the nanoparticles to encapsulate the mRNA, the vaccine will not work. So this is really like critical to have this kind of technology here. And I think, I mean, I was really like pleasantly surprised yesterday to hear that the strategic vision of Saudi Arabia, I mean, starting from the prince all the way to all the ministers, I mean, they have a real clear vision to engage and to invest in the knowledge. So, and this is really key and important. So now, I mean, when you have like the government that is really believing on this strategy, so I think it's going to be really easy to develop all those different technologies. What we need to do is start with the infrastructure, because this is key. The infrastructure have to be, in my view, not dependent in one company, because if you want to be a real player, you have, for example, for vaccine or therapeutic antibodies, you have to have all kinds of technologies. I mean, mRNA is good, but sometimes adenovirus give better result, attenuated virus. So, and those infrastructure are different between one technology to the other. So I think we should build the infrastructure first that will cover all these type of technologies. Then you can work with different companies, large pharma companies, startup companies, to bring and license technologies. I think with the power that Saudi Arabia have and the fact that they are leader, I mean, for the whole MENA region, you can not only partner with big pharma company, you can also compete with them. Because the big pharma company, it's true they have R&D, you know, big, but their main strategy is to look for small startup companies and try to license from them from phase 2. So if you compete at that stage, bring a lot of technologies, you know, invest on those so you can move really faster. And I'm glad to learn also that now you have the first clinical trial, you've done all the regulation. So this is really good. So what you need is really look for startup companies that are promising, bring the clinical trials, compete with those big pharma, and start having your own personalized medicine, you know, for the future.
Yeah, totally agree with you, Doctor. So we'll move like to David. David, like, I mean, with RNA therapy, I mean, so how do you, like for us in KSA, I mean, how do you see us? How we could integrate this kind of technology here in our country?
Yeah, it's a wonderful question. And if you'd asked me that 4 days ago even,
yeah,
I would have given a very simple answer. And, you know, as you know, simple answers often come from ignorance. When you actually understand something better, your answer gets more complex. And just visiting with our colleagues and learning what I've learned in this short time, the reality is so much is already here. We've talked about this, and so much of where the KSA can contribute is independent of modality, right? It's clinical trials, it's helping understand patients. There's a knowledge of diseases, both rare and common here, that the rest of the world doesn't have because of the intensity of the research here, the passion of the physicians. And so I think each point of drug development, of biotechnology development, can be enriched here through collaboration. I think when I first would have thought of that question, manufacturing is something that we don't have time to go into, but it's a real challenge. This is a modality that just a few years ago was micrograms, that a PhD student, I know some are here because I saw them asking questions, would use in cells. And now we're dosing, you know, millions of people with these drugs. And so it's an entirely different challenge. But I'm very confident that the elements are already here.
Yeah. So I'll move, like, I mean, because as I said, like, because of the time. So with Steve, I mean, like, from your opinion, how many cell and gene therapy medicine are in the clinical today? So and what the impact of are they having? So is there any like gene therapy, I mean, available today, and even, I mean, so like for the future, for how many, like, I mean, we're expecting to get?
Thank you. I mean, there are very few clinically approved gene therapy medicines today, just a relative handful. There are, however, many more in earlier phases of clinical studies. And a great example with CRISPR. So CRISPR was identified just 10 years ago, 11 years ago. There are CRISPR medicines in phase 2 clinical studies that are treating patients with sickle cell disease and beta thalassemia and essentially curing those patients. And that, I think, we will see an explosion of that application in rare diseases in the years to come. And you're reflecting on opportunities for the kingdom. There are many, many rare diseases that are prevalent within different ethnic populations around the world, that are prevalent within the Arabic population. With the genomic sequencing efforts that I know are in place within the kingdom, I think there is a huge opportunity to bring forward gene therapy medicines for diseases that are particularly prevalent within the Arabic population to help transform the treatment of these individuals. And again, touching on some of the comments that have been already made, what needs to happen to make that happen? Well, I think the foundations are in place in that the genomic sequencing is there, healthcare system is extremely high quality, the patients have been identified, the science strategies are becoming validated around the world. What needs to be put in place is joining all the bits together and then increasingly building that ability to manufacture those medicines, to identify those patients, to conduct clinical studies, to do the follow-up. But I think there is huge potential, and the same would be true for cell therapy medicines as well.
Thank you, Steve. So I think because of the time, I need to wrap up. But because before we close the session, I just want to stress on what the extension speaker talk about. They took us through different modality, different treatment approach. For us in Saudi Arabia, since we have the vision and we are planning to be kind of hub of whatever most of the pharma company at different stage. The big pharma, the startup, and the medium-sized pharma and different disease. Because what we have here, we have a good infrastructure, we have a research center, we have a clinical. From primary care to all the way to the tertiary hospital, highly specialized hospital, different centers in cancer, in cardiology, in liver transplant. We have a young population here, and when it's come to data, we have plenty of data that it could be used. To establish a new approach for different treatment, especially for monoclonal antibody treatment, for gene therapy. As you know that we are a unique population because we have high rate of consanguinity when it comes to marriage. We have certain or unique genetic makeup. When it's come to the healthy population and even for the affected patient. So I think this country is a good opportunity for whoever for the big pharma to invest here, to build a collaboration, to build a partnership, to establish by themselves, to establish a center here in the country. So I would not take it further. I would like to thank you all. Thank you, Taylor. Thank you, Linda. Thank you, Steve. Thank you, my guest here. And I hope I would like to thank the audience, and I hope you found it very useful discussion. Thank you so much.
Thank you.
Thank you. Thank you, esteemed panelists, for a great session and in-depth insights into therapeutic development and manufacturing from the past, present, to the future. Ladies and gentlemen, it's time for a lunch break. If you're joining us virtually, we expect having you back with us at 12:30 p.m. Riyadh time. We have an interesting agenda in store. Stay connected. Enjoy your break. Ladies and gentlemen, welcome back. I encourage you to keep up the enthusiasm for the second half of our agenda. An interesting one, of course. We'll now hear from Dr. Paolo Martini, Chief Scientific Officer, International Therapeutic Research Center, Moderna, USA. He will share with us an insight of a new era for rare genetic diseases. It's my pleasure to welcome onto the stage our keynote speaker, Dr. Paolo Martini. The stage is yours.
Thank you. Thank you so much. I'll come this side this time, so I'll change. It's me again. Sorry, hopefully you're not bored to see me. But I thought that today, for the first time, I think, in the kingdom, I'm going to present some data that, some clinical data actually, that we have in a clinical trial ongoing in the US, showing that we can actually use messenger RNA in a rare genetic disorder as well. So I'll go over a little bit of how we modify the messenger RNA. As I mentioned before, when we were talking about vaccine, RNA generally is recognized as a virus. And so we don't want, obviously, an immunogenic response towards our drug because we need to use it to replace genes. And so we want to be sure that the messenger RNA is modified to evade the immune system and to become a therapeutic. So let me show you first what we do at Moderna and why we think that messenger RNA has the ability really to become the next generation of medicine. Mainly because I think we can meet many unmet medical needs, meaning that we can produce many different proteins utilizing the human body as a system to create the protein. So at this point, we don't have the limitation anymore of a recombinant protein, of making soluble protein, but we can really make anything from secreted, transmembrane protein, cytosolic, mitochondria, ER protein, really everything that you want to do. So that's great. As we say, we have a high probability of success. Obviously, the vaccine seems to work very well. Now, I'm going to show you that we can, in fact, use it in other type of therapeutic applications. We can accelerate the research and development timelines. As you've seen, we were able really to produce a vaccine in 42 days from the concept to the clinic, and we can do potentially the same, although with a little bit of more careful study, since we are talking about really fragile patients with rare genetic disorder. And obviously, as we talked this morning, we can actually have a great manufacturing efficiency. So we can produce the drug, we can support clinical trial, and more importantly, we can support post-trial, the marketing right away. So, as I said, it is a sort of a revolutionary technology, if you will, because at this point, what was the limitation in the past of making certain type of protein, now it gives us really the opportunity to make every type of proteins. So any type of replacement now is not a stop anymore, but it becomes really a reality to replace any type of defective proteins in any monogenic or multigenic disorder. So at Moderna, obviously, because we think that this is a platform, we wanted to understand every aspect of our drug. And it's important to know that, because the moment that you produce a drug, you need to be sure that not only you can make it, you can actually make it suitable for human body, but more importantly, then you can manufacture it. And there are certain success criteria that we define initially in terms of what the drug would look like if it had to be a therapeutic rather than just a very immunogenic RNA for vaccine purposes. So obviously, one of the key features of the messenger RNA, compared, for example, to gene therapy, is that you have a predictable dose response. What does it mean? That X amount of messenger RNA that you give to the body, you always get X amount of protein. If you double the amount of messenger RNA, you double the amount of protein, and it's reproducible. So that gives you a sense of what is the quantity and the amount of drug that potentially you're going to give to a patient, and that's very important. And the other thing that is very important is that it has a reproducible pharmacology. And so I'll show you, obviously, our clinical data where we actually treated patients and they are under chronic treatment, and we see a reproducible pharmacology every time we give a messenger RNA. We do actually, we can achieve a therapeutic potency. Every time we develop a drug, we look at the animal model, we look at therapeutic index, and the therapeutic index is going to tell us if we are going to make enough protein to be able actually to restore the physiology of the body and potentially to change the phenotype of the disease. And obviously, more importantly, we have safety and tolerability. I'll show you some data that, in fact, shows that patients can tolerate in a very safe way chronic doses of messenger RNA. Obviously, as I mentioned this morning, knowing and having the ability to scale up the process, it gives us the opportunity eventually not only to support clinical trial, but as you know, in a rare genetic disorder, is unethical, particularly if you have a drug that is working in a patient, is unethical to stop. So we need to be prepared to have enough drug to support post-trial and during trial all the patients that they are responding to the drug. And as I said, because we have a highly reproducible manufacturing process, it gives us the opportunity to get a great quality control of the drug. And so we engineer every aspect of the messenger RNA. We don't change the protein, so the protein that eventually is made by our own body is going to be the wild type. In some instances, yes, we do engineer the protein when, for example, we want to extend the half-life of the protein. But in reality, the protein that we make is exactly as it's supposed to be. But the messenger RNA obviously needs some modification because even in my early stage as a graduate student, this was 30 years ago, we had the biggest fear of using messenger RNA because we knew that was very unstable, and then any nucleases could break it down, and sometimes you would look at your gel and that you had nothing. And so we always thought that messenger RNA was basically unreachable as a potential drug. But now we actually have done enough studies that they give us the opportunity to understand the stability, longevity of the messenger RNA. We can manipulate it, and more importantly, we can also, in some instances, increase the half-life of the messenger RNA as well as of the protein, as I mentioned before. And more importantly, we can actually work on the immunogenic response of the messenger RNA. And that's very important, particularly if you think that this is going to be a chronic, obviously, therapy. Is not going to be gene therapy. Messenger RNA has a short half-life, even though when we can extend it, we go up to 72, 96 hours, but no more than that. So obviously you don't have the benefit of the longevity of a gene therapy, but you do actually have the opportunity to have a very safe drug because the moment that you have some issue, you stop, and then the RNA disappear as well as the protein. So in that sense, it's actually a valuable drug. On top of the fact that you actually can reproduce the pulsatility of the human physiology by giving the drug at the right time. Our body doesn't work with always a huge amount of protein made constantly, right? There's always a peak and a trough. And so messenger RNA actually give the ability to sort of reproduce that type of physiology that we have in our body in a very natural way. So, the messenger RNA that we make looks exactly what you would expect to find in the human body. It's capped as a 5' UTR. the open reading frame that will encode for the requested protein, 3' UTR and a poly-A tail. Where we do modification actually is that the 5' UTR and 3' UTR, these are standardized because they give us the opportunity really to increase the ability of translations of the messenger RNA into a protein. And one of the things that we have learned in studying messenger RNA is that in some region, certain regions of the messenger RNA are very important, for example, to a better translation, to the point that if you take the endogenous messenger RNA versus ours messenger RNA, the amount of protein that you make versus the one that we give is actually 3 to 4 times higher, sometimes even 5 times higher than the endogenous one. So you can really capitalize on that. And also we can make it a tissue specific because sometimes in the 3' UTR we have the ability to encode microRNA recognition sites. We know that microRNA have the ability really to recycle messenger RNA. So in some organs we have a very specific microRNA. So for example, if we want the messenger RNA not to translate a protein in the spleen, what we'll do, we'll actually encode a microRNA recognition sites that are going to recognize microRNA that are very specific for the spleen. So the moment that the messenger RNA gets delivered to the spleen, gets recognized with a high affinity by the microRNA and goes into a degradation path. Therefore, we don't have any protein in that particular organ. So we can make a tissue specific in that sense, utilizing once again our own body to sort of process it in the right way. And that's also the most important things, as I mentioned. Unfortunately, RNA is recognized as a virus, right? And so in order to evade the mechanism of recognition of the messenger RNA as a virus, like binding to the toll-like receptor 7 and 8, or other system where you have a double-stranded RNA, and that's why, as I mentioned early in the morning when we were talking about vaccine, it's important to have a fully made RNA to avoid the small pieces of RNA that can actually create double-stranded RNA that eventually would trigger the RIGI system and would trigger an interferon response as well as cytokines with then an immune response from the immune system. So it's important that we do modification of the messenger RNA to understand how we can evade the immune system. And what we have found, in fact, that the difference between DNA and RNA is in one single nucleotide, which is the uridine versus thymidine. While DNA is not immunogenic, RNA it is, and so therefore the culprit must be the uridine. And so what we did, we looked at the synthesis, at the natural synthesis in the body of the uridine, and so we found that either a methyluridine or a methoxyuridine in this position 5, if they contain their little variation of the original uridine, that has the ability really to create a steric hindrance. Therefore, the uridine won't be able to bind to the toll-like receptor 7 and 8, and therefore trigger an immune response. And that's why we can create now, by just a simple modification, a messenger RNA that is evading the immune system. More importantly, once you do the changes in the messenger RNA, you can imagine that the secondary structure, which is very important for translation, is going to be affected, right? And so what we want to be sure that in our system, not only we use enzyme that the ability to recognize this uridine and therefore create a fully integrated sequence, but more importantly, we want to be sure that we create the right secondary structure that has the ability to increase the affinity to ribosome. Therefore, we can get a much better translation of the desired protein. And by doing all of these with a special algorithm, we actually found out that sometimes we have so much more protein made to the point that we can even treat now autosomal dominant disorder, meaning that even if we had an autosomal, if you have a dominant negative protein produce, the amount of our protein is going to be so high that the stoichiometry between the dominant negative and the wild type is going to be overcome by the amount of the wild type. And that's why we can also treat autosomal dominant disorder. So obviously the messenger RNA, if it was delivered as a naked, it would be once again degraded, as I said, is very liable of any type of nucleases. So obviously what we did, we wanted to protect with lipid nanoparticle, also because lipid nanoparticle have the ability to deliver RNA in specific organs. Our lipid nanoparticle looks very much like a spheroid that, let's see, yeah, you don't see very well, but it looks like these spheroids, and they're composed by ionizable and also hydrolyzable aminolipids, cholesterol, obviously phosphatidylcholine or phospholipids that are very typical of a cell membrane. And also we use a PEG attached to the lipid nanoparticle. And that's only for the stability of the lipid nanoparticle. So when we go in front of FDA and we show the quality control, the FDA see these beautiful spheroids, very much consistent in size and in shape, that they think that we have a high quality control. In reality, the lipid could be a problem because the PEG lipid could be a problem because every day we brush our teeth, we use cream, and all of these elements trigger IgG antibody in our body. So you can imagine that if you have this PEG lipid attached to the lipid nanoparticle, we would have all this IgG right away from the system attacking our lipid nanoparticle, preventing the delivery of the messenger RNA to the receptor. So what we did, actually, once again, we created these hydrolyzable PEG lipid. So the moment that the lipid nanoparticles are injected in the system, the PEG automatically shed because they are hydrolyzable. The blood is aqueous in substance. Therefore, they shed completely from the lipid nanoparticle, releasing the lipid nanoparticle and preventing the IgG to bind to them and blocking their ability to then to bind to cell membrane receptor to be internalized and to create the endosome, etc., etc. And so this is the way, once again, to use our body to capitalize on the chemistry of our body to deliver the lipid nanoparticle in a smart way without actually any chemical or extra modifications. And so that's why in our drugs, and now I can say in our therapeutics, because I'll show you the data after, and I'm very proud of that because it's actually the first time that I present this data in the kingdom, but it's incredible that every component are biodegradable, including our lipids. So our lipid, as I said, the one that they constitute really the spheroids, are hydrolyzable as well. So the moment that they get released into the cytosol post-endosomal escape, they are degraded between 8 to 24 hours. So in diseases, for example, in rare genetic disorder where we have steatosis or accumulation of the lipids, you won't see accumulation of our lipids in already a steatotic liver. And there, obviously, it's important because we don't want to exacerbate potentially other type of diseases. We have, unfortunately, I don't have it with me today, but we have an example in glycogen storage disorder type 1A, and we've done studies in the mouse, and the mouse has a very steatotic liver. The moment that you transfer the protein with lipid nanoparticle, the liver deployed fast the triglyceride from the steatosis, and so the liver become like a bloody and plush and like a normal liver within 24 hours without any accumulations of triglyceride or any lipid. It's actually beautiful. And once again, we are utilizing our body to ask them to do what we want without any particular chemical escamotage to get there. So, and this is what it looks like once we give the lipid nanoparticle in the system. Obviously, as I said, there's a dissociation of the pegs. Then, obviously, by exocytosis, they have the ability to bind to the receptor by opsonizing with ApoE and binding to the LDL receptor. And that's why the first generation lipid that we have is very suitable for liver metabolic disorder, because there's a tropism for hepatocytes, right? And so, therefore, then the lipid gets internalized. Once the pH of the endosome start to drop, being also ionizable, you can imagine that they have the ability to escape the endosome and release the messenger RNA that then is getting recognized by the ribosome and producing the basically required protein. And this is a system that we don't push, so it's occurring very naturally, and it gives us the ability really to get to any type of protein translocated in any type of organelles without any limitation. And that's why we think that this is the next generation sort of therapy for the moment. Until obviously gene therapy will resolve the issues of, for example, multiplexing messenger RNA. We can put as many messenger RNA in a lipid nanoparticle as we want, and I'll show you an example for propionic acidemia where we add two messenger RNA to reconstitute an hexadodecamer that is very important to metabolize amino acid and protein. And so at Moderna, we have our own proprietary lipid nanoparticle that they give us the ability really to deliver to with different routes to different organs. We have a collaboration, for example, with Vertex Pharmaceutical, where we do inhaled aerosolization of the lipid nanoparticle. Obviously, we do intratumoral injection with certain lipid. You know that we do sub-Q and intramuscular injection, and these lipids are very different. So we have different lipids for different needs. Obviously, immunogenicity is required for a vaccine, and that's why we want to exacerbate that sort of immune response. Certainly not for obviously regenerative disorder or for other type of diseases where we want to tame, in fact, that type of reactivity. And so we can do also intracardial and intratumoral, and obviously IV injection. We have actually right now two different lipids in the clinic that are tested. The lipid that we are testing for propionic acidemia and methylmalonic acidemia are different from the lipid that we are testing for example, for glycogen storage disorder. And the reason is that we wanted to understand as much as possible this lipid nanoparticle, have multiple options and multiple shots on goal in order to understand how well we can actually deliver and be efficacious with our therapy. And so with this, obviously, you can imagine that we have a very rich pipeline at Moderna that covers, obviously, as you know, prophylactic vaccine, personalized cancer vaccine, intratumoral immune oncology, localized regenerative therapy. Obviously, we have secreted protein, as well as inhaled pulmonary, and then systemic intracellular protein. So we can really do it all. And we are trying hard to understand how we can deliver to different organs. It's not easy by any means. We haven't resolved the CNS, we haven't resolved the other organs. We'll get there eventually, but so far we have been able at least to enter the clinic also for regenerative disorder and oncology as well. So what I want to do today is for the first time to present some clinical data, and I think we are the only mRNA company to date to have actually clinical data in human looking at a rare genetic disorder like propionic acidemia, which, by the way, is highly prevalent here in the kingdom. And that's why yesterday we signed an agreement with the Kmark to be able to collaborate and to actually include the patients in our phase 3 trial for the use of the propionic acidemia messenger RNA. What is beautiful just for the one that do not, there's a high prevalence of this disease in Saudi Arabia. It's caused by mutation either in the propionylCoA carboxyl-CoA enzyme or the propionylcarboxyl CoB subunits, the two of them generally come together to create an hexadodecamer. So patients that they have mutation either in one of the subunits or the other subunit, actually they have a defect enzyme. And as a result of that, they cannot process either protein or amino acid or anything that will give them the opportunity really to have a normal life. As a matter of fact, many of these patients have a very short lifespan. The majority of them, they die by age 6 with severe complication, mental retardation, seizure, high level of ammonia circulating in their blood, and you can imagine that it is not a good quality of life. Some mild mutation allow some patients to live longer, but also with a very limited quality of life. Right now, the only therapy available not only is diet, which is horrible because these patients cannot take any protein or amino acids, so it means that the only things that they can eat are carbohydrate and fat. That's all, which is horrible. But, and the other option is obviously to go into liver transplantation, which still has some complication, particularly, unfortunately, it doesn't offer the longevity that we would like to see when you do transplant because of the heavy toxicological profile of some of the immune suppressants that are used in order to integrate the new organ in the system. And so we think that this therapy potentially could be a breakthrough in avoiding potentially liver transplantation and potentially liberalizing the diet for these patients. And so this is the enzyme. That's what the enzyme looks like. It's an hexadodecamer. Has the ability really, it's a critical enzyme because it's a mitochondrial enzyme that has the ability really to process propionylCoA to methylmalonyl-CoA, which is eventually is processed to succinyl-CoA. Succinyl-CoA is one of the key substrates for the Krebs cycle, which is one of the key respiratory system of our cells. Our cells survive because of the Krebs cycle. So you can imagine that by the time you have a disruption of this enzyme, you're going to have a very critical symptoms and very important effect on these patients. Obviously, these patients obviously suffer from metabolic decompensation. They have a high level of ammonia, which has created brain damage, and they create also propionic acid, which is very harmful to the body. So we want to be sure that all of these gets resolved with our therapy. So this is what our therapy looks like. Slightly different from what you expect. We combine, in fact, the two messenger RNA encoding for PCCA and PCCB. Once they are translated inside the cells, they actually are translocated because we don't change anything from the protein. The protein has its own element, so therefore is going to be translocated naturally to the mitochondria because it has a leader peptide that will transfer the protein to the mitochondria. So once it's transferred to the mitochondria, then it's going to produce this big hexadodecamer that then has the ability at this point to process protein and amino acid correctly. So we did a study, and this was actually inspired by one of the investigators here, actually, few investigators here that collaborated. One is right here, Dr. Isar, and then Dr. Majid Al-Fadel. They did a study looking at carglumic acid and the ability of carglumic acid to drop the level of ammonia in patients. So we wanted to use that as sort of as a benchmark to understand if our therapy was in fact reducing the level of carglumic acid, so the level of ammonia in patients, potentially similar, or in mice in this case, similarly to the carglumic acid. And as you can see here, in fact, in the orange bar is carglumic acid dropping the level of ammonia in mice, and in blue is our messenger RNA therapy that obviously dropped the level of ammonia in the mice. But more importantly, what was beautiful of this, that was actually reducing the critical biomarker, the critical toxic substrate that we found, like propionic acid, 2-methyl citrate, also the C3, C2 propionyl carnitine ratio. So everything was actually starting to look like a real therapy. And one of the important things also that we noticed, not only that in the mice we could do basically multi-dose and dosing, in this case, every 3 weeks, and we could actually have a reduction of substrate. This is just one of them. If you want to see the whole panel, it's actually published in Nature Communications. It's a beautiful paper that shows, in fact, the ability of the combination of these two messenger RNA to really have an impact on the biomarker, the critical biomarker on the disease. One of the things that I didn't mention at the beginning when I described propionic acidemia was that this patient also developed cardiomyopathy, as some of them also renal disease. So we wanted to look at the heart, particularly in the old mice, because the old mice, although it's a model and is not probably the best model to reproduce propionic acidemia, they actually develop an enlarged heart. And what we have seen, basically, that if you keep treating the mice, and this was a long-term treatment for up to 6 months, we actually started to see a reduction in the heart weight, suggesting that not only we have a benefit at the level of the biomarkers, but we're actually having an impact also on the potential cause of mortality, which also could be cardiac arrest or cardiomyopathy because of this therapy. And so with this data, we actually developed the leading candidate, and we decided obviously to go to the clinic. And that's where now I'll be able to show you, and I'm very proud of this because this is really the first data ever generated in a rare genetic disorder, and now we have clinical data that suggests that we might have an impact in human with this combination. So just to summarize what we saw from the mice and that give us the opportunity really to go into the clinic, we actually were able to identify lead candidate, which was a combination of PCCA and PCCB. One of the things that is important, when you have a mutation in one of the subunits, you can imagine these two subunits are important to come together to create the hexadodecamer. If you have a mutation of one, and if you have a reduction of one of the two subunits, also the others start to disappear. So you cannot really replace only one or the other, because it might take time by replacing only one for the system to start to reproduce the other one in a timely fashion. So it's important that in some instances, in certain diseases, we do have the opportunity to give them both so that we can restore right away the physiology of the activity of this enzyme. And so what we saw also, we had a long duration of action. That complex, that hexadodecamer, when it comes together, it stays in the body for about 21 days in the body, in the mice for 21 days. So you can imagine that this could give us the opportunity really to have a nice regimen for treatment. I'll show you that in reality, in the human is a little shorter. Still, with the high unmet need that we have in this disease, an infusion every 2 weeks for these patients, I think, is more than tolerable, I think, given the quality of life that they have. But more importantly, what we saw a significant improvement in ammonia level comparable to carglumic acid. We saw a reduction of serum biomarker, and more importantly, we saw an impact on the heart weight. And interestingly, you would expect that like enzyme replacement therapy, you know, when you give enzyme replacement therapy, for example, for Fabry, for Gaucher disease, for Hunter, or more importantly for Pompe disease, patients start to develop anti-drug antibodies, right? And this is a problem because then you have to increase the level of protein in order to overcome the amount of antibodies. By giving messenger RNA, what we have found that we had a reproducible pharmacology throughout the dosing, meaning that the protein was starting to be recognized by the liver as its own. And so we didn't develop anti-drug antibody. Even after 6 months of treatment in mice, they kept responding to the protein. And we've seen in some gene therapy studies, for example, from Duke University on Pompe disease, where GAA is a highly immunogenic protein. And yet, when you have a high production of the protein coming from the liver, even though delivered by gene therapy, you don't see anti-drug antibody happening. And so we were very happy to see that we can actually reproduce the pharmacology of our drug. And that's why we decided to go into a clinical trial. The trial started, unfortunately, we were ready to start just a couple of months before the pandemic. We started to recruit the first patient from Boston University, and the pandemic happened. And you cannot jeopardize the life of these patients. So we stopped all the clinical trial, including the one on methylmalonic acidemia, mainly because we didn't want to put in jeopardy our patients for potentially being exposed to COVID, given that the hospital were, in fact, crowded with, unfortunately, with dying patients at the time. So we waited until 2021 when it was safe because of vaccination from Moderna. Just kidding. But we were able to actually get these patients finally into the clinical trial. And so now what I'm showing here, this is actually a little obsolete slides. We presented these slides at the R&D day back in September in the US. We are actually fully enrolled cohort 4, and we are finishing cohort 5. Cohort 4 is 0.6 milligram per kilogram. Cohort 5 is actually 0.9 milligram per kilogram, and we are pushing to the next cohort because so far what we have seen is that we have safety and tolerability of the lipid nanoparticle. So far, and this is once again, is obsolete, this is the data that we presented. I cannot give you yet the new data because it needs to be obviously vetted and be verified. But so far, we had 10 participants that have been dosed, 3 participants that have been for more than 1 year dosed with the drug, so they mean that they receive every 2 weeks an infusion and they're still receiving it, right? And we're responding to it. And we did basically, at that time, over 120 intravenous doses administered, so you can imagine now that they are more than that since we are a cohort 5. And so far, all the participants that are eligible have decided to continue eventually with the open label studies. And sometimes, you have discontinuation, right? Because patients don't tolerate the drug, safety profile is difficult. In this case, so far, every participant has decided to continue with the therapy, and they will be moving into the phase 3 open label study. And so we have, for the moment, no dose-limiting toxicity, no drug-related serious adverse event. We actually sort of adjust the protocol, ensure that we didn't trigger an infusion reaction in patients. As I said, no discontinuation due to safety, and the only drug-related events were mild to moderate and due to infusion-related reaction, which is less than 10% of the doses. And these are mild infusion, meaning that reaction, you have a little bit of fever, you have a little bit of discomfort. We don't even pre-drug these patients. They just get the therapy. There's no pre-drug, there's no immune suppressant, there's no Tylenol, there's no antihistamine, nothing like that, right? So you can imagine that this is actually quite safe and well tolerated. But more importantly, what we started to see is that the first dose, as you can see, is 0.3 milligram per kilogram, which, by the way, milligram of propionyl carboxyl-CoA and 0.15 milligram of propionyl carboxyl-CoB. And that's what they make up to the 0.3 milligram per kilogram. And we did the study the stoichiometry has to be 50-50 in order to get to this level. But as you can see here, there was a lot of variation in the patients. Now, once we started to increase the dose, that actually, and obviously we are implementing these as more doses are coming in and more patients are coming in, but you start to see that at 0.45 milligram per kilogram, the biomarkers start really to decrease to the point that we have about a 34% reduction in the key biomarker. I'm actually curious to see the next dose, which is 0.6, which is double of these, which will mean 0.3 of one and 0.3 of the other, because I think that will give us a much better sort of outcome in terms of reducing biomarker. And so the other things that we discussed with FDA, what is the best clinical endpoint to have and to be sure that not only we have efficacy by biomarker, but we also see an improvement. And metabolic decompensation was actually discussed with FDA and MHRA, mainly because metabolic decompensation has obviously increased acidosis in the blood. When you have acidosis, you have a high anion gap, high production of propionic acid. You have acute hyperammonemia. And so these patients generally get hospitalized and they have to go to the ER in order to reduce all these biomarkers. And it takes about a week with a severe diet, with severe monitoring these patients and trying to reduce this level. So obviously the healthcare agency now, they are inclined to use metabolic decompensation as a clinical endpoint because also prevents basically hospitalization of these patients, particularly in the ER setting. But one of the things that we saw, and that's why metabolic decompensation becomes important, and even if in the early doses, so these are the patients that we chose, that they had obviously different level of metabolic decompensation before being treated. Now, when they are in treatment, you start to see that we reduce by 48% the metabolic decompensation events in these patients. When we talk to many physicians all over the world, they say that a successful therapy for these patients will be at least a reduction of 20% of metabolic decompensation. We are at 48% reduction, right? So I'm hoping, and keep your finger crossed, that we continue to see that, particularly with the higher doses, because that will be phenomenal for this patient. It would mean, in fact, a change in quality of life, and not only for the patients, but also for the family. You can imagine that the burden on the family, particularly of these children, being on a monthly basis sometimes, particularly here in the Kingdom, in my conversation with many physicians, they are hospitalized on a monthly basis, and this is not fun. No, it's fun for the family, not fun for the kids. They're intubated. Every time I come and visit, I leave in tears because you see this little child with tubes going everywhere, seizing sometimes, is just devastating, right? So starting to see some impact on the disease, I think, is going to be transformational for these patients. So I'm going to be done and giving you a summary with hope that in fact we continue to see these encouraging data, because if this is the case, I think we can go to accelerated approval once we have defined the dose, and in the meantime continue the open label studies for safety and efficacy, but more importantly, what we have seen from so far from the studies, which is still small. Right now, I'm presenting data from 10 patients. We are at 15, so it might get more robust. And I would love, in fact, to include patients from the Kingdom, given that they have a more homogeneous background, so it will give us a better sense of the responsiveness to the efficacy of the drug. But as you can see, obviously, we met every endpoint that we were looking for with encouraging trend in biomarkers and clinical endpoints. So we are continuing to enroll, and more importantly, we are hoping that the patients will be part of the open label studies, the phase 3 open label studies, because this will mean an important milestone for this type of organic acidemia. So with this, I want to thank all my colleagues at Moderna. As you can imagine, from the concept to manufacturing to lipid nanoparticle to everything that we have done, many people were involved. It's impossible to list them all. This actually was a project that was exclusively carried out at Moderna. So we had the animal model at Moderna. We did it all ourselves. And I'm so proud to see that finally we can get some clinical data. I know that I have a couple of minutes. If people have questions, I know that I'm looking at the MC and she's like, I have 2 minutes, 5 minutes. If you have any question, please ask. Otherwise, I'll leave the stage happy. Thank you. Yes. Yes, please. The only thing is that I cannot hear you well.
My name is Charles from Germany. So great talk. One question, if you are dosing that often, why don't you use the circular RNA?
The
circular?
We are, for the moment, we are going old-fashioned, right? Mainly because there are so many new parameters here, right? Messenger RNA, lipid nanoparticle, infusion, everything that is completely new. So that's why we started with a regular infusion, with a regular pump. We just modified the timing, etc. The future is going to be different. We are also looking at a different route of administration that will be more suitable for patients potentially in the future. But for the moment, given the severity of the disease, we are going with the classical way. Also because when you start to change too many parameters, it's going to be problematic to see if you have any issues. The future is going to be different because Moderna is working on many different aspects of delivering and devices, etc., etc. So,
you are developing the circular RNA in other projects. I know that by fact, but the point is here, as you are inducing this expression through this linear RNA, I was wondering, why don't you just transfer this technology that you have already established in other projects, in these projects where the expression will be longer, and for these patients, it will be much more beneficial?
It's possible, but you have to be very careful, right? As I said, this system work in a physiological manner, right? So sometimes I know that longer is better, and potentially it is a better way. But as we have seen in gene therapy, right, particularly for some of the rare diseases, where you have a production of protein that stays constantly in the system, you don't have extra beneficial effect. And the reason is that our body doesn't work that way. You saturate receptor, you are saturating, in this case it's an enzyme substrate that goes together. The moment that you don't have any more substrate, the enzymes start to degrade. And then you start to rebuild the substrate, so then the enzymes start to get back. So understanding the physiology of the disease and the physiology of the body is very important, and that's why gene therapy sometimes is not the solution. I know that people want the long term, but understanding how to modulate the biofeedback that we have, it's critical in order to actually have a continuous efficacy. I think I'm running out of time at this point. Sorry. Thank you so much. I appreciate it. Thank you.
You're having a great productive day, Dr. Paolo. Thank you so much for your great informative presentation. It seems like we're ushering a new era of treatment for rare genetic diseases. Yes. Medical devices and mid-tech development and manufacturing, the title of our next panel discussion. To start, please join me in welcoming the chair of the session, Ms. Bushra bin Saud, Danaher Corporation, UAE, who's joining us virtually. Ms. Bushra, can you hear us?
I hear you. Not sure whether you hear me.
Yes. Okay. Wonderful. And we'd like to welcome her panelists, Ms. Monica Rasai, Welcome to the stage, Dr. Matteo Giborg and Dr. Hisham Feniri and Dr. Biril Opperheim. Welcome all to the Riyadh Global Medical Biotechnology Summit. I'll pass the mic to you, Ms. Bushra.
Thank you very much.
You're welcome.
And good day to our esteemed audience. Welcome to the Riyadh Medical Biotech Global Summit. It's really an honor to be here and sharing this session. We will focus today on the medical technology and diagnostics. And just for the intro, my name is Bushra bin Saoud, and I am the regional president for Danaher Corporation in the Middle East, Africa, and Turkey. And I will just start with giving you some context. I hope the slides are on at the moment. Just to explain, give you a bit of information about who we are, what is Danaher, and what we aim at doing. Danaher is a corporation that is a global and science and technology innovator. We are committed to improving quality of life. I think you can move to the next slide. And among that, we have at least 20 operating companies that are doing their best to do that and to improve quality of life in the world with one single purpose, shared purpose, that is help realize life potential. So for us, we want to drive meaningful innovation and in some of today's most dynamic industries, very fast growing. I can give you an example. We have been at the forefront of the COVID fight. We were a key player in producing one of the most reliable COVID tests, among the first to get FDA approval. And at the same time, we are also playing in the field of vaccine manufacturing. And I think this morning there was a session and we also contributed to that. Now, importantly, you can see in the next slide that Danaher is somehow, half of the footprint is life sciences, where we want to make really leading edge scientific research that make possible, this kind of scientific research. And also our diagnostic solutions and workflows somehow are, also like 1/3 of our footprint. So those tools and workflows are helping us to inform and treat accurately millions of patients. So today you will see we will have 3 operating companies that will hopefully share with us their pipeline of innovation, what's the mission they want to have, and also bringing that to what can we do in Saudi Arabia and what can we improve. At the same time, as part of our speakers, we have Dr. Feniri. He's online from Morocco, and he will also help us understand how, as a leader in the academia world, do we look at diagnostics, biotech, in the other part of the MENA region, to see also his perspective and how we can learn from that. So moving on, today you can see our diagnostics somehow business is beyond 10 billion globally, and the key operating companies in this business are present with you, and that are Beckman Coulter Diagnostics, we have Leica Biosystems, and Cepheid. Those are the leaders that are today sitting and will take us through their own views and perspectives. So again, welcome to the speakers. We would have wished to be with you face to face, but at least let me start introducing our first speaker, Mrs. Monica Rasai. Monica is our Vice President of Marketing and Strategic Initiatives. She covers what we call high growth market for Beckman Coulter Diagnostics. And in fact, she has more than 25 years of leadership roles in the medical device industry across US, Europe, Asia. And she's particularly interested into the research and development along with strategic marketing for Beckman Coulter Diagnostics. So without further ado, I will invite Monica to share with us her views on how Beckman Coulter is reimagining healthcare in the future, and how her personal views on this topic. Monica, please be welcome, and the floor is yours.
Good afternoon. It's a pleasure to be here, and I have the honor of speaking about Beckman Coulter and who we are. We are one of the Danaher diagnostic companies, and we have a very strong mission of relentlessly reimagining healthcare one diagnosis at a time. We operate in a global base, and we have about 11,000 passionate employees that are working with us across the world. We serve our healthcare community, our customer, and our patient by providing 1 million critical tests at any given hour, and this is the responsibility we do not take lightly. At Beckman Coulter, we do have about 80 years of legacy. We stand on the giant shoulders, Dr. Beckman and Coulter brothers, that most probably you all have heard about them, and how they really created solutions that mattered. Today, we have this noble purpose of wanting to make the world a better place with our solution offering and upscaling our laboratories we serve. So let me elaborate a little more on that one. During the last few years, we have launched many game changers in the healthcare market. To call a few, you can see it also in the screen, many macro panels, many instruments, many assays, many automation and clinical informatics. However, where I want to bring your attention to is how we are pivoting from an engineering culture to a clinical outcome culture. That mission of relentlessly reimagining healthcare is extremely close to our heart, and I would like to explain what each words mean to us. Relentlessly, the very first one, speak about our Danaher rigor. DBS, or Danaher Business System, which is our secret sauce, inspire us to always, at any given moment, look at opportunity for continuous improvement. We believe in order to impact patient pathway for better, we need to go to places we have never been before. Reimagining healthcare speaks about the fact that we want to be forward-thinking. We want to come up with solutions that move the needles, that address the community challenges. And last and not least is one diagnosis at a time. That is about you, I, our mothers, our fathers, our spouses. That means in every single drop of blood, every tube that we get, there is a life there. There is a person that is waiting for an answer, and we want to remove the burden of waiting and worrying and replacing it with the knowledge and insight to act now. You might think the pictures on the screen are random pictures. They're not. One is a father I lost 30 years ago to heart attack, and that's why I'm here and why I serve the healthcare community. Thank you. The other one is the mother, a cancer survivor 2 times. And inshallah, I would be able to provide her the best life she can have with the best diagnosis she can get. This mission is very important, not just to me with what I shared with you, but also with many of our associates, from president level to everyone in our manufacturing level that feel that connection with what we do. We have been launching game changer product, but what is the most important when it comes to lab is we want to make every single lab out there the most efficient they can be, by providing them with a solution that addresses their toughest problem. Because of that, we ask ourselves the why questions, like why should the power of automation only be limited to large size or medium size laboratory. We are in it to make sure any size laboratory has that automation line and can reach that productivity as we go through. Another one is, what if we would be able to eliminate daily maintenance so our laboratorian can focus on what matters the most and help the physician to make the best decision for the patient by the information and insight that they provide. So that is the ultimate goal, to serve the laboratories and bring them to the excellence from operation level. But what about the patient? The patient that we all feel very connected with, all of us. The way that we go about addressing it is very much about patient care pathway and the clinical outcome. We look at the community health challenges and we ask ourselves, what if multi-inflammatory syndrome in children could be caught way in advance? What about AMI? What about sepsis? What about cardiacs? What if a physician would be able to get more insight from that tube of the blood, which is matched up with the health information of that patient, and provide antibiotic solution 2 days in advance? You all can imagine that every minute counts. So these are the ways that we are looking into revolutionizing this space. If you look at our industry, there are so many things are happening, and we are living in a great time and exponential time. Digitalization of healthcare, personalization of healthcare, many other trends that are coming through. But if you look at it, in many cases, they are episodic. What if we would be able to provide a solution that is proactive, that provide insight about not just now, it's what about is going to happen next. For the Kingdom of Saudi, first, your vision is so inspiring to us. Amazing to see all the great work that has been done. We have been looking at the community health challenges, and we believe with our solution, we can provide solution that matters to you and address the community health challenges. First, by providing and investing in the local talent here and also the supply chain. Second, by making sure automation for all is a reality, and we are providing automation and clinical informatics solution to upscale all the labs. And last and not least, by providing a menu that matters, that address the community health challenges. Please join us. Be our partner to relentlessly reimagine healthcare, one diagnosis at a time. Thank you.
Thank you, Monica. Thanks for a great and inspiring presentation. And I think now it's time to move to our second speaker. I will introduce Mr. Mathieu Guy-Bourget, Senior Commercial Director, EMEA for Leica Biosystem, another Danaher company. I'd like to say a few words around Mathieu. He's leading the Digital Pathology and Workflow Organization, supporting digital transformation from workflow improvement to whole slide image management. And Mathieu has more than 20 years in the medtech industry in North America and Europe, where he's based. Welcome again, Mathieu, and please, the floor is yours.
Thank you, Bushra. Good afternoon, everybody, and thank you very much for the invitation. Yesterday, Dr. Ronald DiPino from MD Anderson talked about the way of how to improve clinical innovations and how it is important to mitigate risk when you talk about preclinical trials. Today, I want to talk about practical example if you do cancer innovation, working in the pathology environment, whether you're working on companion diagnostic, immunotherapy, or artificial intelligence. So Leica first. Leica is the global leader in pathology environment. We are an organization of 150 years who's been providing everywhere in the world all instruments, software, and consumable for pathology labs to be able to run. As we have today quite a diverse audience, I just want to remind that a pathology lab, they transform tissue tumor material so that a pathologist can do a diagnostic and research can be executed. We have around 4,000 associates in the world, all of them working to ensure that we provide all the best support for the laboratory so that the pathologists can provide to all of us the best diagnostic for the cancer. What is about digital pathology? So digital pathology is simple by principle. It's the fact that you scan a glass slide with a scanner. This is the latest one we recently launched in 2021 called the GT450DX. It's already the most acclaimed in the market, reinforcing the fact that we are still and we will continue to be the global leader in digital pathology with the largest install base of scanners everywhere in the world. We are proud to have multiple customers and reference, whether in Europe, Middle East and Africa, such as the Curie Institute, Charité in Berlin, the TÜV in Munich. We also work with the whole hospitals of the government in Kuwait. And in North America, we work also with Sloan Kettering, MD Anderson, and we are also very proud to have allowed the Mayo Clinic group to digitize all their activity, which needed 45 scanners. So why digital pathology is critical? Unfortunately, there is a global shortage of pathologists and the trend is not good. So it's very difficult to find new pathologists available. And on top of that, because cancer is complex, you need subspecialties. So for instance, neuropathologists. And unfortunately, at a local level, in your city, it might be challenging to have the right pathologist in front of the right glass slide. The other element is the rising of digital pathology and artificial intelligence. Artificial intelligence in pathology finally arrived. There's already some good successes. Tools which allow to reduce the workload for the pathologist, some tedious tasks relative to counting cells, for instance, on a glass slide. Some algorithms now are able, and some clinical approved, to provide second opinion for the pathologist to define on very complex cases. And even now, you have new generation of algorithms which are even challenging other technologies such as molecular diagnostic. However, deploying efficient digital pathology is not that simple. So I'll give you an example. You want to do a clinical development to develop an artificial intelligence on breast cancer. This is an example on the slide. You want to do a multicentric study, and you're going to work in a supervised environment. So you're going to ask pathologists to work on different slides from different labs so that we establish the ground truth to educate the algorithm. Unfortunately, when we're going to do that work, there's going to be challenge to reach concordance. There's going to be challenge for pathologists when they look at the same glass slide to provide the same diagnostic, the same annotations. Why is that? Because in reality, pathology labs are less automated than many others. You still have a lot of manual steps. You still need a lot of lab tech, human interactions. At every step, you can have a different providers with different technologies. And on top of that, unfortunately, lab technicians are a more and more scarce resource also. It's very difficult to keep your workforce, and so you have rotations, differences in expertise, and so on. So how to address that, and how to be digital readiness then? So the way we do it at Leica, because we provide end-to-end solutions, we have consultants supporting the laboratory to transform their practice. What you need to realize is that pathology labs are empirical evolution, which is normal, and for all their life, they had to generate a glass slide, which is good for a microscope. If there are some adjustments to be done, the pathologist on the microscope can adjust. If you put a glass slide in the scanner, it's digitized and it's over. If you send it to a pathologist 200 kilometers from that, that pathologist doesn't know exactly how the lab is running. So you really need to work to optimize the workflow, to standardize, to work on quality, so that you deliver consistent, high qualitative glass slide so that you can put into the scanner, which can be therefore leverage, whether clinical or your research. We also have, obviously, a portfolio to support in terms of innovation. We're not just working on the workflow optimization. But our abilities is that we can work with the lab to improve from their current state to their future state with the different elements. We have also the capacity to start from scratch. So we work in multiple projects where we are from the origin of the project. We design the lab with the architects so that we can define the best workflow according to the need of the hospital, the private institutions, and so on. Because we are a Danaher organization, we have the chance to use the Danaher business system tools, which are fundamental of the Danaher culture and extremely critical to our global success. We use those tools not only to deliver on the promises, but also for the benefit of our customers. So obviously, digital pathology is also about IT readiness. It's from integration, implementation. We have IT experts who help the laboratory from the lab floor, on the traceability, on the data management, up to the discussion with IT management for integration, the best integration possible into the hospital environment to reach the standards such as DICOM, which is the norm in terms of image management in the environment to ensure that IT management is optimal and cost-effective. Thank you very much.
Thank you, Mathieu. Thanks for a great, and concise review of what Leica can offer. I think at this stage we will move to Dr. Hisham Feniri. Dr. Hisham is with us remotely. Dr. Hisham is the Vice President of Research in Mohammed VI University in the Kingdom of Morocco. He also holds several roles in this university, including Professor, Chair of the Technology Development and Business Transformation Department, and Vice President for Research and Innovation. At the same time, Dr. Hisham is also the Scientific Director of the Moroccan Foundation for Advanced Science, Innovation and Research. Dr. Hisham, welcome, and over to you.
Thank you very much, Bushra. It's a great pleasure to be here with you today, to be participating in this very important event. The Kingdom of Saudi Arabia is doing amazing things, and it's a tremendous inspiration for the Arab world and the African continent. So very pleased to be here today. The University Mohammed VI Polytechnic was inaugurated by His Majesty in 2017 and is already ranked number 3 in the country out of about 25 or so universities. And it's growing very fast. Our focus is on research and innovation, particularly applied research. And as you can see here, our focus is mainly on empowering talents and empowering Africa in the areas that I have mentioned, that are mentioned on this slide, but particularly for this conference, I just mentioned the biotechnology and biomedical engineering and the medical application interface. Next slide, please. So the university is organized into 4 clusters, one in science and technology, the other one is medical and paramedical, and the third one is humanities, economics, and social sciences, and finally the business and management clusters. So the different clusters have been created to respond to specific needs of the country and the continent, and we develop academic programs in response to specific needs, especially that we are affiliated with a major corporation in Morocco, is the OCP Group, And so all our work is oriented towards use-inspired research. So if there is a need for it, if there is a new technology to be developed, then we develop programs around those needs. Next slide, please. So the university has currently about 4,500 students. We're aiming to reach 6,000 by 25. Our aim is not to exceed about 10,000 students in a way similar to the MIT or Stanford model. But the community of postdoctoral fellows and graduate students will continue to grow. As you can see here, we have various programs, but particularly we have on the right there, we have specific program dedicated to coding. We have about 2,000 young coders in a very unorthodox educational program. It's a program that has been very, very successful, and it's growing. We have opened now 3 coding schools across the country, and the 4th one is in the pipeline. In terms of distribution of students, you can see that the female students actually is the majority in our university, and we have as you can see here, a population of graduate students currently that's at only about 18%, but ultimately this community is going to grow quite significantly. On the next slide, just to give you an idea about the openness of the university towards the world. We have partnerships across the world. And the idea here is to create this mirror laboratories with partners in various universities, where we have a faculty in our university and a faculty on the other side working together to address specific problems. So since this is a medical biotech conference, I would like to focus on a very, very small portion of the work that we're doing. On the next slide is the medical biotech at the Moroccan Foundation for Advanced Science and Innovation and Research. This foundation was created in 2008 and is focused on high TRL levels, so 4, 5, 6, and above, and has spun out a number of startup companies already. And this foundation is now part of the ecosystem of University of Mohammed VI Polytechnique. So on the next slide, you'll see that the laboratories have been certified ISO 9001, 17025, 13485, and so on, to do very high quality medical biotech research that allowed us to develop a number of diagnostic kits for Morocco and for the African continent. Just to give you some context, on the next slide, you'll see that in Morocco, until we got into this business, all kits were imported, usually from Europe or America. Same for the biomedicines. And so our mission was to create the first medical diagnostics and first biomedicines coming out of Morocco. And we are also interested in developing diagnostic tests for animal diseases and for developing platform of gene sequencing. On the next slide, I'll just give you a few examples of the kits that have been developed. For example, during the COVID epidemic, we developed the first kit, Moroccan COVID test that has been distributed, approved actually in Europe, and is currently obtaining FDA approval for North American distribution and worldwide distribution. We have sold already over 5 million kits just in the African continent. We have also the Hep C, TB, leukemia, breast cancer, prostate cancer kits that are in the pipeline, as well as rapid tests for COVID-19, Hep C, and HPV. And so on the next slide, the advantage of these kits is that, as you can see, they're all based on LAMP or PCR. They are innovative and simple, accurate, and have been clinically validated and patented. And they are we are open, obviously, to distributing these kits across the MENA region and beyond. So with that, I'll just finish with a couple of pipelines coming soon. On the next slide is our biosimilars. Those are antibodies that were currently in the production stage for breast cancer. And we're also developing vaccines, mRNA-based vaccines for animals. And with that, I would like to thank you again for the opportunity to speak to you and to get to connect with the community in the Kingdom of Saudi Arabia. Thank you. We cannot hear you, Bushra.
Oh dear. Can you introduce?
Yeah, maybe.
Maybe you should go.
There's no sound.
Go ahead.
Introduce.
Go ahead. Go ahead. I cannot hear you. She's going to do it.
Yeah, Beryl, Beryl, would you mind to introduce yourself?
I will.
Please, go ahead.
I think it's been difficult to hear Bushra, so I'm going to introduce myself. My name is Dr. Beryl Oppenheim, and I'm a Senior Director in Medical Affairs at Cepheid. If I could have the first slide. So to start, just to say, I am truly privileged to be able to be presenting here at this very exciting summit. And what I would like to describe to you today is how Cepheid is leveraging a continued focus on innovation in order to deliver on our mission, which is to expand access to our tests and systems in order to improve patient outcomes throughout the globe. Cepheid is indeed a global company. We have a presence in more than 100 countries, office locations in 17 countries around the globe, and manufacturing in the United States, in Europe, and in India. And our tests and systems are used in extremely diverse settings, in very large hospitals and smaller and medium-sized hospitals. In the hospital, of course, we're used in the laboratory, but increasingly closer to the patient, for example, in the emergency room or in intensive care units. And then our tests are used outside of the hospital, in clinics, and in very unconventional community settings. But regardless of the setting, the solution is the same. It's the GeneXpert platform, an instrument that can be leveraged up or down, flexed depending on the need, to have an instrument with a very small footprint, running just 2 tests at a time, all the way through to our very large Infinity, highly suited to high throughput testing. And regardless of the size of the instrument, our users can be reassured that it's the same technology, the same tests, giving the assurance and ease that they get from knowing about the accuracy, the performance, and the simplicity of use, suitable for this very diverse setting and users. And just in terms of the range of tests in our pipeline, we already have more than 30 CE marked tests available, focusing across pretty well all of infectious diseases and also oncology and human genetics. And just some examples would be our respiratory tests, of course our test for COVID, either having SARS-CoV-2 as a standalone target or in combination with influenza and RSV. We also have tests that detect the main organisms causing hospital-acquired infections and causing antimicrobial resistance. We have tests for blood-borne viruses, for women and sexual health, and of course our tuberculosis tests, which I will say a little more in a minute. Just thinking about the pipeline, we are going to be leveraging our innovation in a couple of ways. Firstly, to go deeper into multiplexing so that we're able to detect more targets, think more about syndromic testing, and the first tests available will be for respiratory and gastrointestinal infections. And then we're thinking about updating existing tests to make sure they meet the needs of the future. So when we think about our innovation pillars, what is the strategy? And there are three main areas to our strategy. Thinking about improving access to decentralized diagnostics, but at the same time moving into diagnostics for complex care, and finally, and extremely importantly, continuing our focus on diagnostics for global health. And within these pillars, we need to think about a great many angles. Of course, the expansion of our test menu to be able to focus on more and more infections and diseases. Improving the systems themselves to be easier to use and more able to innovate for new tests. Focusing on our analytics and informatics, and also focusing on improving the technology within the cartridge. And I'm just going to give you some real examples, particularly focusing on our work on global health. And a couple of examples that I'd like to give is really something that there's now a completely new line of research on in terms of infectious diseases, which is rather than just detecting the pathogen, the organism, there is a focus on also having a look at how the host is responding to that infection. And it may be that the host response is able to give additional information as to what type of infection it is and also what stage the infection is at. We are also thinking very hard about moving into mid and high-plex, deeper multiplexing that allow us to look for a wider range of pathogens or targets just in the same cartridge. We very much, for the decentralization, want to make the time to result as short as possible and are working on technology to improve this. And finally, we are thinking about alternate sample types, again, that may be very useful close to the patient. And a big example would be to use fingerstick blood to be able to make a diagnosis in the sorts of settings, for example, where venipuncture or blood processing is not available. So just some examples of tests that are either available or near to being ready. And a good example of our multiplexing technology is our new 10-color technology, which changes the module so we can do much deeper multiplexing but maintain that high level of accuracy and performance. And the first test on the market to use this 10-color technology is Expert MTB XDR, a test that allows clinicians to take a look at the most important resistance mechanisms for the major drugs used to treat tuberculosis. And it's been heartwarming and delightful to me to see that this test is already available and in use here in the Kingdom. And we really hope it's going to make a big difference to patients. I was talking a little bit about this concept of focusing on the host response to infection rather than necessarily only the organism. And this is the first direction that Cepheid is going into trying to develop the expert TB finger stick host response test. Now we know that immune cells in the blood demonstrate TB-specific changes in gene expression. And what this test wants to do is use mRNA gene signatures in an algorithm to give a score looking at the host response and attempting to firstly rule out that that patient has got tuberculosis, but also hoping to say this patient is very likely to have tuberculosis, and also what is their risk of developing clinical infection in the near future. And we already have some very exciting results from a prototype cartridge. Another example of how our 10-color technology may be leveraged in the future to think about pandemics and important emerging threats is a test that we are developing really to keep available in case of need, which is the pan-coronavirus assay. And the intention of this test would be to cover broad-range coronaviruses and then call out those that we know. And this test could be leveraged if we started to suspect that there was a novel coronavirus coming out and causing problems. And that leads me very neatly on to explain to you about Cepheid's continual focus on being fully prepared for the next pandemic. And our plans are to work with governments, but not only governments, with regional networks of key opinion leaders, with NGOs, other organizations, to be sure we will be right there as soon as there is a suspicion of a new outbreak or pandemic. And then we would leverage all the different functions within Cepheid, but also working with third parties, if that is the most appropriate thing to do, to bring a new product to market as quickly as possible, and also to ramp up the production to make it available where it's needed. So I'm going to just wrap up by saying the last 12 months we did have some very exciting innovations and new launches around our hematology oncology portfolio, respiratory, women and sexual health, and some launches around our systems and software. We have some very exciting tests in the pipeline, and we are very excited to be working with you to make sure all these tests are available and helpful to you here in the Kingdom. And thank you so much for listening.
Great job.
For the technical glitch earlier, so I will still take time to introduce properly our Dr. Beryl. Dr. Beryl is our Senior Director Medical Affairs in Cepheid, as you have seen. She has extensive experience in microbiology in the National Health Service in England, and that for many years. At the same time, she has been the director of 2 large public health laboratories as director of infection prevention and control. And her main interest goes to antimicrobial resistance, infection prevention, and specifically infections in the critically ill and immunocompromised hosts. Thank you very much, Dr. Beryl. And at the moment, I think we're going to change gears. We are going to open the dialogue with our speakers. And probably I'll start with my first question. Monica, my first question would go to you. And again, thank you for your inspiring presentation. Now, I think we have seen that you mentioned the menu that matters. And of course, we want to be a bit like zooming onto the Kingdom of Saudi Arabia. And if you can describe for us what would be, to your eyes, the menu that matters for Saudi and how it will impact the patient's care in the future in the kingdom.
Absolutely. Thank you for a great question, Bushra. So what do I mean by the menu that matters? Every patient at any given time when they go to see a doctor or to the hospital, lab touches them. Means the blood test that gets back to the physician is from the lab. So lab is the first to know. And we want to make sure we equip our laboratories not only with great solution and analyzer and automation, but also with a menu that address the community health challenges. When we look at the Kingdom of Saudi, just to call a few of the community challenges as we see them, and I'm open to your suggestion if we are missing any, diabetes, AMI, sepsis, just to call the few. And we do have comprehensive tests, and some of them, for example, in the area of sepsis, are new to the world, biomarkers that are second to none, that can help the Kingdom of Saudi. So that's what I meant with the menu that matters. Lab has the gem, the gold in the data, and they can provide impactful insight to physician, and the physician can change the patient pathway. Thank you, Bushra, for a great question.
Thanks, Monica. Wonderful answer. And let us also probably give a chance to Mathieu this time to tell us about one element of your presentation, Mathieu. You mentioned that Leica Biosystem has reference sites for digital pathology, and I have noticed that we had North America, Europe, and even in the Middle East. You mentioned some sites in Kuwait. So I want you to share your thoughts on what does it take to have an established site in Saudi, and what do we need as prerequisite to succeed in having that kind of a site here in the kingdom?
Thank you, Bushra, for the question. Indeed, I hope I explained that digital pathology is a journey. We are at the moment very active, and we hope, and I pray nearly every day, that we'll be able to participate to the virtual hospital in the kingdom. In terms of expectations, if I can say so, on our side, there's already very high standard in this country. Still, for that journey, we believe we need to work in project management mode, because it's a lot of resource and time on top of delivering high-quality diagnostic for the patient. So we would require to have a team and that will be able to work together with the right stakeholders. We would also ask for a good executive sponsor. It's critical to maintain the direction of the project and also ambitious goals. So for instance, being able to digitize 70% of the workflow within 6 months. That will be the basis to go toward that direction. Thank you for the question, Bushra.
Thank you, Mathieu. And I would assume that with your interactions at the moment, you could see that somehow, not, I wouldn't say easily, but fortunately happening in the kingdom in the near future.
Yes.
Wonderful. So let's move now to our Dr. Hisham. And again, thanks for shedding light on what you have been able to establish in the university, Mohammed VI University. That was really impressive to see how fast this has happened, and in the middle of a pandemic. Now, we would really like to learn from your lessons. What were the experience? I mean, how you've been through the journey, and if you were to tell us, 2 or 3 things that you would want us to learn from your experience in this project, what would those be if we were to establish either a diagnostics or a biotech ecosystem in the kingdom?
Well, thank you for this question, Bushra. I think it's important to let me just put it this way. Currently, academic research is very, much separated from the real life issues, the day-to-day issues, whereas corporate research is very close to what the market needs. And then there is a huge white space between academic and corporate research, to the point that the communication between industry and university is very difficult to establish. There are a number of programs that have been created across the world, and very often they are not very successful. There are a number of programs created in the United States, for example, to support innovative translational work, but it's very, very limited. So the idea of UM6P, University of Manchester Polytech, is to fill this space. It's an open space. Very few organizations are actually filling that space. So we are focused on very applied research. So we're, of course, if we come across interesting discoveries and interesting findings, they're going to be published in the proper journals. But the aim here is to solve issues that society needs solution for. And so, for example, in 2019, when we were faced with the COVID pandemic, it was very difficult to get a COVID test in Morocco. And so we had people who mastered the technology of the PCR. We had already developed a number of tests for a variety of diseases, and we could position these researchers to develop a kit in 3 weeks. And so that's the positioning that the university has taken to address the pressing need, not only of Morocco, but also of the African continent. The problems in Morocco and the problem in the Arab world and the African continent are not necessarily the same problems as in Europe or North America or the rest of the world. We have our own issues and challenges that we have to solve ourselves. And so this is the vision that has driven all our programs and our approach to research and innovation. Thank you.
Thank you very much, Dr. Hisham. And the way I have seen the country going through the pandemic is very reassuring that institutions like yours have certainly contributed to the success of going through such a difficult time. So we are left with you, Beryl. I think that Cepheid has also played a wonderful role into fighting against COVID that we know by now, not only in the development, but also in the deployment of the most reliable, but maybe most reliable PCR test in the globe. Now, of course, we are in a phase where we are post-pandemic, and we hope to see, we continue to see this stream of innovation. We, as a company, we know about the multiplex and the likes. So I just wanted you probably to help us understand, if we were to look at the post-pandemic priorities that Cepheid has for itself globally, how do you see that being deployed in the kingdom, and what would you advise us to look at as a post-pandemic and looking forward kind of a view?
Thank you so much for such an important question. And you're absolutely right. I think we have learned a lot during the pandemic, and lessons learned can really be leveraged for the future. I think I've been really impressed and found it heartwarming in visiting some of the outstanding hospitals here in the kingdom to see how our tests are currently being used and the significant impact they're already having on patients. But I think there is still a lot to do to make sure that our tests are as widely available as can be, and making in the settings in which they would have the greatest impact. And for me, the next steps would be that we need to work with clinicians, with experts, and with decision makers to fully understand the healthcare needs and how we can deliver tests that will reach the patient as quickly as possible and make the greatest impact. we know we need to provide scientific evidence to prove the impact that the tests can have on patient care and patient pathways. And finally, some of our innovations will indeed be disruptive to patient pathways. Pathways may need to be changed to deal with the rapidity of results coming and how to use them. And to do that, I believe we really need to provide the clinicians, doctors and nurses who will be using the results with the education and tools so they know exactly what to do with the result and how to action it for the maximum impact. Thank you.
Thank you, Beryl. Thanks a lot for your explanations. So at this stage, we are almost at the end of the session. I just wanted to, first of all, thank again our speakers, whether they are on-site or remote. I wanted to also somehow wrap up a bit the session, just like conclude on what I heard and what we have said and shared. Monica was instrumental in showing us how Beckman Coulter challenged the conventional work to be able to elevate the laboratory role into improving the health of the patients. Then we had Mathieu helping us to highlight the importance of the workflow digitalization, because that enables the clinicians to an accurate diagnosis, and of course, developing the confidence in that diagnosis. Dr. Hisham Ferini brought us up to speed on how academia or an institution or university is able to somehow be at the center of a national program and be able to create that dialogue between the private sector and the need of a country. So a lot to continue learning from that. And of course, our Dr. Beryl was able to let us understand how Cepheid, as a molecular diagnostic leader, is able to play a vital role in battles that are national health challenges, infectious diseases, AMR, AMR, and priorities that are top priorities for many of our countries. And I can include potentially this region, all the countries of this region would be looking at those as main priorities. So with that, I can see we still have time. So in case there is any question from the audience, we would be happy to take it.
Do we have any questions from the floor? It seems no, Ms. Bushra.
No.
Yeah.
Thank you. So I just want to thank our speakers once more, thank the organizing sponsors for such a wonderful dialogue, and wish everyone a wonderful summit. Thank you very much.
Thank you, esteemed panelists, for a fruitful session. Innovation is indeed reimagining the global healthcare. Ladies and gentlemen, now we'll take a 30-minute break. Stay connected. We'll see you soon. Thank you. Esteemed guests, welcome back. Our next session will bring together regional and international speakers for a dynamic exchange of knowledge, expertise, best practices, and global benchmarks for clinical trials. Please join me in welcoming co-chairs Dr. Majid Al-Jraisi, Chairman, Research Office, Director, Clinical Trial Services, KMARC. Welcome, Dr. Majid. And Mr. Walter Klim, Chairman and CEO, Moleculin Biotech. Welcome, Mr. Walter. And Dr. Elio Ribello. Welcome, Doctor. To you, Dr. Majid and Mr. Walter. Thank you.
Assalamu alaikum. Welcome back after the break. Today our session is about the one of the attractive sessions, which is the clinical trials, and all the biotechnology, population health are ending with supporting the conduct of clinical trials. So without further ado, I would like to introduce my co-chair, Mr. Walter Klim, and also with us is Professor Elio Ripoli. So we will start with Professor Elio Ripoli. He's chair in cancer epidemiology and prevention at Imperial College London. He is also founding director of the Cancer Epidemiology and Prevention Research Unit. He initiated the European Prospective Investigation into Cancer Nutrition, EPIC, that recruited over 500,000 in 10 European countries and established the first dedicated large-scale population biobank. Currently, he is Director of Cancer Epidemiology and Prevention Research, Director of Public Health Research, Imperial College Healthcare, National Health Services Trust, UK. Professor Ripoli will be talking about the role of population biobank in medical research. Please join me to welcoming Prof. Ripoli.
Well, first of all, thank you very much it's a pleasure being here. It's my third or fourth time in the kingdom, and it's always a great pleasure being back and meet a number of great colleagues. So the session is essentially on clinical trials, and you may wonder what population studies have to do with that. So I'll try, and we will try, to show you that there is a continuum going from initial observation at the population level about risk factors to better understanding the mechanism of disease to eventually identifying possible targets for new drugs, and happily at the end go to the clinical trials to see what works. So in 3 seconds, I have given you the sense of my presentation. I could walk away, but I will now, with your attention, try to go through the logic a little bit more in depth. So medical research over the past 2 decades have brought us to a completely different vision of what causes the diseases that eventually will kill us. So we have little by little understood that the epidemiology of disease is changing around the planet. Infectious diseases remain very important, but non-communicable diseases have become everywhere in the planet, almost everywhere in the planet, the dominant epidemiological characteristics of disease and eventually as a cause of death. And almost 10 years ago, we were asked by the New England Journal of Medicine to come up with a global vision. So we analyzed all the 55 million deaths around the planet, and what came out by using all the knowledge at that time is that, as you can see, as you can see, the number one underlying cause of the disease that will eventually cause us troubles is high blood pressure. Number 2 is tobacco. Number 3 is having high blood glucose as a precursor, obviously, of diabetes, but also of cancer. This is not well known, and the cardiovascular diseases. With an underlying problem of physical inactivity, high energy intake, which translate in high body mass, and translate as a biomarker level in high cholesterol. In some countries, alcohol use as misuse is a problem, and there is a generalized issue of low consumption of fruit and vegetables. The red and the blue are what really has changed our vision of the planet, because the red are the millions of deaths per year in low and medium income countries. So the old idea that cardiovascular diseases and cancer are a characteristic basically of Europe and North America, just to go quickly, is completely changed. Now the majority of cardiovascular disease and cancer cases occur in low and medium income countries. So from this, one could say, okay, we've done the job. We can go home and public health can take over. Where is the problem? The problem is, just by knowing this is not going to change the planet. This is not going to change behaviors. This is not to change what you buy in the supermarket. It's not going to change the way food is produced and what priority is given to food production. You need to go, and to link with this, is not going to provide medications that might prevent the occurrence of more serious diseases. So what you need to understand the mechanism, because epidemiology, to go from knowledge to prevention, needs to go out from the black box of saying these are risk factors, to go to the open box and say these are the causes, and there is a huge difference. When you know the causes, you can do something on this. And to know the causes, you need to open the box. The box is what we do every day in our life, and then one day we will be diagnosed with a disease. But what is in between, what is in between can actually be measured in population-based prospective core study, or in short, biobanks. And what we need is what we have for cardiovascular diseases. What do we have? We have cholesterol, we have blood pressure, we have high hyperglycemia. So we can actually use medication and intervene on intermediate risk factors. You are not going to be killed by having a slightly high glycemia, but something will happen download. So this is why biomarkers of exposure and biomarkers of health and disease changes are fundamental because they are the gate to the occurrence of disease. So with all this in mind, 20 years ago, I initiated what Majed very kindly presented as the European Prospective Investigation on Diet and Cancer. So that was the first time in the world that researchers from 10 countries came together. We collected data on over 500,000 participants, very detailed data, and we collected blood from over 400,000. And it's not, so this is the logic of a population study. The logic is that first of all, you need to make measurements. That is the, let's say, the heavy load, the not exciting part. But if you do that, the blue box, well, you collect the data and you put them in a way you can use them. Then you need a second one, the green box. The green box is the follow-up. You need a health system that allows you to identify that Mr. Mario Rossi in Milan, who entered in the study in 1993, was later on diagnosed with colon cancer. So you need that link that will give you the green box. When you have the green and the blue box, then you can finally get the price of your work because you can link the occurrence of the diseases with what you have measured at the baseline. And clearly the baseline can be sophisticated because you can have every 2-3 years, for example, collection how people change their diet. So this is a machinery that has been going on for 2 decades that has initially produced quite a lot of what you read in public health. Fruit and vegetables are good, being overweight is bad, and so on and so on and so on. But has produced also much more, because at that time we stored 10 million samples from 400,000 people. We have now used, and give you an idea of the turnover, we used 1.5 million for genomics, metabolomics, biomarkers, and so on. And all this is based at the International Agency for Research on Cancer of the WHO in Lyon. And there is always a human factor. The lab technician who is there, his name is Christophe. So Christophe was the first lab technician who started working with us, and Christophe is still there. That there is the element that you must secure stability. To do what? Well, first of all, the value of a biological samples is peanuts unless it comes with phenotypic data. Just to have the blood there, well, blood transfusion bank have plenty of blood, but they don't do research. You need personalized data. You need, as we have done in many places now, in UK with the Airwave Court of 500,000 policemen and women, with Qatar Biobank, with the Helios study in Singapore, to collect intense data. For example, we have done blood pressure, arterial stiffness, spirometry, imaging, ECG, a lot of imaging on tens of thousands of people, data on diet, and so on and so on. So when you have built all this treasury, then you can actually, you have the data of what we sometimes a bit lightly say omics. Because omics need all what is before to be meaningful. All the phenotypic data, the disease data, the behavioral data, and then omics start making sense. Then you can say, okay, I do a genomic study. I led the first NCI consortium on breast and prostate cancer core consortium that brought together tens of thousands of cases. And we have learned, for example, that by bringing together all the genetic variants we have found in 10 years of work, the predictive value of genetics is much less than the predictive value of personal history, reproductive history, height, weight, and physical activity. So the risk of having breast cancer at age 60 is poorly predicted by genetic variants because the behavioral metabolic factors dominate. Now, I just show you some examples, are just, flash, like in the news. With some of my colleagues, recently published in the Proceedings of National Academy of Science, something which is, groundbreaking. Basically, they did GWAS on Alzheimer cases from our courts, identified the number of single nucleotide polymorphism which predict Alzheimer's disease, then went into metabolomics, and those SNPs happened to be on some genes. And what these genes were doing is amazing, are genes that regulate lipid metabolism. They regulate the balance of ceramides, which are a particular type of fatty acids brought around in our body. And so the big surprise is you never thought that fatty acid lipid metabolism, that is known to have a lot to do with cardiovascular diseases, could have anything to do with Alzheimer's. So this is kind of opening a new window to our knowledge. Recently, my colleagues used the same set of data and did a very, let's say, very large metabolic study on 44,000 people and with 5,500 deaths, and came out that there are a set of genes that are related to a set of metabolites that boils down essentially to lipid metabolism again. And these are genes that predict whether you are more likely or less likely to premature death. Premature death is by definition dying at age around 60, 70 nowadays.
You have 1 minute. You have 1 minute?
1 minute, exactly. Another example is to use genetic to calculate the genetic risk score for cardiovascular diseases, but then you have all the phenotypic data. And the good news is that if you divide the population into low genetic risk, intermediate genetic risk, high genetic risk, if people are slim, physically active, have the right diet, don't smoke, don't drink, they have a lower risk of cardiovascular disease. So healthy lifestyle can offset genetic risk. No time to go into this. I say in 2 words, breast cancer, the big unknown from the point of view of prevention, no way to prevent breast cancer, good way to diagnose and treat it. We have demonstrated that breast cancer is linked to metabolic syndrome. So testosterone and estrogens are higher, and free bioavailable form are higher in people who are insulin resistant, have high insulin, low sex hormone binding globulin. We have done the similar things in colorectal cancer, where we have translated obesity, physical activity, and energy balance into specific pathways that translate into increased cancer risk. And have translated already in recommendations, what is green is good, what is red is bad, recommendations that have been adopted, for example, by the American Cancer Society and the Cancer Research UK. My last slide is to say, just to remind that there is a big continuum, was the beginning, in research, understanding mechanism, understanding causes, understanding mechanism, understanding biomarkers, understanding the possible targets, and then my colleagues who are down the road in pharma and biotech take up from there and translate. At the same time, you can say public health has now a rationale for promoting health behavior in the society and at the individual level based on science, not on beliefs. And that is what I think is the message of a scientifically based public health. Thank you for your attention.
Thank you very much, Prof. Ripoll. And we go to the next speaker, Mr. Walter Klemp. He's the chairman and CEO of Moleculin Biotech, a NASDAQ-listed drug development company based in Houston, Texas. Mr. Klump is the founder and chairman of two current NASDAQ-listed biotech companies, one in cancer and drug development and one in the medical devices, with 19 years experience of biotechnology industry. He helped facilitate an initiative similar to Saudi Vision 2030, the country of Poland, which is Vision 2020. Mr. Klump will be talking about the global advancement in clinical development. Please join me welcoming Mr. Klump.
Thank you, Dr. Al-Jarazi, for that kind introduction. And I'm going to ask you to make a little bit of a mental shift here. Dr. Riboli eloquently described for us an opportunity to change how we approach what we study and why we study it, to provide a rational framework, long overdue. I'm going to talk about something very different from that. I'm going to talk about why clinical trial sponsors choose to study in one location versus another. And that has very significant relevance to Saudi Arabia. I'm very passionate about clinical trials. I'm a biotech sponsor. I've run 22 clinical trials of my own technologies. A third of those clinical trials have been outside the US, which affords me the opportunity to provide some perspective on why we would choose a clinical trial location outside of our home country. Now, I realize that it's well understood why we do clinical trials, what the importance is, especially to this audience. We all understand that clinical trials are critical to innovation, they're critical to new drug approvals, and they bring much needed cutting edge technology to a population base. So very important. But I'm really going to shift and talk about the fact that in addition to all of those important things, clinical trials are big business. $80 billion was spent last year on clinical trials globally, and that was just the direct cost. When you look at the overall economic impact, it's probably 3 times that number. And how many businesses do you know that have generated 30% compounded annual growth rate for 20 years in a row? I mean, this is a real economic opportunity. And unfortunately, Saudi Arabia has been missing out on this explosive growth opportunity. And let me give you some examples. The country of Poland, by the way, has a population and a GDP that's very close to Saudi Arabia. But Poland has 7 times as many clinical trials per capita than Saudi Arabia. Now let's look at Australia, though. There, the government instituted some very aggressive programs to attract foreign sponsors like me to conduct clinical trials in their country, and it worked. They conduct 100 clinical trials per 100,000 population point. That's more than double the United States. It's 30 times the amount in Saudi Arabia. So, as I said, I'm a biotech sponsor, and I want to use that perspective to help you understand how we make decisions about where to conduct a clinical trial. In order to have the right frame of reference, I really need to start with the fact that as a small biotech company, my real objective is essentially to exit to a larger biotech company, to sell my technology to that larger company. What that really means, and by the way, to do that before we've even gotten a drug approval. So what I'm really in the business of is selling data. I sell data. And what's important there is it has to be marketable data. And in order to be marketable, it has to be credible. But eventually, if I have a successful negotiation with Big Pharma, we're going to agree on a price. And the price for that data is going to be dependent essentially on one really important thing: length of exclusivity, LOE. That's essentially the patent life plus whatever regulatory exclusivity we might have in a given country. Now, what's really important is to understand that what I just described means that the most important resource to my company, the most important variable, is actually time. Now, I'll invite you to recognize what's not on that list: cost. I think a lot of people would assume that any important decision like where to conduct a clinical trial would involve the cost per patient of that clinical trial. Doesn't really matter. Let me explain why. There's an old adage that time is money, and it's no more relevant than in the area of biotech development. And to understand why, it's important to understand the notion that when we exit as small biotech to larger biotech, what we're really, our objective there is to essentially capitalize on that value of the length of exclusivity. And in order to understand the importance of the length of exclusivity, you've got to pull back, and you've got to look at the reality for drugs and their patentability. So unfortunately, most cancer drugs will only have 7 to 10 years worth of exclusivity left by the time they get drug approval. And the reason that's so important is because when we're negotiating with Big Pharma about the value of that data set, that negotiation comes down to a calculation. It's basically a formula. It's net present value of the future revenue stream that's expected from that drug once it's approved. Now, unfortunately, that value is highly dependent upon the length of exclusivity. The longer the length of exclusivity, the longer the revenue stream, the higher the number. In fact, if that length of exclusivity is increased by a year, the value to my company on average is an additional $100 million. $100 million for extending the life of exclusivity for that drug. So when it comes right down to it, I don't really care what the cost of a clinical trial is. What I really care about is how long is it going to take to recruit that clinical trial. So the reality here is that countries that have a lower utilization of clinical trials within their country, that have a lower per capita number of clinical trials, those countries become more attractive targets to sponsors like me. And let me give you some examples to drive this home. Let me talk about AML, which is acute myeloid leukemia, and soft tissue sarcoma. I pick these examples because these are real for us. My company has technologies that are right now in clinic for AML and STS in both the US and in Poland. So I have direct data to give you. Okay, in the US, AML has a disease incidence of about 20,000 patients per year. There are almost 600 clinical trials chasing those patients. What that means is, on average, it's going to take me 21 months to fully recruit a 100-patient trial. So now let's compare that to Poland, where the number of clinical trials actively recruiting is only 10, okay? Now, lower population, but vastly lower ratio of trials to population. What that means is I'll save 16 months on average recruiting that trial in Poland versus the US. And if you look at the numbers related to soft tissue sarcoma, the savings are closer to 2 years. What this really means is I'm highly motivated to look for opportunities to shorten clinical trial time. Now, if you take this example and apply it to Saudi Arabia, where clinical trial activity is 7 times lower than even Poland, guess what? Saudi Arabia becomes my #1 target for my next clinical trial. And that's not the only advantage that Saudi Arabia has. As a country, you have another major advantage, and that is that you're a kingdom. And because you're a kingdom, you have the ability to make decisions much faster than any other country on the planet. And once those decisions are made, you have the ability to implement those decisions much faster than any other country on the planet. What that means is essentially you have an opportunity to become the destination for clinical trials. In fact, it's so meaningful. Last year at this same conference, a group of us presented a proposal to the royal family for essentially changing the paradigm, changing how clinical trials are started and how they're managed in your country. And I'm really pleased to say that there's now a team here on the ground implementing those procedures and those changes. You are about to make history. And I'm telling you, this opportunity is real, and it's very exciting to those of us who want to conduct clinical trials. So I hope that you'll join me in frankly telling everyone who will listen that now is the time for Saudi Arabia to shine as essentially the destination, the global destination for clinical trials. Thanks for your attention. I look forward to talking to you more about this.
Thank you. Thanks.
So I'm going to try to do this swiftly, but it'll take a little bit of time. I have the honor of introducing my co-host for this panel. Dr. Majed Al-Jarazi is the chairman of the research office and director of clinical trial services at KMARC. And in addition to his Doctor of Pharmacy from the University of Tennessee, he also completed his Master of Science in Medical Education at KSAU-HS in Riyadh. He's currently Associate Professor at the College of Pharmacy at the King Saud bin Abdulaziz University for Health Science, as well as the Pediatric Clinical Pharmacy Consultant for the King Abdullah Specialized Hospital. Please join me in welcoming Dr. Majed Al-Jarazi.
Thank you very much, Wally, for the nice introduction. And thank you for paving the stage for me to talk about the structure of clinical trials in KMARC and hopefully to be the setup for the other part of the country. It's like attractive and you have inspire us with your last statement to be the hub for clinical trials, and we will be, inshallah. So just to talk about KMARC, I'm sure most of you know KMARC. So it was established in 2007 with only 10 employees. Now it's 500 plus employees, and we have 3 campuses, one in Riyadh, and with 35,000 square meter, one in Jeddah and one in Al-Ahsa, and we have also our Saudi Bank here in Riyadh. We have many structures in KMARC, 3 different sections, mainly talk about the developing platforms, departments, and we have the section for supporting researchers and also the entrepreneurship and biotechnology park. Without going through the long list, you can see here from the platforms that we have, and we are opening the collaboration with other institutions, local institutions as well as outside institutions, and we are bound to have these platforms: medical genomics, experimental medicine, bioinformatics, biostatistics, medical research core facilities, biobank, cancer and cord blood bank, cancer research program, stem cells and regenerative medicine, clinical cardiovascular research program, nanomedicine, population health, developmental medicine, infectious diseases, immunology research program, Saudi stem cell donor registry, and we add to this the vaccine development or the vaccine platform. So these are the services that we are providing to all researchers. including the Operation Research Office, Innovation Technology Transfer Management Office, which is really important unit for taking the patent into registration and encourage our investigators to register their patents. Research, training and development, doing a great part in training our staff and the clinical investigators. We have the Advanced Computing and Technology Safety Office, Science and Technology Unit. And also we have the Biotechnology Park with Business Strategy and Bioequivalence Lab. So these are the current existed infrastructure that exists in KMARC so far. And we have to talk about our initiative, which is the National Command Center. And we have named this as the Saudi Enterprise, the Saudi Clinical Trials Enterprise, which is the national initiative that's funded by the NIDLIB. Main charges are to develop and enhance the capability and capacity of clinical trials in the country. Ultimate goal is to attract more sponsored clinical trials to the country and with high impact on healthcare and economy, and contribute in the development of R&Ds and pharmaceutical industry with, again, high quality, maintaining the quality of data that we generate. With this also, we have developed the advisory board that include all stakeholders from the governmental sector as well as from the other companies. Also, our regulator, the Saudi FDA, is on board with the advisory board. And the charges of the advisory board is to oversee the process of establishing clinical trials in the country. And engaging the other stakeholders and institutions also with us is to help in building the infrastructure and the capability in the country. Now, let's just focus more about the clinical trials in KMARC. So, our department of the clinical trials, we have a multidisciplinary team that is highly dedicated and dedicated to provide clinical trial services to investigators, which is the research coordinator. We have the scientific officer to design or to help in designing the study and developing the protocol, doing the feasibility studies by statistics. Also, in addition to this, we help in starting up the studies, site initiation visit, patient recruitment, investigational drug management. Also, in the project management scale, we are assigning clinical coordinator to help the PI in conducting the clinical trials. We do the training to the trial team, site monitoring, data management, safety reporting, archiving, and quality assurance. So these are the services that we do provide to our investigators. And this is what is really important in order to promote clinical trials, maintain the quality of the data that generated from clinical trials. We have to have these services in any institution. I think my colleagues have talked about the phase 1 clinical trial. And this is in line with what Wally was talking about, is how can we be the hub for clinical trials in the region? And these are the differentiating market for us in the country. So first is the location. Geographical location facilitates communication and interaction to bring in the necessary services to build reliable clinical trial infrastructure system. Second is the healthcare infrastructure. I think we have a very advanced system in our country that we should be proud of in terms of the electronic healthcare system and also the well-trained physicians providing these services. Education. Significant investment has been made into the education sector. Numerous universities currently offered these certificates and to help in building and graduating high-quality physicians and also healthcare providers. The fourth one is the cross-sector collaborator, and you have witnessed the collaboration, the agreement that was signed and the opening ceremony. There are so many collaboration with local as well as international, well-known and recognized parties that are unique in their services in terms of providing research facilities or sciences. And the last but not the least is the governmental support and structures. Existence of research funds and graduate scholarship programs, this is what makes us unique in terms of providing funds to our investigators and also providing support to the researchers, which will produce eventually high quality clinical trials. So this is in line of what we want to be in the near future to build the biotechnology park as well as and help and facilitate the clinical trial infrastructure. This is just some of the statistics that we have in KMARC about the variety of clinical trials that we do. So we have cardiovascular, endocrinology, GI, infectious diseases, oncology, asthma, COPD, surgery, procedures, pediatrics, as well as other disciplines that we do clinical trials, in particular with some of the well-known sponsored pharma industry. This is the map for taking the product from the basic science into preclinical up to the marketing and the commercialization. So our part here is to help and produce that high quality data in order to reach the commercialized product, which is the clinical trials. This is not my slide. I think it's Dr. Abdel Ali, and thank you for giving me the or lending me the slides. So this is what we are talking about, the ecosystem. I think, in my opinion, the most important thing that we have to consider and we have to think about is developing ecosystem. When we talk about ecosystem, our Saudi leaders has demonstrated strong commitment towards developing a national R&D agenda, and we have seen that yesterday. The Saudi government has increased funding towards R&D and increasing funding available through center capitals and also by establishing the Saudi NIH. Evolving regulatory and government sector with Saudi FDA working closely with international industry to develop regulatory framework for clinical trials, and you have seen the engagement of our regulator in that. Saudi also has attracted many multinational pharma and biotech companies in recent years, as well as the growing local capacity. In addition, we have increased the academic research by the Saudi industry, which is the NIDLB. And last but not the least, increasing the support for Saudi entrepreneurs and innovation into the form of accelerator program and innovation hubs. So these are all systems that we have, and we have to connect all these dots, government, pharma industry, in order to have ecosystem supporting our initiative of building the clinical trial in the country. And thank you very much. So with being the last speaker, we would like to open our panel discussion for our panelists. And I would like to start with Prof. Elio about the population and how can the population and biobank help in conducting and also facilitating the clinical trials.
Thank you. Thank you for the question. Obviously, it's part, I would say, it's part of the long process that goes from understanding diseases, mechanism of disease, to identifying possible drug targets, and eventually going to the development of something that can be tested in a clinical trial. What one might say is that looking back in the past 10 or 20 years, there has not been let's say, an optimal connection between what is done at the population level, with often discoveries of important biomarkers, and how this knowledge of new biomarkers might one day be taken up and developed. So we live in a super specialized medical scientific environment, and opportunities like, for example, this conference for connecting between different sectors are probably have not been sufficiently used. So I think that communication and also a little bit of risk-taking from the private sector. I led in 2001, which is 20 years ago, the publication of studies clearly demonstrating that to be insulin resistant, to be pre-diabetic, is associated with an increased risk of colon cancer and postmenopausal breast cancer. But all this could, in a way, be one day translated in some kind of chemoprevention, but there is a hesitation of taking the risk, of doing the research that might one day bring to actual a product that you can be prescribed in a drugstore.
Great. I totally agree. I think this is really important. I think having the connection between the biobank and the population health, and also paving the way for the investigators to look for the gaps in the population and try to find the opportunities in filling those gaps. And in our country, we are. Our statistics show that as epidemiology in terms of diabetes, we will reach probably up to 35% by probably in the next few years. So without filling this gap of having enough data of a population and see where we can do involve in clinical trials in terms of helping the problem of the diabetes in the country, without having the data, I don't think this is going to happen. Let me move to Wally, and since you have bring the experience of Poland, I think this is something that we have to benchmark with, since the session is clinical trial benchmarking. I would like to ask you, Wally, in your experience, since you have contributed and facilitated in developing their 2020 vision in Poland, where you can see the link between the Saudi vision and the Poland vision in terms of building the infrastructure of clinical trials?
Sure, this is a very important subject. Poland started out actually pretty close to where Saudi Arabia was. 10 years ago, they just had a trickle of, 15 years ago, had just a trickle of clinical trials. But there was this institution of Vision 2020, and the notion that if there was a collaboration between government funding, incentivized funding, and existing institutions to work together to attract private industry, it had to really be a three-way collaboration. That's really what sparked the opportunity for Poland to just skyrocket to the point of they're really the leading clinical trial center in all of Eastern Europe. And it wasn't by accident. First of all, they made a huge investment in infrastructure. I first came to the kingdom 3 years ago, and what struck me immediately was the fact that you have a hospital system, an infrastructure that's second to none. I mean, it's top class in the world, right? I saw the same thing in Poland. The difference was that the right combination of collaboration between private industry, government grant funding, incentivized clinical trials, and the existence of this high quality medical infrastructure, all those ingredients were required. The fact is, you now have all of those ingredients. Because of the 2030 initiative here, you now have the government role of incentivizing this collaboration. And I can tell you that with the institution you have, the infrastructure that you have, and now the government support that is forthcoming, that private industry, players like me, are standing up and taking notice and saying, we want to be involved in that process. Yeah.
Great, great. Well, let me say, if you face this problem in Poland, one of the major obstacles that we are facing right now in KMARC is attracting investigators to do clinical trials. And we know our clinicians are doing a great job with their patients, but unfortunately they don't have enough time. I know the research prototype time is not applicable in our system, but how can we overcome this challenge by attracting more? I mean, incentives, money-wise, is not enough to incentivize the investigators. So what could be done better to attract more investigators?
So I totally agree. Incentives alone are not enough. They need to be there. That's an important component, but it's not enough all by itself. What I like to say is there needs to be the absence of resistance, okay? I'll use the U.S. as an example. You know, the FDA has a very important regulatory role to play. Very important. Unfortunately, though, the government mandate to our FDA is make sure that sponsors like Moleculin don't do the wrong thing. Well, it's fundamentally a negative message. In other words, you need to be the watchdog to make sure nothing bad happens. Okay. Instead, if the mandate were your job is to help ensure that new drugs are developed safely, that's very different. I'm saying the same thing, but from the positive aspect. In other words, make the regulatory authority a collaborator in this process. You have a unique opportunity to do that. I believe the SFDA, in fact, when I was here 3 years ago, within one day, after meeting the great folks in KMARC, in the same day I met the head of the SFDA, and in addition to the head of several government institutions that are there to support economically what's going on. So there's already this collaboration within the country. I think if you make sure that the mandate to the SFDA is to become a partner in this process. Yes, we have to be safe. Yes, we have to follow the rules. But if we have a partner that we can reach out to, that's not a black box, that we can pick up the phone and say, we have this issue, can you help us solve it? That's very different than saying, I need to talk to the FDA, but I have to make a submission and wait 30 days before I can even ask a question, right? It's just a difference in approach. And I think Saudi Arabia has the opportunity to get that right where we got it wrong.
Well, I'm not sure if we have representatives from Saudi FDA, but speaking on their behalf, they are supportive in terms of the bylaws and expediting process. Sometimes they give some exemptions, but yet, again, I totally agree that there are some areas that need to be improved in terms of the approval process.
Right. So, as an example, I mentioned that last year we presented a proposal to the royal family. That proposal included some very specific suggestions. For example, a standardized contract, clinical trial contract across all hospitals, across the entire country. We could never get that done in the US. And I can tell you, I have seen clinical trials held up for almost a year because of negotiating a contract with a hospital to get the job done. You have the ability, because you're a kingdom, because you're so cohesive, to say, nope, there's going to be one contract. And it's standard across all the hospitals, so we only have to do this one time, right? Instead of 15 times for 15 different hospitals. In addition, streamlined application processes where reviews happen in weeks instead of months. And when there's a question, it's a live question. You can have real dialogues with real people to solve whatever questions are out there. Those sound like small things, but when you add them all up, the average time to begin a clinical trial in the US is really about 9 months. And often it's more than 9 months from start to, from the initial request to start a trial to when the trial's actually started. That's terrible. I mean, think about what we discovered with COVID, right?
Mhm.
I mean, in the case of COVID, everybody stepped back and said, we've got to solve this problem together and we have to work together to solve it. Even the FDA and the EMA. Everybody sort of joined hands and said, we're in this together, let's get the job done. So all of a sudden, all the nonsense about 30-day review periods before we can ask a question, all that just got pushed to the side, right? I'm just suggesting that the same thing we did for COVID, we should do for cancer research and for diabetes.
Right.
I mean, for all indications. Why would we just single out COVID and say we're only going to be efficient for COVID-19, but then we're going to go back to being inefficient for all these other diseases? I mean,
it does not make sense.
Yeah, it's crazy.
I totally agree. Well, we still have time. We can engage our audience. If anyone has a question for our panelists. Yes, go ahead, please. I have the mic.
Thank you. Steve Lockhart. I'm with Pfizer. I spoke earlier about vaccine trials. Can I just say I agree with absolutely everything you've been saying. I think a key factor that we look at when we choose sites is startup time. And that's an aggregate startup time. It includes everything from regulatory, site management, contracts. My other thought is that one thing that maybe would be an advantage here is dedicated site management resource. Because you're absolutely right, busy clinicians don't have time, and trials, frankly, are a pain. There's a lot of paperwork and data entry, and it has to be done very well, and it will be audited. So it has to be done very, very thoroughly. So I think a dedicated national site management organization, be it commercial or state managed, would make a big difference. Thank you.
Thank you very much. I think this is a great suggestion, and we can adopt this suggestion to have a site management organization. It's probably might be easy to do at national level, which I think it's one of the ways and maybe shortcuts to help in probably alignment of the institutions in terms of their processes, and also developing the ecosystem.
Right.
Yeah. Any other question from the audience? We still have 2 minutes. Yes, please.
I also agree, we're in violent agreement with respect to all the points that you were made. I would just make a comment and add further, and I emphasize this in my talk. We've entered into this era of precision oncology, precision medicine in general. I think it's extremely important for innovative clinical trials and for patient care in general that there be extremely robust profiling of patients, and not just pre-treatment, but longitudinal profiling of patients. And that we should include, at a minimum, genomic profiling, of course, but also immune profiling, to understand which patients are suffering from which mechanism and to enlist the right patient into those clinical trials. And that will dramatically, if you have knowledge of the issues in that patient, and a pharma comes to you, that pharma is going to know that you can actually then pick those patients and enlist those patients into the right trials.
Thanks, Ron. You know, I'm really glad you brought that up. I would add to that one of the recommendations that we made last year, in fact, Ron, you were instrumental in making sure that these recommendations got in front of the right people. One of the recommendations we made was to create a central database. The Royal Marsden in the UK, you may have interacted with them before, so I may be speaking at a school here.
We are part of the same academic health science center.
So as a sponsor, what was so attractive to me, so impressive about them, is they created a central database of all their known patients. And again, respecting GDPR, but within those guidelines, they were able to essentially tell us almost instantaneously how many patients they had access to that had the right genomic profiling, that met the inclusion-exclusion criteria of our trial, and we were able to estimate on the spot, within a day, make a real estimate of recruitment rate from that institution. That kind of centralized database goes hand in hand with what Ron DePino was saying in terms of utilizing data to make better decisions about who to include in a trial and who not to. So I'm very much about speed. I think hopefully I've communicated that. But what I would say is it isn't just about speed, it's about quality of execution. And by being best in class in all of these practices, I think Saudi Arabia has a chance to really nail it in terms of quality of execution, as well as being the fastest clinical trial location on the planet. So
Agree.
Yeah.
Time is up. If you
Just a point, just to say that when we think about clinical trials, we tend to think of patients in a hospital.
Yeah.
Obviously, there is something where epidemiology can contribute. We, for example, in London, and we are part of the Marsden is part of our academic research center. We create a database which is called Chariot. We registered 40,000 NHS persons through GPs. So we have a database of 40,000 people who have done a pre-consent to be part of trials. Trials not on the serious disease diagnosis in hospital and so on require hospitalization, but trials, for example, on cognitive decline, hypertension, glycemia control. So that is a component which I haven't seen touched for the moment, where I think here with the Saudi Biobank, the kingdom could be in a prime position, because by collecting data on tens of thousands of participants, of people living in Saudi Arabia, with their data, you might have the underlying population from which invite people with particular conditions that might be the object, might be the ideal subject of a trial. So the population-based, I would say, drug testing.
Perfect.
Well, with that, I would like to thank our panelists for their great contribution and sharing experiences, and thank you very much for your listening.
Thanks, everyone.
Thank you all for this fruitful, informative session. It's always great and valuable to hear from industry leaders. In continuation to this session, our next panel looks at the clinical trials, real-world evidence, real-world data, and artificial intelligence. To start, it's my pleasure to welcome co-chairs Dr. Ra'ed Al-Hazmi, Executive Director, IT Department, Ministry of National Guard, Health Affairs. And Dr. Lamia Al-Amir, Head AI and Bioinformatics, KMARC. And their esteemed panelists, we have Dr. Adrian Cassidy, he's joining us virtually, and Mr. Torben Sandgren. Welcome to the stage. I pass the mic to you, Dr. Ra'ed and Dr. Lamia. Thank you.
Assalamu alaikum. Good afternoon to all of you. We have a great session for you. It's session 8, clinical trials, real-world evidence, real-world data, and artificial intelligence. We're going to start with Dr. Adrian Cassidy, who's going to join us virtually. Dr. Cassidy is the head of global evidence generation at Novartis, which aims at advance the use and application of integrated evidence solutions across the life cycle of pharmaceutical development. Dr. Cassidy is an epidemiologist with 20 years of experience building and leading data analytics, evidence teams, and platforms, an academic, public health and industry settings across international markets. I'm going to pass it to you, Dr. Qasdi. The floor is yours.
We cannot hear him.
At this meeting, and especially very happy to follow the previous session, which I think I heard the panel really interesting discussions on advancements in clinical trials. So for this session, we'll switch gears a little bit and talk a little bit about what we see as the future state for real-world evidence and integrated evidence. So next slide, please. So what is the difference between real-world data and real-world evidence? Well, the real-world data are actually the data that are underlying RWE. And the real-world data is translated into evidence by essentially applying appropriate analytical techniques to address a specific question. So the typical sources of real-world data would include data that's coming from electronic health records, from claims, disease registries, or even patient-generated data through apps or surveys. And of course, the increasing interest in real-world data is because it's becoming increasingly increasingly rich with clinical and genomic data, as well as increasing in quality and veracity. So real-world evidence is essentially the translation of that data, and real-world evidence can be generated using a variety of study designs, such as retrospective or prospective observational studies, and really thinking more in the realm of advanced analytics, how we are applying advanced analytics, machine learning, AI in real-world data settings. Next slide, please. So real-world evidence actually complements the evidence from randomized clinical trials. And to understand this, we first have to say that, of course, randomized clinical trials provide the highest quality evidence and essentially are the gold standard for evidence-based medicine. However, we also know that patients who are recruited to RCTs are highly selected, and also their management is really tightly controlled in a defined protocol. Therefore, RCTs are not necessarily reflective of real life and are also difficult to generalize into routine clinical practice. So in this context, real-world evidence actually complements and can also supplement findings from randomized clinical trials, and can, of course, provide evidence from larger, broader patient populations where patients are treated in standard healthcare practice in normal settings. And so whilst we recognize that real-world evidence is less robust or rigorous than the results from well-designed RCTs, it has high external validity because it targets a broader patient populations and generally more representative of how patients are managed and treated in real life. Next slide. So, the timing for this talk is excellent because, as you can see from this slide, many global health authorities are issuing guidances and frameworks to help support decision making, particularly in a regulatory setting, using real-world evidence. So specifically, regulators are exploring when and how to use real-world evidence and how it might be appropriate for regulatory decision-making. And I'm going to give you an example later in a few slides of how we at Novartis have actually used real-world evidence to gain approval for in a rare disease setting in a new indication. So, whilst I talk about real-world evidence, actually what we need is truly integrated evidence, because we know that as the healthcare system evolves, our diversity of stakeholders also demands that we diversify our evidence to ensure that we meet their need. So we have multiple needs from both health authorities, payers, healthcare professionals, as well as patients. So, at least from our perspective at Novartis, we achieve this by switching our strategy from essentially what we initially focused on in real-world evidence to basically this integrated evidence strategy, which is essentially aimed at providing a holistic evidence package for the range of stakeholders that require this evidence for our medicines. Next slide, please. Sorry, next slide, please. In our organization, we've done this by creating essentially a unified strategy to ensure that we both are able to generate the right evidence and also have the infrastructure to do that. So there's essentially three pillars. One is where we really take this approach to actually integrate evidence or evidence approaches right throughout the life cycle. And that can be from informing clinical trial design to actually innovating clinical trial design, where we would include a real-world arm in that study for long-term follow-up of patients. Or we think about tokenizing clinical trial patients in which to actually generate long-term outcomes on those patients. The second approach, and second and third pillars are also equally important. You have to also have the right data to ensure that you can generate rigorous and robust evidence, and that can be achieved through a number of ways. And the final aspect is essentially the analytic platform in which that sits, on which it allows you both to hold the data, but also work with external partners, as well as really conduct advanced analytics that translate these data into rigorous evidence. Next slide, please. So this will be my final slide, where I want to show you really an example of all of this coming together. So I mentioned earlier that actually real-world evidence can be used to provide evidence of clinical benefit for new therapies. And we've particularly seen this for rare diseases or difficult to treat populations. In this example, we actually, it was in a group of patients that for a very rare disorder, it's called PIK3CA-related overgrowth spectrum. And in these group of patients, we were treating them with a licensed drug in a managed access program. And within that managed access program, we already saw a benefit for the treatment. So we actually engaged with FDA and asked whether, or submitted a proposal essentially, whether they would accept the real-world data from this program as a demonstration of effect for these patients. So in that context, we then conducted a multi-country study to essentially extract the information from the patient's files. And this was the evidence that was submitted for approval. So we received approval for this medicine that's called Rejoice in April last year. And it really is one of the first times where we've seen real-world evidence being solely used for approval, at least in the US. So this was for FDA. So, with that, I want to end there because I'm conscious of time. What's really, I guess, important to leave as a key message for this audience is that really integrated evidence has the potential, from my perspective, to transform patient care. We see multiple applications, not just from a research and development perspective on targeting therapies for treatment, but we see multiple applications both for health authorities, for regulators, for payers to understand the effectiveness of treatment in a real-life setting, as well as healthcare practitioners and ultimately patients who, at the end, we're really driving the use of these data and evidence to improve their outcomes. So with that, I really thank you for your attention and look forward to the discussion.
Thank you, Dr. Adrian, for this insightful presentation. And we need more information in the panel discussion. Now, Dr. Ra'ed Al-Hazmi will give us his speak. Dr. Ra'ed Al-Hazmi is the Executive Director of the IT Department in the Ministry of National Guard Health Affairs, and he is the IDC CIO of the year 2021. Dr. Al-Hazmi has been involved in the management of the IT department since 15 years right now, and by his lead, the Ministry of National Guard reached quadruple stage 7, highest level in HEMS digital maturity model as the only healthcare organization in the world to achieve this. Academically, he is adjunct professor, assistant professor in health informatics department, King Saud bin Abdulaziz University for Health Science in Riyadh, and also adjunct professor in biomedical informatics department, Nova Southern University in Florida, United States. Dr. Ra'ed research actives as a focus in digital transformation, health informatics, and data science, which involve publishing number of scientific literature and registered 4 patents in the United States. Dr. Ra'ed, the floor is yours.
Thanks. The potential of AI in real-world evidence, and AI requires a lot of data. And over the last number of years, we have been generating a lot of it. There's no shortage of data. According to some sources, the data that was generated, 90% of the data since the dawn of humanity was generated just in the last 3 years. Other sources were talking about the data that was generated in 1 year, 2019, is more than the data that all humanity has generated over the last 5,000 years. And what's basically challenging about this data, we're generating a lot of it, but what's challenging is most of it is unstructured and semi-structured data, very difficult to deal with, very difficult to analyze. But the good thing is we have big data technologies that would allow us to tap into this huge amount of data that gets pumped in from many sources and would allow us to handle what we call the 4 V's, the volume, velocity, variety, and veracity of data. And it has been proven. There are a number of entities that employed big data at a very large scale. It used to be in the past that even if you scale out your resources, we used to call it scale up, which is ending up with larger servers and larger back-end equipment. It used to be that you reach a point where the performance and the capacity does not go up, no matter what you do, to the point that the whole thing just collapses. But with big data, it's a linear kind of increase. You add more resources, you get more capacity and more performance. And we need this technology badly, now and in the near future. there are a lot of new sources that going to add and generate a lot of data. Take, for example, the health passport, which it was experimental and was implemented at a small scale in some countries, but this will be increased in many countries to include not only the providers' data, hospitals, primary healthcare centers, et cetera, but also IoTs, Internet of Things. You have consumer medical devices that patients are going to have at their homes with the increased adoption of remote patient monitoring, RPM, that's going to basically generate a lot of data that you have to store somewhere and you have to use for transactional, probably within the hospitals, regulators, and other transactional purposes, but also for research as an RWE. And there is also the digital twin of the person that was implemented, again, still experimental, according to the hype cycle of Gartner and other sources, but it will be expanded. You're going to end up in the near future with an exact replica of the patient, a digital replica that you could simulate safely and you could experiment with, and then once those experimentations are successful, you could implement in a safe way and accurate way in the real world. And all of these are going to open the RWE floodgate. The old way of obtaining the data for research and even for transactional purposes are going to be obsolete in the end. And that's probably going to be more futuristic here. But at some point, probably, and we saw this with some technologies that are available nowadays, we probably could test multiple hypotheses at the same time, not just focus on one and just gather data and just do the analysis and then work on the publication, which takes a long time. And the findings of those kind of studies would yield not a lot of outcomes. You could analyze the data fast, maybe automate it in an automated way. And then you could maybe even automate the publishing process. Those of you who were experimenting with ChatGPT, one of the generative AI models, myself, I was very impressed. You just give it a couple of lines, and sometimes it's more of a scientific lines for the AI to write about. And me as an informatician for around 20 years now, I can distinguish the writing here, which is about the impact of telehealth on patient care, with citing some citations, although the AI was a little bit cautious and told us that it's not trained on publications and literatures. But the writing is amazing. But this shows us the future, obviously. If we train this generative AI model on the publications and also on raw data, it might be able to tap into this raw data, come up with hypotheses, and by the way, you could ask it about hypotheses right now. It could come up with some of them, but cannot proceed further. And it could analyze the data and maybe write some publications with some citations. That's probably the future. And we probably have to cope with the future that would come, and all of the indicators are indicating in this direction. Thank you so much for listening. And I'm going to hand it over to
Thank you, Dr. Rad.
Dr. Lamia Al-Umair.
Yes.
Dr. Al-Umair is the head of AI and bioinformatics at King Abdullah International Medical Research Center, KMARC, and an affiliate faculty member at George Mason University in the United States. Her work has been featured in several international media channels, including the first edition of MIT Technology Review, where in 2022, and due to her expertise in AI, she was picked as one of the top 30 leading Arabs in AI. Dr. Ahmed, the floor is yours.
Yes, thank you. Thank you for the introduction. Thank you very much for attending here and staying until this time. It's Thursday night. Today in this presentation, I will explore with you how these two technologies, AI and bioinformatics, support and help clinical trial. couple of decades ago, we have a new science called computational biology, bioinformatics. This is coming due a lot of information that we got it from biology information. We got the DNA sequencing, the structure of protein. So we have to have this computational biology science to help us to understand the new era. And when we merge it with the AI, I'm sure all of us know the word AI, and usually I have this introduction because most of the conference I go, it's medical conference, and people come from the medical background. They used to have the call AI, artificial intelligence. I like to say it's a computer intelligence. It's not artificial intelligence, which is using algorithm from the computer and try to analyze it in a way that the human mind is thinking, but in a faster way, accurate way, and give us more insight than what we can give in a time manner. In AI, we have different algorithm and different technology that we can use. We can use machine learning, which is we can train the machine of different information, and we have a natural language process, NLP, which is also in the text information, and we have computer vision. I'm sure a lot of you during the pandemic, we have the vision of the computer, and we have the voice recognition also technology. We have the expert system that we can use also. Robotics that mainly can simulate the muscle of the human and try to do a lot of action planning and helping human to analyze and make decision making. So what is clinical trial can have impact? The combination of the AI and computational biology is already happening and transforming and having impact in clinical trial. By taking the advantages of the computational biology and AI, we can accelerate the time, and especially in the drug development in the beginning, and managing and recruiting patients, and end up of analyzing the data. So there is some application in AI. AI can use to from identification the drug discovery, as I told you, accelerate, finding a drug target, and choosing the right patient for the right crew. Now we can use all human choosing, and this is taking a lot of time. By having health record and by having AI, we can recruit patient very fast and very efficient. And also we can analyze the data, choose the right drug for the right patient, cognitive information that we can manage to merge all the information and get the right information that we need. Also simulation and in silico. Now from anywhere we can get to the lab using in silico information. You don't need to have the wet lab. We can do it in silico. This will minimize the cost and will make us much more efficient. As my colleagues, Dr. Raed, talk about the digital twin. Digital twin is now the new evolution in AI. So they have the human body copy, and the clinical trial, they can do the test for the new drug on this digital without harm for the patient. And at the end, analyzing all the information that we got it from the clinical trial and have the right decision. The impact in AI, as I mentioned, so it will improve the accuracy and the speed. It will improve clinical decision making. It also will increase the efficiency and the cost efficiency, especially here with Saudi Arabia. We try to minimize the depend on the oil, and we are trying to increase the depend on the data. Using the data will increase the economy in Saudi Arabia and achieving Vision 2030. Also, it will simplify the work instead of having much of work, just analyzing this data, choosing this data. Now, the scientists will take more time in advancing the science, and it will simplify their work. Absolutely, there is some challenges like the data security and privacy and regulation and ethics that is we have to look at, and sometimes limitation to access some AI technology like the cloud for right now. So to have this, we have to have an effective strategy. Effective strategy is starting with building a strategy that, first of all, we have to know which task that suitable for the AI application. We have to approach the data collection to see the right data collection, and also effective collaboration between human and the system. Since we have this strategy aligned together, then we can involve training the AI system with the human, and then we can achieve, set up the mechanism of AI clinical trial driven. In the last slide, I would like to talk about the future of the AI. I was reading a book, it's called The Future is Faster Than What You Think. So we have to be ready. AI is and computational biology in clinical trial just beginning. And as more as organization will adopt the AI technology, the opportunity to improve efficiency and accuracy will be increased. Saudi Arabia adopting AI-friendly ecosystem by adopting 5G and security data access. All of these information to improve the innovation and clinical trial. And at the end, thank you very much.
Thank you, Dr. Al-Amir. Now we're going to hear a speech by Mr. Turban Sandgren. Mr. Sandgren is the CEO of UN EEG Medical. And prior to this, he was the VP of Global Sales, Marketing, and R&D at Convatec Group and Ono Medical, Inc. Furthermore, Mr. Sandgren has held international leadership roles in Coloplast AS in China and Denmark, and BP Carl Oil in Denmark and Austria. He holds a BCom in Marketing Economics from Copenhagen Business School. Mr. Sandgren, the floor is yours.
Thank you for the kind introduction. And I will also just wait for my slides to be ready. I will be the one to talk you through a case how to implement real-time data and evidence by means of a small brain chip. Just a little bit about our background. Here we are. We actually started the company 10 years ago, where we got the CE mark for our solution in 19, just before COVID went berserk. We are employing 125 people and actually owned by the families behind the third biggest hearing aid company in the world, WS Audiology. So that's basically where we are getting a lot of our know-how about technology from, electronics and so forth. There is an unsolved problem. We actually have a quite large group of patients whom we are letting down. It's people with uncontrolled epilepsy. They are basically not helped, not even with medication. And we have addressed that by means of big data. Today, 1% of the population are having epilepsy. And 30% of those are actually having uncontrolled epilepsy, where they are drug resistant and they are basically left alone with a quite poor life. They are actually among the worst cases within the medical field. Today, we know that people are offered medication. We know that they can go to the hospital, admitted to the hospital, and have an EEG. But honestly, this is not real-world data. It is in-hospital data for a couple of hours, maybe a couple of days. So what do you do? Yeah, from half an hour to maybe a couple of days. So we are actually inventing a system which is now in an IDE study in United States as a de novo, and we already, as you can see, got the CE mark, so we are now distributing this technology in 10 countries in Europe, meaning that we are achieving a lot of real-time data, which we are then transforming into real-time evidence. This is the system. I'm standing here with a little device, the implant, which is slivered under the skin just behind the ear. You have a logger. The logger is recording up to 1 month of 24/7 brain data, not loop recording, 24/7 data. It is then transferred by the patient to the hospital via our cloud, and then the physician is able to sit and look into up to 1.5 year data at the same time. And the screen shown here is actually having 1 year and 3 months data if you take the upper line. So this is a totally new way of giving objectivity into the treatment of severe epilepsy. So what are we talking about? We are talking about not deep brain or anything. It's just subcutaneous. It is just behind the ear, and then you are having a small wire going down to a recorder, exactly as I'm wearing a wire now with a microphone. And it's a very small, minimal, invasive procedure. It takes 15 to 20 minutes under local anesthesia, and then you are basically discharged from the hospital again. So it's a little bit the chicken and egg, because we had to implant quite a lot of patients before we had the data for making our advanced AI-supported analysis. So we actually got, as a starting point, 60,000 hours of data from the classic system, and then we got another 60,000 hours of data from our own two-channel system, from the temporal lobe area. And then we were actually able to establish an advanced algorithm actually annotating a person's objective seizures for months, for years. So that's quite extraordinary. And that means that the patient, the physician is able to adjust medication based on facts and not feelings from a diary. It's fantastic standing here in Saudi Arabia. We have had a strong focus in Europe and in the United States because of the FDA. I'm just mentioning some of the hospitals where we are having our cooperation and implantations taking on, and that's, as you can see, quite well-established organizations. So I think that was my time. Thank you very much for listening.
Thank you, Dr. Turban, Mr. Turban. So thank you very much. Now we'll move to the panel discussion. I will assure you it will be light questions. I will start with one question, maybe Dr. Adrian will answer it. For the real-world evidence and real-world data, how it will contribute for optimizing the clinical trial and achieve the outcome?
Yeah, no, thanks for the question. I think that there's already been actually really nice examples for where real-world data will help optimize the trial and improve outcomes. For sure, the data, because it is broader and also a wider understanding of healthcare practices, it can help inform trial design as well as define outcomes. But equally, the real-world data can also be used to recruit patients into trials. And if we think about how trials are also evolving in a more decentralized way, especially since the pandemic, elements of decentralized trials are essentially moving away from the traditional approach of actually more like in the real-world data sense rather than the RCT sense. So, I think that from our perspective, we have, from a pharmaceutical perspective, we use real-world data a lot in the development of drugs. And this is either to better understand the patient's medical need, their journey through the healthcare system, the elements within that healthcare system of why patients are not being treated. And all of these evidence actually also are very beneficial and also can improve the probability of success of clinical trials. So this is how we think about also using data from a pharma perspective to improve clinical trials.
Thank you, Doctor. Maybe Dr. Raed, you can elaborate on this question.
Yeah, if I could elaborate on Dr. Kazdi's point. I mean, using RWE is almost like bridging the chasm between the real-world delivery of service and research and having the cycle more seamless. It used to be you have the delivery of service is done, and then maybe you plug to it in a separate way, a way of obtaining the data for research. Then you take the data on the side, you work to analyze the data and do some publications and come up with findings, and then feed this into the transactional realm to the care delivery of the patient. I think nowadays this is dysfunctional, and this should not continue. I mean, you could have the continuum going seamlessly. You tap into the real-world data from multiple sources, and then you do the analysis based on certain hypotheses, and then you come up with findings, and maybe in a very fast manner, you could feed this into the delivery of service in a much faster way. And I think maybe an added advantage to the use of RWE is also the accuracy of data. If you obtain the data from what people are documenting, the clinicians, the providers, definitely that's going to be much accurate than you have a process on the side that tries to solicit what was done to the patient, what are the outcomes that the patient basically was having, and then you take this on the side, which is almost like a copy, but it's not an exact replica of the transaction and what was done to the patient. So these are just a couple of maybe advantages of using RWE as a development when it comes to research.
Okay, so you mentioned that having the RWE from different resources, this is maybe one of the things that the bias will be there if we take it from one source?
Well, that's one source, and I think in the near future we're going to keep adding to the sources. According to some studies, RPM, remote patient monitoring, the patient that had access to remote patient monitoring had much less mortality rate during COVID-19. It was 6% and compared to 20% for the patient that didn't have access to RPM during COVID-19. And with these advantages, just by having these advantages that impact outcomes, you're going to have more and more adoption of home use medical devices. You're going to have IoTs all over the place. You're going to have the smart devices, the wearables, all feeding in data into the reservoirs of RWE, so you could tap into this and harness this useful data.
Perfect, thank you. So now technology-driven is everywhere right now, although human is the center of this. Using and utilizing AI in clinical trial. So Mr. Trubin, there is an issue of ethics and regulation. Can you give us some of your experience in this field?
Yeah, it's a great question because we, especially being from Europe, governed by the European Union, we have the so-called GDPR, where we actually have to ask for a kind of strict consent for whatever we are doing. So if we are asking for access to a patient's data within, for example, seizures, we are not automatically allowed to use it for other purposes, like analyzing sleep patterns or other biomarkers. So we have to get a very firm consent from each patient, otherwise we'll end up in quite a difficult situation as a company. So it's governed pretty tough within the European Union, and that means that when we are talking about real world data, we have to be very clear about what we would like to use it for strategically, because we have to ask for that consent upfront. If you're not able to get it anonymous, and that's not always the case. So I would say we are spending quite a lot of time on considering the ethics within this.
Is there a new regulation can be adopted to expedite the using of the data?
Sorry?
Is there like any new regulation maybe in Europe? So
I would say that we are trying to cluster the data we need and to understand, and then we try to make it in a way so it can be anonymous, so it's not a person related. And then we are also negotiating with the authorities how strict it should be, you know, from theory to practice. So that's the focus we have.
Thank you. Dr. Raed, as an IT Executive Director Department, can you elaborate on this question, how we do the ethics and regulation of using the data for clinical trial?
There should be actually a lot of regulations that protects the patient privacy. And the last event of Davos a couple of days ago, one of the questions that they were basically asking and discussing is how do you protect the people's privacy and also the intellectual property with the increasing adoption of generative AI. With generative AI, I mean, you have data that's available on the web without getting consent from people. You train the model based on it, and then you generate findings. And this changes the way probably research would do business in the near future. In the past, or until now probably, you would need to get consents after you get the IRB approval, and then you start collecting data. It's going to be the other way around, obviously. You're going to have this data lake, and that's going to be your RWE, and maybe you're going to get the consent prior to getting the data and the data leak. So you're going to get clearance from the get-go to start the research process. So I think it's going to be almost like a paradigm shift in the process itself.
Yeah, I'm sure this will take a long time to discuss. So I will have this executive summary from the AI and ethics regulation. I'll come back to you, Dr. Adrian. I'm sorry, I didn't lift you back. I just want to know what is the health and economy impact in real-world evidence and real-world data? Can you elaborate on this?
Oh, sorry, you're speaking to me. Sorry, apologies.
Did you hear the question?
Yeah, I heard the question.
Okay.
Yeah, thank you. I think from a health perspective, I take that aspect first, and I take the example that I presented on how we used basically real-world data in a rare disease setting to essentially gain regulatory approval. And now we know particularly real-world data has good utility for rare diseases, because these are also the settings where it's less feasible to conduct a large-scale trial or a randomized trial. And so in this setting, in this particular setting where these patients were suffering from a very rare overgrowth syndrome, the use of real-world data to demonstrate a benefit of an existing treatment actually enabled those patients to access an effective therapy that they probably would never have been able to access because a clinical trial may not have been feasible. So that's a very specific example of how you can also improve health outcomes, specifically for rare diseases, by using evidence, real-world evidence, in a robust. The other aspect. Oh, sorry. Go ahead. I'll leave it there. Maybe let someone else speak.
No, it's okay. You can continue. We can hear.
Oh, Yeah, no, sorry. Okay. The other element to that is, I guess, the economic aspect. And here, again, coming from a pharmaceutical company, you think about the element of enabling evidence that enables value-based healthcare, right? So where you can really demonstrate the effect of your treatment in a broader patient population, that it's effective and safe. And in this setting, when you can do that, then health authorities and payers, reimbursement agencies are actually quite willing then to pay for the therapy, right? But you have to be able to use the data to also demonstrate that it is effective. So I think this is also where I would come from the economic part, because at least from an industry setting, this is critically important of how we would do that.
Well, thank you very much for this insightful information. So before we have a question from the audience, Dr. Adrian, do you want to send a message in 1 or 2 minutes before we take the question from the audience? Dr. Adrian, do you hear me? Just a takeaway message.
Oh, yeah, sorry. Yeah, sorry, I'm having problems with my connection. Apologies for that. Yeah, maybe takeaway message for me is I've worked in this area for around 20 years. I've never been more excited by the opportunities that lie in front of us for using real-world data and evidence, as well as the advanced applications to that, to actually improve patient outcomes in healthcare. So I see, huge opportunities to do that in different settings. And yeah, I think that one of the things that I'm particularly excited about, we talk a lot about the differences between real-world data and randomized clinical trials. I think in the future, and this is also links to what was also said earlier, I think in the future we're going to talk about data, right? And data is becoming increasingly available, increasingly better quality, and there won't be this distinction, I think, anymore when we think about data.
Yes, or they say, thank you very much. They say the data is the new oil. Dr. Raed, any message before the question?
Yeah, I mean, maybe complementing a little bit Dr. Adrian's statement. I mean, things will change a lot. The way we do business and research, obviously, will change significantly, and we have to cope with this in the near future and also the far future as well. There are regulations that have to be put in place to protect intellectual properties. One example of this, I mean, if you train a model similar to ChatGPT on all of Wikipedia and many of the libraries, books, and a large portion of the internet, who is going to own what the model produces? You could actually write books using ChatGPT, but ChatGPT itself did not come up with all of those ideas, did not come up with this intellectual properties. So the question is, I mean, who owns what? Is it the entity that developed those AI models? Is it the original authors of this intellectual property pieces? Again, it's a big question, and many discussions are happening. I highlighted the discussion that happened in Davos in the last couple of days. So that's really one portion of it. The other portion is we have to cope with how do you handle data and monetize data. It used to be that you have the data and you give access to certain kind of entities for fees. Now, all what they want is just one-time access to all of your data, and they could generate as many as they want, hypotheses, and analyze the data in a different way and publish countless number of literatures based on the data. So maybe not monetizing the data itself, but having some sort of a shared kind of profit in the process if you want to have access to the data using those advanced technologies such as generative AI. So all of those aspects have to be put in place for us to cope with what's going to happen in the near future.
Thank you, Dr. Raed. Mr. Turpin?
Yeah. I think we will in the future see a much bigger demand for the biomarker, the brain. We are used to get a lot of data from Fitbit, other stuff, but we have actually never got ultra long-term knowledge from our brain. And we know that Elon Musk is trying with Neuralink to support the brain by AI, but actually imagine the brain over time to understand what is going on from the source itself and not indirectly. Imagine what that could bring to drug development, that you are actually having evidence of what you are doing over a long time. You can correlate sleep patterns, not from a wristband, but from actually the brain. You can add a lot of knowledge from the brain, and we haven't even started yet. So I think the brain will be the source of big data, and very reliable because it's directly from this one.
Interesting. I just want to say, no matter our world become technology-driven, human will play a very crucial and important role in this. So we will not forget that we are human who is the one controlling this technology. I think we have 2 minutes for question. Yes.
Not 2 questions.
السلام عليكم.
السلام.
مهندس ماجد غيمش. الحقيقة أنا مش كويستشن عندي، أنا عندي كومنت وأبغاكم تسوون كومنت على ماي كومنت. أنا أول شيء أشكر الباناليست على their outstanding presentation, and my question is macro picture. Now you are talking about the big data and the AI and the contribution in the medical care. We know that the medical care in the global GDP is around 7%. So global GDP is around $80 trillion. Now we are talking about the digital economy. And digital economy is completely different from the knowledge economy or the green economy. So there are technologies implemented in this digital economy, around 20 technologies, including AI, blockchain, IoT, and so on. From the macro picture, there is no law internationally governs the digital economy so far. And the idea behind it, there are 2 obstacles: the residency of data and the sovereignty of data. And this is not resolved yet and too difficult to be resolved. This is one. Second, the criteria for these technologies, there is no criteria approved yet for only one technology, which is IoT. And these criteria is adopted from 3 organizations in the world, which is the International Telecommunication Union and the ISO, International Standard Organization, and the International Committee, Technological Committee. They have a joint technical committee to approve any technology. So yet, the IoT the only technology. So the cloud is not approved yet. And there is no criteria for that, and there's no law is governing the digital economy. Second, Dr. Lemia, she said one slide about the challenges, and she put 3 challenges, and these challenges really is still challenges. And Dr. Raed, he says that in 2019, that the data created is more than 5,000 years, and that's true. So the idea, which data we need. So maybe less than 1% of the data we use is analyzed. So when you use the AI, the idea is to have the data that is refined. You have crude oil, this is an oil, but if you want to use the fuel for the car, you need to refine it by a refinery to benzene. So at the same time, the same model for the data. So now what I'm saying, how can you handle these challenges? Still, we are in a mess in the digital economy, in the digital world. The security for the data is not resolved yet and is not secure. So now in Europe, when they say about this, the sky, the cloud, they have a project called Gaia X, because all the European companies or the government, they use their data in the American data and it's controlled by the American law. So this is a big risk for the Europeans, as the same for the whole world. So how can we handle this dilemma? Thank you.
Thank you. Thank you very much. This is very insightful information that we got. We can discuss the answer after the session because actually our time is done. We'll come back to you and answer your question. Thank you very much for this, your time, and I hope you have a good Thursday night.
Such a great, lavish session. Thank you all. Ladies and gentlemen, what a productive long day. We have reached to the last session titled Genomics, Precision Medicine and Artificial Intelligence. We'll discuss in depth transforming science through data and artificial intelligence. To start, it's my pleasure to welcome the co-chairs, Dr. Majid Al-Fadl, Deputy Executive Director, KMARC. Welcome to the stage, Dr. Majid. And we have Dr. Patrick Tan now, yes, Executive Director, Precision Health Research, Singapore, and their esteemed panelists, Dr. James Weatherall and Dr. Madhuri Hegde and Dr. Philip Larson. Welcome all, and we look forward to hear your insightful views. I pass the mic to you, Dr. Patrick.
Hello, I hope that people can hear me. Firstly, thank you for coming to
absolutely,
this.
and likewise, absolutely. And let me give you my business card then.
Oops. Wow, okay. Anyway, back to the start. Thanks for attending this session. It's going to be a very exciting session on genomics, precision medicine, and artificial intelligence. I would like to thank the organizers for inviting us to this very important summit, and also to the audience for staying with us until this is the last session of the conference, but it will be the best session. It's going to be a bit of a different session because, believe it or not, we actually don't have any slides to show. So what we want to do is to actually have an open conversation about the challenges, the opportunities for genomics, precision medicine, and AI in healthcare and biomedical research. We have a very cosmopolitan mix of individuals from practitioners, big pharma, biotech, as Philip will explain, and from the public government areas in different types of countries. And what I would like to do is to also ask the audience, if you have any questions, please raise your hands, and then we would like to have this as a very open conversation with all of you. What I'd like to do now is to ask each of the speakers in turn to maybe introduce themselves, say who they are, their passions about the area of genomics, precision medicine, and AI. And then after that, we will go in and have a series of question answers. We also have a speaker from overseas, Dr. Hegde. Thank you very much for dialing in. I don't know what the time is over there. So maybe we'll start off by the sequence. Can I ask Dr. James Weatherall to give us, say a few words about himself and what draws him to this area? James, over to you.
Thank you, and pleasure to be here closing the summit after 2 exciting days. I'm Jim Weatherall. I'm head of data science and artificial intelligence at AstraZeneca, and as you said, we've got a mixture of companies and representatives here today. So I just wanted to start with some opening thoughts based on our experience over the last 3, 4, 5 years of really trying to do artificial intelligence at scale. So some learnings I'd like to pass on, maybe some tips, some pieces of advice. But anyway, it's some reflections. Firstly, I think reflecting back, it's important that we've taken advantage of the huge potential for artificial intelligence. So there's a huge potential based on the ever-increasing volumes and diverse variety of data that we're seeing right across the biomedical and health sciences. I think there's been a recent opportunity with availability of modern machine learning methods and high-performance computing. And I think also important is digitization and automation of lab methods. So now that we have the benefit of doing this for a few years at scale and looking back, what we've realized is that to truly transform the way we do science with artificial intelligence, you don't just drop an algorithm into your current process, you have to disrupt the process as well. So you have to bring those new lab assays, those new ways of doing science with AI, not just adding AI on top. So that's about harnessing the potential. The second point I was going to make is about the number of places we tried to implement artificial intelligence. So we looked right along the R&D chain of how we develop a medicine from idea to human impact. And we probed that process in as many different places as possible to see which ideas and which implementations of AI would work, whether it's using computer vision to identify biomarkers in images, knowledge graphs to enable better understanding of biological pathways, or reinforcement learning to help us design our molecules. We do all of these today, and it's because we trialed it out in so many different places to find out where AI works. And finally, the thing that we found really important was interdisciplinary working. So this is the human in the human-machine loop. Again, in the same way that you don't just drop AI into an existing process, you don't assume that AI is going to figure everything out for you. You need talented human beings to make it work as well. And the most effective way we've found of doing that is interdisciplinary scientific working. An example of that is an AI academic centre we set up at the University of Cambridge in the UK, which covers 6 different university departments and 2 different companies. And we believe that will take us to the next level because the diversity of thought will help us to produce the next generation of solutions. So just a few thoughts from me of having implemented AI at scale, harnessing the potential available, probing lots of different ways of implementing AI, and making sure you bring as many diverse thoughts to the table as possible.
Thanks, James. I think it's going to set the stage for different ways that AI can really disrupt the way that we are going to be thinking about how medical research is conducted. Maybe I'll pass it over to Dr. Majid Al-Fadal to introduce himself and to say what he does and how does genomics, precision medicine factor into his day-to-day work, which is very, very important, saving lives. Dr. Majid?
Yeah, thank you, Patrick. This is Majid Al-Fadal. I'm the Deputy Executive Director of KMARC and the Chairman of Medical Genomic Research Lab, and also the Chairman of Genetic and Precision Medicine Department at NGHA. My passion is the translational research and how to translate the patient need into research and then back to the clinic. So our philosophy in our departments is bench to bed and bed to bench, and how we could help our patient through the biotechnology and other technology found in the industry. We see currently the huge advancement in the medical genomic in the last decade, and we are going from a decade of diagnosis to a decade of therapy. And we are currently in the most exciting era of genomic medicine over the world. We are seeing discoveries of more than 20 to 30 gene therapy every year, and also we are seeing advancement in the big data of genomic and artificial intelligence as illustrated clearly in this summit. This is, I think, my background and my passion.
Thanks very much. I think we'd like to pick up some of the in the Q&A about how rare diseases is really one of the poster child for the application of genomic medicine. I'm now going to introduce our online speaker, Dr. Madhuri Hegde, and maybe to say a bit about, introduce yourself to the audience here, your journey and your passion about genomics and precision medicine, and both for yourself and for your company. Over to you.
Thank you, Patrick. Good evening, everyone. It's not a bad time here. It's 9 a.m. in the U.S. Okay. So it's a good time. Thank you for having me. My name is Madhuri Hegde, and I'm the Senior Vice President and Chief Scientific Officer for PerkinElmer Genomic Laboratories, global laboratories. And when I say global, I think it really sort of sends a message of where these labs are. The labs are placed globally. So I run genomic laboratories for PerkinElmer in India, China, Sweden, UK, and US. And that really gives us that exposure of doing genomic testing globally. I have been doing genomic testing for most of my career, about 25 years now. And I've seen this transition from doing genomics to omics. And we just heard that this is a decade of therapeutics now. So we have developed tools for diagnosis and diagnostics in general, and the genomic methodologies have helped significantly. But it is time to go from genomics to more omics-based approaches. In our own laboratories globally, we have been able to deploy a lot of new methodologies. But one of the key things which we have learned is having a multi-ethnic and a multi-omic approach is the best way to go forward. And if I just give the example of US, in US population is no longer a homogeneous population. It is a heterogeneous population. People from all over the world live in the US, which means that the samples that come to our laboratory for testing are from different ethnic backgrounds. And this is where AI and machine learning becomes really important. Having a global laboratory setting allows us to create a database from which we can draw information for interpretation. So it is really layering AI on the top of the entire database of different kinds, bringing it together, creating an ecosystem, which then can be used for, which can be then layered on or used for AI type approaches. This is really important because, talking of AI in isolation does not really go anywhere. The data that we generate and how we create that modular data for use for patient care is absolutely critical. So this has really been a very exciting time for me. My background is from academia. I have been at PerkinElmer for 6 years now. Yeah. So I've really worked in both in academia and industry, and that translational aspect is absolutely stunning with what is going on with global collaboration.
Thanks very much. I think you actually alluded to an important area that when we say genomics, it actually is a much larger phrase for omics, where we are capturing not just the genome, but the transcriptome, the epigenome, single microbiomes, proteomes. So I think that this is really going to be, and all of this will be the bedrock for studying new discoveries. I'm going to ask Philip to introduce himself also, and to maybe talk to him about how you see this field in the area of big pharma and also in the biotech, for biotech companies. Maybe Philip?
Thank you, Patrick. I'm Philip Larsen. I'm very pleased to be here. It's a great conference, and I'm obviously honored to be invited for this conference. I've been in the industry for the past 20-some years, and recently taken a step into lead a biotech company, Six Peaks, that's focusing on what you could call common people's common disease. And I've taken that step at this point in time because I think the time is ripe to really harvest all of the technological advancements we've seen in the past 10, 15 years to understand, more complex diseases in a far deeper way than we've managed to do previously. Especially in the area of oncology, but also in rare disorders, we've been used to live with the term precision medicine. We've recognized that in oncology, we're treating not just one disease, but actually probably 200 different diseases. The genomic revolution has helped us understand monogenic disease to a level we quite frankly couldn't dream of just a decade ago. And these insights have helped especially to improve productivity in discovery and in the pharmaceutical industry. We've seen a massive gain in productivity over the past 10, 15 years. But we also have to be honest, because that gain in productivity have predominantly benefited relatively small diseases. And I think we have to think deeper about how we can use that insight to innovate also when it comes to bring new medicines for people with cardiovascular disease, for people with diabetes, for people who have obesity as their primary medical concern. And the thing is that the genome-wide association studies we've seen come trickling in over the past 2 decades have probably confused us more than they've actually brought clarity. But I think now with the advent of especially new ways of mining data and coming out with sort of artificial intelligence to really bring order into all the swarms of data we have access to, the time is now ripe to really understand diseases at a much deeper level of understanding. And why do I say this? I say it because we have in general medicine this tendency to say people suffer from something that is common, and maybe the symptomatology is common. There's a high blood pressure here, there's an elevated glucose here, there's a decrease in cardiac output here, but that's a very superficial way of looking at disease. We need to go layers deeper and understand there are many different reasons why patients show up with an elevated glucose or a higher blood pressure. And first, when we start understanding the different molecular crossroads that leads to these manifestations, we also have a chance to introduce new medicines that are far more precise in their approach to the patient. And why is that needed? It is needed because the most recent introductions of medicines, especially in the area of cardiovascular medicine, have not really moved the needle when we are talking about how it affects the population with the disease. The PCSK9 inhibitors that were touted as, God-given medicines for people with elevated LDL cholesterol, they're quite frankly not very efficacious in a population setting. It's a medicine that has about 60, the numbers needed to treat is about 60 people need to be on the medication to prevent one cardiac attack. And that's obviously, from a healthcare perspective, a very wasteful way of coming up with innovation. So we have to bring that numbers needed to treat down so the efficiency and the cost effectiveness, again, is attractive for the healthcare systems around the world. And I think that's where, pharma, clinical medicine, and biotech can come together and improve our ability to innovate. So we also introduce precision medicine in the area of general medicine.
Thanks, Philip. I think you already raised a very provocative challenge of moving the work from rare disease to common variants, and the idea that some of these phenotypes are terminal endpoints, really, of the myriad different causative upstream mechanisms that then can be perhaps targeted in a more stratified fashion. I'll say a few words about myself. My name is Patrick. I'm from Singapore. Singapore is a very small place. It takes half an hour to get from one part of the country to the other point, and that's the long drive. So I am here for two capacities. My first capacity is that I head a research institute in Singapore called the Genome Institute of Singapore. It's about 400 people working on all aspects of omics, DNA, population sequencing, single cell, epigenetics, sequencing not just humans, but flora and fauna, microbiomes, and this is really a passion of mine. The other area that I have responsibility for is running the Singapore's program in national precision medicine that I'll say a few words about, because it's really our attempt to try to think about how can precision medicine be brought to scale in a way that ultimately benefits the country. So in the Singapore has a very unique composition of three different groups of individuals. We have Chinese, we have Indians, and we have Malays that represent Southeast Asia. I hope that some of you have had an opportunity to come there. So it's really one of the only countries in the world where you have these three different ancestries coexisting together in one space. The life expectancy of Singapore is very high. We have a pretty good healthcare system. We're about 85 years old, but we have a lot of challenges in our healthcare system. Many of our people, even though they live long, they will spend the last 7 to 8 years of their life in poor health. They will encounter the hospitals, and this is driving up a lot of healthcare costs. So really the question for Singapore government was, can we use precision medicine or precision health as a way to trigger changes in population health, to make the population more healthy, so that people can live longer, but not just live longer better, be more productive. So we did it in 3 phases. In the first phase, we tackled the genome. We did whole genome sequencing of 10,000 individuals. 10,000 is enough to capture what we call the normal variation in the population. It tells you what is different between individuals, but probably not relevant to pathogenesis. You need that for any reference genome, and because the reality is that compared to studies from the Western population, many of the other populations in the Middle East, in Asia, are underrepresented. So that was a start, and we learned how to sequence at scale. We're currently in phase 2, where we will be establishing a long-term population cohort. So this, if you want to understand factors that predict disease, you have to start off with a healthy population and work onwards. So we have a population called SG100K. It's 100,000 individuals. We're doing whole genome sequencing, whole genome methylation sequencing to look at epigenetics. We are doing research phenotyping. So every individual goes through a 6-hour phenotyping exercise where we image, we take questionnaires, and we follow them as they progress, and we link them to national electronic health records, so that anytime any of these individuals sees a doctor or sees a clinic, we get a readout. We know what's the event, we know what's the blood test, we know the prescription, and that allows this to identify the factors that are important and to suggest interventions that then can be brought to bear. And once we know those, we then go to phase 3, where we target 1 million individuals, or 1/6 of the population, where we will bring it on the cusp of clinical implementation to roll out to the entire country. It's a 10-year program. For people who are interested in it, a recent perspective of this was published in Nature Genetics last week. And so we hope that some of the experiences on this could be brought to bear for other population genomics programs. And so maybe with that introduction and to my illustrious panel, we'll open up for questions. So I'll start off with Dr. Majid. As you know, as most of you, he came here from the hospital, and maybe you can set the stage for rare diseases in the kingdom. What is the prevalence? What are the conditions? Are there particular genetic diseases that are more common in the kingdom compared to the West? And how do you think, as a biotechnology enterprise, one can the nation can capitalize on this, not just to improve the health, but also the economic prospects for biotechnology? Maybe I'll ask it to you. I'm sure that other people have questions, and we hope for questions from the floor also. Over to you, Majid.
Yeah, thank you, Patrick. Actually, according to our studies in 2016 and 2017, we find the prevalence of rare diseases in Saudi population is around in one study 1 in 600, and in another study 1 in 1,000. But with more, this study has its own limitation, but with more and more data and from our observation in the newborn screening program, that number, which is 1 in 1,000, is become more and more accurate. What does this mean? This means, if you translate it in life, that 1 in each 15 Saudi, he is a carrier for a pathogenic mutation for one of the rare genetic disorders. And this is really, if marry from someone who is carry the same mutation, then you will have a rare genetic disorder. So the autosomal recessive disorder is really prevalent in our population, and if you compare it to US or Europe, it's 10 times Europe and it's 10 times US, United States, and 4 times Europe. Just name any disease and you will find this prevalence. And we did that exercise for many of the diseases. And this indicates what? That the rare diseases in our region is common. So it's not anymore rare. So it's a great place for clinical trials, for any new medication to be tried and run a clinical trial. This region is the most suitable region to run randomized clinical trial for any new medication. And this summit, I think it's a great opportunity to try to emphasize on that and building bridges with the industry in order to run more clinical trials in this region in rare diseases. And the other thing we found also in our region, which is really exciting and sad at the same time, that you can find more than one disease in one family, and also more than one disease in one patient, not only family. So, and when we measure that, we found it around 10% of our patients, they have more than one disease. And what is this meaning? This meaning that the usual approach, which is done in Europe and USA, is not anymore here, is applicable. So the first, I think in our population, the first line of testing is whole exome sequencing and whole genome sequencing, rather going to the single gene and gene panel like other population, because you find that this couple may have another disease, and to prevent the recurrence of the disease in the future pregnancy, you need to discover all the diseases in the family. And when we did our study recently in 2020, we find that 8% of Saudi population have secondary incidental finding. And this is really indicate how this disorder is prevalent in our region, and how our need to have more strategical approaches to fight this devastating disorder and prevention them, and also find a treatment for them.
Thanks very much. And it's quite interesting that from your explanation, many of the genetic diseases are not in the West where they are arising de novo, but actually inherited through the families. And looks like in Asia, we also have things like beta thalassemia, which is not as prevalent in the Western. And so, what do you think about applications like carrier screening, for instance, before couples get married? Do you think that is an application to try to find the carriers and provide counseling? Or is no? Would you? What are the types of attitudes that one has to think about?
Yeah, I think certainly this is a good question. I think we need really to expand our panel regarding the premarital screening for these disorders. But also, in order to do that, also you need to consider the ethical issues and also the social issues, because you want also people to marry, you don't want to prevent marriage. So you really need to do this carefully and also with a great strategical plan. But also the other things I think that make this region is also excited and sad at the same time for the rare genetic disorder, that our population here is homogeneous. So and if you see if we see like in the last decade, we sequence more than 5,000 exome and around 2,000 genome, and our data showed around 90% of the cases are homozygous, not like Europe and US. So in US and Europe you will find compound heterozygous and autosomal recessive, but here in our country we found homozygous, and this indicates homogeneous population. And homogeneous population also, it's good for clinical trials because it minimizes the the biases regarding the sample itself. So, and also it's a good model for discovering the medication, even for the complex genetic disorder. So always I am saying that the rare disorder are the key for the complex genetic disorder, because there is a lot of pathway connected, and at the end we are one human.
Thanks a lot. Maybe we can also have the flip side of this on the biotech sector. Maybe I can ask Philip and Jim. You know, there have been some biotech companies coming up that are focused on genetic disease, rare genetic, Shire, for instance. You heard about Moderna. From the point of view of a drug development standpoint, is this something that is viewed as mainstream, or is this something that is viewed as kind of like its own a niche area? Because the numbers of patients, and I guess we'd have to talk about the cost of these therapies. And Novartis has the $1 million a patient for the SMA. So can I have any thoughts there on how industry looks at, I guess, rare genetic disorders, even though they're not rare?
Sure. Well, my view is that the prevalence of rare disorders and coupled with the genetics and the insights, the tools that have evolved over the past decade or so has really helped improving the productivity of the pharmaceutical industry as such. And I think it would be very difficult to provide or come forward with a value proposition of that nature if no one's willing to pay for the innovation. So obviously there is a will, there is a path, but I think the bigger question is whether this is a sustainable path, because I don't know how many different rare disorders there are, but if we say there's something, between 7,000 and 10,000 at the end of the day, I think we also need to come out with very different business models, different payer models, because at the end of the day, we could end up simply exhausting the means that are available for healthcare if we're exclusively focusing on these quite rare disorders. And we collectively, that's not only decision makers in insurance and public health, but also in the industry, needs to have a better understanding of what, future opportunities will be rewarded by also access for the medicines. And it's not an easy discussion because at the end of the day, it's how we prioritize healthcare.
Thanks a lot.
If I could add,
yes,
it's a great point, Philip, and to build on that, I sort of think of it as risk management along those lines. What's the incentive to get involved? So, is it mainstream? I think it depends on the company and the strategy and the pipeline and the risk. So, what we did at AstraZeneca is we acquired a company that were already mature in this space, and so that made sense from a risk perspective because a lot of the technologies and the disease understanding was already done. And so, that's now become an entirely new R&D unit for us. But, to your point, Philip, I think, we need to see how this is going to play out over time, how this is incentivized around the world, how the innovations come forward. But I think perhaps what's in our favor is many rare diseases do have a clear genetic cause. Many of those clear genetic causes are clear genetic targets for therapeutics and medicines. And so, that's where I can do a short advertisement for our collaboration, I guess. So the clinical trials that Dr. Majid mentioned, the kind of therapeutics we want to test there are things like CRISPR gene editing for, improving the condition or even completely curing some of these rare diseases in the patients that carry them. So I think that's, to move from the broad global perspective back locally into the region, I think technology transfer and knowledge transfer to bring some of those technologies here will be of great assistance in the future in terms of really addressing some of those rare diseases. And I also look forward to seeing us run those clinical trials.
Thanks very much. I think, you actually led to a question that I can ask Dr. Hegde. And, the genomics used to be about reading and what nature has given us and reading a lot of it. But now we have the ability now not just to do that, but to edit and to write. And, maybe I can ask Dr. Hegde, how do you, and maybe how do you see this, a future and the ethical issues with actually going in and editing? So you can do it on the somatic genome, but now sooner or later we will talk about editing the germline genome. Give us a sense of the crystal ball on where you think the future is in this whole area of gene editing therapeutics.
Thank you. I think that's a great question. It's a very loaded question. So I'll try and divide the answer into two parts. I'm going to start with talking a little bit about newborn screening, because newborn screening is really an ecosystem which we have all come to understand over the past 50 years. And there are about 70 babies that are saved globally today by just some nutritional supplementation for many of the rare diseases. If you use the same concept, because newborn screening has taught us a lot on the ethical implications of doing, every country approaches newborn screening very differently. And in PerkinElmer, we often talk about in-country, for-country approach using the genomic approach. And what does that mean? Because each country has a different approach to how genomics is implemented. The cultural aspects of that country have to be also taken into account. And when I just look at some of the disorders today that big pharma and small pharma are addressing, it's pretty phenomenal. In our labs globally, we also participate in clinical trials. And we are actually right now in about 20 clinical trials with large and small pharma. And the way we address this, looking at something like a CRISPR editing approach, and if we just take one example, I think it's easiest to talk about this CRISPR editing or gene therapies using some example. And I'm going to use the example of Duchenne muscular dystrophy. Globally also you can call it one of the common genetic disorders because there's 1 in 5,000 boys who are going to be affected. And there are a variety of different approaches today that are being used to find a therapeutic answer for DMD. And the way we look at it is the CRISPR editing is one of the sort of very exciting approaches that has been used. But you have to go back to the genome for that. It is not about just editing the DMD genome to correct the defect, but also finding out all the other impact that can happen through CRISPR editing. So we are doing genome sequencing to see off-target hits, actually making sure that the CRISPR target is hitting the right target in the DMD gene. So there's a variety of ways of doing this. I think the ethical aspects are really important here, and we see this on a day-to-day basis globally because we get approached by families globally who want to enroll in the clinical trials. But the questions each of these families ask us are very different from what we are sort of projecting ourselves as in terms of enrollment. So there's a lot to think about here, but the aspect that sort of I would leave all of us to think about is how do we address it in country for country, use artificial intelligence to layer upon that approach and keep the understandings and the education we have all gained from newborn screening, because it is done globally. It is one of the great examples of how to deploy rare disease approaches globally.
Thanks very much. And maybe I can extend that to our industry colleagues. Comment on the use of these technologies to intervene on targets that previously were not tractable by other therapeutic modalities, small molecules. And is that something that you think is being opened up, targeting the undruggable proteome? Or, can you have comments on that?
Yeah, I'd be happy to give a shot because we think about small molecules as a relatively traditional sort of modality or entity, and actually there's been a lot of development in the area of small molecule chemistry over the past couple of decades. So, today's small molecule chemistry, that covers chemoproteomics, small molecules targeting RNA, small molecules that, with the use of ubiquitins, facilitate protein degradation, stabilizes transcription factors to either promote gene expression or decrease gene expression. So small molecules, as of today, are, like other modalities, amenable for a lot of diverse applications. And, my view is that I think some of the fairly crude modalities we are currently using to either knock in or knock out genes will become available also through small molecule chemistry in the future. So we have far more tools in our toolbox to really address ways of manipulating gene expression, so on and so forth. So I really think, the traditional medicinal chemistry is evolving as we speak. And this is clearly an area where, artificial intelligence is helping enormously. With AlphaSight or DeepMind's, new way of looking at proteins, all of a sudden, all the 20,000 human proteins are essentially available in public domain. So everyone who wants to model and target any of our proteins in the body, also the mutated ones, can do that. And, the database is there. It's essentially just, our imagination that sets limits.
Yes, so I think this is an amazing, because with the new technologies, you can actually, technically, any targets can be used. I'll pass it over to my co-chair to continue the discussion.
I think also for the gene therapy presented, since 1970s. And the problem with the gene therapy since long time was the delivery inside the cell. So with discovery of a CRISPR, we also have improvement in learning how to deliver these, corrected genes inside the cell through viruses and also through different other non-virus vector. So there is advancement nowadays in the area of delivery of this molecule inside the cell. And once we are, I think, successful with this approach, maybe you will see like one approach could be applied for all diseases, you know. So it's an exciting coming decade in the era of medicine.
Thanks a lot. I'll pass it over to Dr. Majid to run the rest of the session today.
Okay. So I think we are good in time. We can open one question if anyone has any question now. Yes, in the back.
My name is Dr. Khouda Masoudi, PhD in medical genetics from University of Tabuk. Thank you for this great discussions. My question is, how do you deal with very rare, like variants? Sometimes a rare disease has like one common variant, and companies around the world tend to develop a gene therapy for that variant. But what about the other variants? I'll give example for retinitis pigmentosa. So the most common, like 30% of patients in the world has RHO, a mutation in RHO gene, and many efforts in different labs around the globe are developing against RHO. So, but we have like about 80 variants for this disease. How do you deal with this, like maybe one case in the world, like 5 cases in the world, the very rare variants? Thank you.
So if I understand your question, you are asking that how we deal that some diseases has one only mutation that common for the whole world, and the other diseases having many mutation. So how we can deal in developing medication for such a mutation? Is that your question?
No, actually, no. Retinitis pigmentosa has 80 genes. So any mutation in these genes can develop retinitis pigmentosa.
Yes.
But when you look at the statistical data, 30% of the patients with this particular disease has RHO gene, mutated RHO gene. So I can see many research groups around the world develop against RHO. Yeah, I mean, they just sounds like they don't want to waste money on like one patient in the world, so they find it it's better for them financially to develop for the most common variant. So RHO, they all focus on RHO. So what about the other variants?
Okay, excellent question. So
thank you, Doctor.
Yeah, so I think anyone want to answer this question?
It's a hard thing. Maybe Madhuri Dr. Hegde can answer that. Try to answer that.
Sure. So yes, it is a difficult question. I think let's just talk a little bit about the variants itself. So, we have a lot of genes, but most of the genes have variants in them which are difficult to interpret, especially when you find a single variant in a single patient. And as you know, all of us follow the American College of Medical Genetics and Genomics sequence interpretation guidelines. What happens in this situation is that unless the variant is actually a truncating variant, we interpret those variants as variants of unknown significance. So going to your question about why the companies are approaching a single pathogenic variant and not going over the entire spectrum of variants, this is a fundamental problem in how the therapeutic approaches are being developed, because it is really necessary to be certain about the impact of the variant on the gene and its gene product before you can have a therapeutic approach. Many of these pathogenic variants are private pathogenic variants, so it's very difficult. It's not just the cost of developing the approach, but understanding the impact of that particular therapy on that pathogenic variant, and therefore, the downstream impact of developing such an approach. But I think what is going to happen, today this is the scenario. In the next 5 years or so, as we understand more and more about what is the impact of these therapies on the gene and the protein, the approach can be then expanded to the different variants in the same gene. So it will happen. It is not happening today because we are still very early on in understanding these therapeutic approaches, especially CRISPR-Cas9. But I'm sure that as we understand more, these approaches will be spread throughout all the variants in the same gene.
Great, thanks.
Thank you.
And maybe to add to that, it really depends also on the genetic defect, because if it's a loss of function, gene replacement may not necessarily have to recognize the actual mutation leading to loss of function. You can still repair with kind of a universal construct. And one example is also you can do it even with small molecules. Vertex therapy for cystic fibrosis is obviously efficacious in many patients, irrespective of the nature of the mutation. So at least there are examples of a single drug actually helping patients with different mutations.
Yes. Also, we can have also other modality of a treatment other than gene therapy alone, focusing on the pathways or chaperone therapy or whatever available for that specific phenotype. Now we have the third question in our session. How is genomic and precision medicine contributing to better impact on health and the economy? For Dr. Patrick.
Okay, so I'll try to be brief because I do want to hear from Philip and Jim. So the question is, how can a national program in precision medicine help on health and the economy? When Singapore looks at these programs, we look at what is the legacy of that program after it ends. So we invest public money, but it has to have a long-lasting impact. And the way we think about these programs, we have 3 letters. We call it R, I, and E: research, innovation, and enterprise. I think for the research part, it's important to recognize that for many of these diseases, particularly those that are more prevalent in the non-Western countries, we actually have a very poor understanding of their pathophysiology, their basic mechanisms. And the omics and the precision medicine really is the only way to give you that molecular interface that can reveal new drug targets. So I think the research, if you have good data sets, good databases, good linked, this will power our research for many generations. For I, there's innovation. How can we use this to actually improve the health of the nation? And this is where it becomes tricky, because you need to choose the right clinical use cases that can scale, that ultimately when you engage the health economics people, they can look at this and think about how do you price health? What is the cost of one human life? And I think this is a conversation that we're having in terms of how, and those are the clinical pathways that we need to bring to bear in the innovation system. For enterprise, it is another area. Through these programs, you can extract public-private partnerships to bring new capabilities to your country. You can work with pharmaceutical companies to bring in skill sets and capabilities. And all of these things ultimately will catalyze your next generation of homegrown industries that can then learn from them, acquire new capabilities, and accept new intellectual property that is now embedded in your country. So that's the way we think about those 3 aspects. I think I'll stop there.
Okay. Any other comment?
Yeah, I was going to add a couple of points actually. Is this on now?
It's off though.
Yeah.
It's off though.
No.
Maybe while Jim is doing that, we can, because in the interest of time, we can. Oh, is it on? Test. Yeah, maybe Jim, you can see. Yeah.
Yeah, I think also we could remember that with the Human Genome Project, which was done in 2000, we can see currently the countries who invest in Human Genome Project in 2000, they are currently the leader of economy and genomic recently. So we think as a geneticist now that the countries who invest currently in the genomic data and precision medicine and big data and genome sequencing, for sure in the next decade they will be the leader from the economy part of view. Because currently now artificial intelligence is entering every field in our daily life. And the more data you collect as a country, the more, economic impact will happen in the future.
So hopefully I'm back now. Yes, I am. So I just wanted to build on that point about, if I sort of broaden the question slightly to how do you maximize value for precision medicine approaches across a healthcare system. I think one thing is really interesting is the sort of open source movement. So as an AI scientist, everybody sees the buzz around OpenAI, whether it's ChatGPT or DALL-E or the natural language processing. Great strides are being taken, I think, to open source and democratize AI, and I'm fully supportive of that. I think part of democratizing our technologies and making them more available and more accessible to researchers so that every research institute is not just reinventing the wheel, I think is incredibly important. So an example that we've done with AZ, for instance, if you look up DrugNome, NOME.ai, that's where we did a drugability assessment of the genome by annotating it with additional data, and that's essentially open sourced AI. You can just, there's a published paper and it points towards a website, and anybody can access that website and interrogate it and say, which portions of the genome could I target with different types of therapies? And that's something we've done because we believe science, within certain commercial limits, should be an open, collaborative process. The second point I was going to make, it wasn't in my notes, but it occurred to me as you were speaking, is you said something about how do we improve the sort of cost base in a healthcare system? How do we minimize or optimize costs? Well, AI is very good at optimization and achieving certain objectives. So an intriguing question for us all, I think, is if you gave AI all of the data from a healthcare system and set it the objective, how do I minimize costs?
That would be interesting.
I wonder what it would tell us. It may give us an answer that we didn't know. It may give us an answer we don't want to hear, but it would be absolutely fascinating to try that out.
It would be, yeah.
So Jim, can you tell us what is the main development of AI in genomic medicine currently exists?
Yeah, I think I've covered a lot of it in the previous questions actually, and we're almost out of time. Maybe I'll just add one more and then open up for the other panelists. So, I mean, AI in genomics more generally, I think for me, it's a lot about linkage of different data domains, and multi-omics has been mentioned a couple of times, so I think I'm partially repeating that, but making genomics somehow more dynamic so that we look not just at genetic origination of disease, but downstream transcription, translation. And then as we see these different layers of omics being built out, organizations are coming along to plug in the gaps. So I've worked with a small company that now specializes in epiproteomics, and they're starting to fill in all these different parts of biology and plug it together for a deeper understanding. It is incredibly complex. No human could understand it, but AI is great at finding patterns in that kind of stuff, so that then we can understand as scientists what we want to do with those patterns. So that's what I'd like to see, more dynamic and holistic understanding of biology.
So Philip, how precision medicine and AI will give better opportunity for the biotechnology to develop in the coming future?
Well, I think when we're talking about precision medicine outside oncology and rare diseases, the challenge has always been, as I was alluding to a little earlier, to find the patient who would respond to a given intervention. And that's also about, improving cost efficiency, but it's certainly also about improving the benefit-risk balance for the patient and for the system. And this is where I think AI can really help, building on what Jim just said, understand the complexity of data far better, pairing omics with genomics, but also understanding that the central dogma, so to speak, one gene, one protein is by no means correct any longer, because there are so many smaller proteins, so many different things that are happening at the level of the genome that, we have to understand that far better. But this is where AI can help us at least understand patterns and connections, not necessarily giving us the finest layer of detail, but at least giving us an opportunity to tell which patients would respond well to a certain intervention and who will not. Because we may already have drugs and tools available that can be used in a far more efficient way, and that in and of itself would be a massive gain to maybe, be gained over the next 5-10 years.
Thanks. I think we're out of time already, but I wanted to thank, it's been a great conference. I'll highlight that a lot of this stuff we're doing is only on 1% of the genome. It's all the protein coding genes, and I hope that there's so much more to do. 9% of the genome, we don't know what they do. So there's a lot of opportunity for new discoveries to be made in this space, and I think this is a reflection of the excitement that all of us feel. I think we'll draw today's session to a close. I want to thank all of my speakers and also Dr. Hegde for dialing in. Thank you very much for your time, everybody. Thank you.
Thank you.
Thank you, co-chairs and esteemed panelists, for your valuable contribution and insightful views. Ladies and gentlemen, thank you for being with us till this time. It's Thursday and the weekend already started. And for that, I just want to tell you thank you. You are a remarkably interesting crowd of people. To officially close our proceedings, it's my pleasure to welcome onto the stage His Excellency Dr. Bandar Al-Gnawi, CEO, Ministry of National Guard Health Affairs, President of King Saud bin Abdulaziz University for Health Sciences. The stage is yours, Your Excellency.
Thank you very much for being around all for the last 2 days. It's been a great pleasure to have all of you here contributing to the success of this summit. It's an opportunity that we have seen brought all of us together to discuss a very, very vital, important project for the country, which is biotechnology. And we're really privileged to have, major international biopharma companies and leaders and senior executives who are being able to join us and present the latest advances and the advances in the field of biotechnology, cutting-edge technology, and as well making sure that we network together for the future, network together to be able to enhance our ability, not only for Saudi Arabia, but for the global benefit of human health. And truly it's an honor and a privilege to work with all of you, and very excited to see that all of us are coming to the close of this program, and as well excited to see that yesterday we've been able to sign few of the agreements between the different parties, and to see as well there has been a lot of meetings at the summit exhibition and as well at private boardrooms to discuss as well the future and as well planning for advancing the whole objectives and goals of this particular gathering. Truly it's a marvelous event, and you made it very successful. You made it really as well become something that all of us will look forward for the next year. Without your support, without the support of our colleagues from everywhere around the world, we will not be able to reach to the stage. And as I said yesterday, we're looking forward to see that we can make it even better next time. We're looking forward to make sure that as well we follow on all of these issues that we have discussed and the panel discussions and all of the learned lessons and the shared experiences that we have seen over the last 24 hours. It's truly an honor as well at the end of this closing remark to invite my colleague Dr. Ahmed Al-Askar to really come forward and bring some of the recommendations that we think has been developed, co-developed with you to see that we are here as well looking to make sure that we don't leave without creating a roadmap for the future, creating a roadmap for the collaboration, integration, and as well ensuring that we are cooperating together for this particular biotechnology advances in Saudi Arabia and worldwide. Thank you.
Thank you, Your Excellency. Thank you for your support. And what a wonderful 2 days, great program, rich discussion that we learned a lot from, lots of inspiration through these panel discussions. Made by experts in the field, by leaders, by thought leaders in the field of biotechnology, innovation, research and development. We really value such great contribution. These discussions and your input was taken in consideration and has made us to develop what we will call Riyadh Declaration, and its recommendations basically now given the leadership in this country who are giving us their support and their engagement into this conference, into this summit. We ought to come up with recommendation, and these recommendations are coming from you, from our participants, from all those who discussed, contributed, and exchanged their expertise with us. So allow me to present to you an executive summary of this Riyadh Declaration with recommendation out of this summit. So, achieving a knowledge-based sustainable economy and investing in R&D are at the forefront of Saudi Arabia National Vision 2030. As such, the Saudi government recognizes the need for science, research, and innovation in achieving its Vision 2030 goals. In light of the Kingdom of Saudi Arabia current focus on economy diversification and job creation, local production, technology transfer, conducting clinical trials locally, and training and education of the Saudi labor force are becoming the new norm in the biotechnology and pharmaceutical products industry. To contribute to the Kingdom efforts in developing a thriving biotechnology ecosystem, King Abdullah International Medical Research Center at the Ministry of the National Guard Health Affairs has seized this exceptional opportunity in collaboration with the various national stakeholders and international partners, and organized in September 2021 the first Riyadh Global Medical Biotechnology Summit, which has witnessed an exceptional success. Building on that success, the second edition of Riyadh Global Medical Biotechnology Summit is being concluded as we speak now. The Riyadh Global Medical Biotechnology Summit, in its 2 editions, was organized under the auspices of His Royal Highness Crown Prince Mohammed bin Salman and inaugurated by His Royal Highness Prince Abdullah bin Bandar, Minister of the National Guard and Chairman of the National Biotechnology Strategy Committee. The summit aimed at presenting the current status and future opportunities for global partnership in key strategic areas of high priority to KSA in medical biotechnology, including vaccines development and manufacturing for infectious disease and cancer, cellular and gene therapy, therapeutics development and manufacturing, genomics and precision medicine for rare diseases, as well as clinical trials and real-world evidence. In alignment with the National Vision 2030 and industrial individual strategies, institutional individual strategies, Riyadh Global Medical Biotechnology Summit has identified the following recommendations as strategic cornerstones to transform KSA into a global gateway to thriving medical biotechnology, which includes, number one, a cross-functional medical biotechnology steering committee should be established to agree on an implementation roadmap that outlines detailed actions or detailed action plan and assigned roles and responsibilities, milestones, and expected deliverables. Number two, legislation to enable individual researchers to create biotechnology startup companies, including those affiliated with public and governmental institutions. Policy, that's number three, policy to encourage, support, and protect in-country R&D and manufacturing for biotechnology products based on the main therapeutics area to focus on. Number four, effective regulatory system that strives to ensure safety, efficacy, and quality of medicines and other medical biotechnology products, building on the existing Saudi FDA standards and aligned with these international standards. Number five, government policies which incentivize biotechnology entrepreneurs for accessing convenient startup funding and policies that will encourage growth of venture capital sector, ranging from financing facilities to equity participation models. Number six, support the creation of international partnership and alliances in medical biotechnology in order to attract new foreign direct investment. Number seven, develop incentives to attract and retain top talents, national and international, to establish and grow global collaboration and to foster public-private partnership in R&D, innovation, and application of biotechnology. And the last, the one before the last, number eight, earmark a dedicated startup fund to support the creation and expansion of medical biotechnology companies across the biotech ecosystem. Number nine, establish an international independent advisory group to work with the Biotechnology Steering Committee to develop clear KPIs and to assess and monitor the development and maturity of medical biotechnology ecosystem in KSA over the next 5 years as a result of the initial investment. So with that, that concludes this executive summary of our recommendation, and it's all, again, contribution from all the participants, distinguished speakers, biotech leaders across the world, and academia. So with that, I would like to, again, attract your attention to the next, which is a video highlight of the two-day summit. And then I would like to invite you after the video to a group photo. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh.
صاحب السمو الملكي الأمير عبد الله بن بندر بن عبد العزيز وزير الحرس الوطني، أصحاب المعالي، السيدات والسادة، حضورنا الكرام، نرحب بكم في هذا اليوم المبارك في حفل افتتاح قمة الرياض العالمية للتقنية الحيوية.
Biotechnology is going to be the highest growing sector in the next few decades.
Biotechnology is something that's growing rapidly. Today, Saudi Arabia is privileged to have a great vision, leadership, direction.
إنجازات كبيرة جدا في الأربع سنوات الماضية، نحقق أكثر إلى 2025 ومن ازدهار لازدهار إن شاء الله.
Thank you. May I invite Your Excellency and to the stage for a group photo, and everybody, the stage accommodates all of us. Please.